Everyone, how's everybody doing? If I am audible and visible, please let me know. And we shall begin with the NEET PG marathon. We're going to start with the most easy peasy, most beautiful subject, which is microbiology, right? So the end by the end of this marathon, what are we going to feel about microbiology? That it's easy and peasy. Okay, so stay tuned for a whole marathon of mnemonics and picture mnemonics and visual cues, and we are going to quickly rush through the entire the syllabus. So this is like the best way to revise entire huge chunks. So let's not waste any time. If you have joined, which I see a lot, many of you have, I would love to greet you. Do drop in a hi and hello in the chat box. If you have any doubts, drop in your questions. Even if you want to answer, please drop in your answers also. So let's keep it interactive so that I can find out what pace you want me to go at. Okay, so we're going to start with the most easiest thing. Everybody knows this. Hi, audible, visible. Thank you so much. Good morning. Good morning. So the marathon is going to be a week long. In a single day, we are going to cover 2 to 3, subjects. So today you have micro followed by derma and then peds. So that is how it's going to be. All right. So I mean, I am sure that this photo is something that every one of you can recognize. This is a Zn stain. This is a acid fast stain. So what are you seeing? You are seeing these acid fast bacilli, which are slender, thin, and beaded appearance. Why do these bacteria which give this acid fast appearance? Because they have something called as the mycolic acid in their cell wall. Of course, you know that this is something which is so the workbook can be found from the link which has been shared. It must have been shared with you. All right. Chalo. So, mycolic acid. Yeah. So, what was I saying? I was saying that there are some modifications of this Zn stain which is asked. What are the modifications? You know that it depends upon the percentage of H2SO4 which is used, right? So, there is not just Mycobacterium tuberculosis which is acid fast. So, 25% H2SO4, Mycobacterium tuberculosis is able to withhold. Okay. But what about the others? You know, you have Mycobacterium leprae also which is acid fast. Then you have nocardia also which is acid fast. Then you have the coccidian parasites also which are acid fast. Then you have the head of the sperm, spore of bacteria, okay, bacterial spores, sperm head. What is all the modifications of Zn stain used? Yes, correct, you're writing mycolic acid, 1% nocardia. So 25% is MTV, 5% is M. leprae, and 1% is nocardia. Yes. What about coccidian parasites? Coccidian parasites is 0.5 to 0.25%. And what is about the spore of bacteria and sperm head? 0.25%. Another important thing about coccidian parasites is that you use the cold method. What is the cold method called as? The Quinones method. The Quinones method of staining, right? So there's nothing special about it. One special thing about gram stain that you have to remember, that many students tend to remember, that Legionella, no, leprae is 5%. Yes, many students tend to remember that Legionella is acid fast. But there's a correction. It's not Legionella pneumophila which is acid fast. It is acid fast. What is acid fast? It is acid resistant, right? So which Legionella? Sabka daddy. Okay, you know, Mick daddy. Legionella Mick daddy, Sabka daddy is actually the one which is acid fast, not Legionella pneumophila. So just to jog your memories and to sum up everything right here, what is Legionella pneumophila? You know, we remember I teach the students ki you have guests at home, you turn on the AC for them, you give them cold water and you say lijiye lijiye. And what have you given them by saying lijiye lijiye? Lijiye lijiye, Legionella lijiye. So Legionella pneumophila is associated with AC vents and water coolers. It leads to outbreaks. It is a cause of atypical pneumonia, right? Infiltrative interstitial kind of pneumonia. That is not acid fast. This guy, Sabka daddy, Mick daddy, is acid resistant or acid fast, right? Now we have, yes, you're definitely going to get the annotated PDF later also. You're going to find the link in the description or in the first comment box, which is going to give you the link to the annotated PDF. Right. So now we have gram staining. Gram staining, everybody knows you have two types of bacteria in this world. इस दुनिया में दो तरह के लोग होते हैं सेनोरिटा. कैसे? ग्राम पॉजिटिव और ग्राम नेगेटिव. नेगेटिव वाले रेड होंगे, पॉजिटिव वाले ब्लू होंगे, right? And this is a type of a differential stain. Okay, this is a type of a differential stain because this is making me differentiate between a gram positive and a gram negative bacteria. A question is that what is a mordant that is used in gram staining? What is a mordant? Mordant is something which is which fixes your friendship. Okay, so it is the Fevikwik of your friendship. First Sunday of August we tie friendship bands. What is that? That is a mordant. Okay, you're signifying, you're solidifying your friendship. So the mordant here which is used is the iodine. What is iodine doing? Iodine is combining with crystal violet, which is the primary dye, and it is making big complexes with this crystal violet, which is not allowing the big complexes to get washed off. So when I add the decolorizer, which is acetone here, this acetone is not able to get the crystal violet out of the cell wall of a gram positive bacteria. Why? Because the gram positive bacteria, their cell wall has a thick and complex peptidoglycan layer. A very thick peptidoglycan layer. Yes, chipka diya. Yes, correct. है ना? So this thick peptidoglycan of the gram positive cell wall is the reason that acetone or decolorizer is not able to decolorize this, because this iodine has behaved as a mordant and kept it all inside. Okay, kept it all inside. So the question is, which are the bacteria which do not get gram stained? Okay, which do not get gram stained. You know, there's a saying we say that gram stain is the stain of the poor people. Even if you have the poorest of laboratories, this is the most basic stain. You know what is the cost of iodine? Iodine costs 1 rupee per ml or something. Like one bottle can last you a whole year. So basically, why am I telling you this useless story? So that you can remember that gram stain is something which is a cheap thing. Okay, now people जो बहुत ज़्यादा पढ़ लिख जाते हैं, people who are highly educated and have gotten higher foreign degrees, they don't care about cheap things, right? So what is a higher foreign degree that people go and get? MRCS. Yes, so MRCS people won't care about cheap things like gram stain. So what are these guys? Mycobacteria. Mycobacterium tuberculosis especially never takes up the gram stain, or if takes very weakly gram stain. Then you have the rickettsial species. Oh my god, you must have been getting trauma right now thinking about the rickettsial species. So many weird names. But don't worry, we have a beautiful picture mnemonic and the story of a railway station which is going to help you remember the rickettsial group of bacteria all around. Okay. Then you have this guy, chlamydia. You know chlamydia ka problem kya hai? What is the problem of chlamydia? For the longest time we thought that this guy is a virus because it is so small and it is so obligately intracellular and it does not leave the bacterial cell, that we used to think that it's a virus. End of the day, we realize that it's a bacteria. So obviously no viruses take gram stain, so doesn't this guy. Mycobacteria, rickettsia, chlamydia, and S for spirochetes. Are spiral bacteria which do not take up, they're so thin, they're so small that they cannot take up the gram stain. So they need to take the silver stain on top. Okay, so we are done. So what is this? This is antimicrobial susceptibility testing, and we are looking at the Kirby-Bauer disc diffusion test. What are we seeing? We are seeing a cation adjusted Muller-Hinton agar plate, and we are looking at discs. So this is a disc diffusion test. What do we measure? We measure this zone, this diameter. So what do we measure? We measure the zone or diameter of zone of inhibition. Zone of inhibition is what we measure. Okay, and when we have all of these different antibiotics, you can see this is cefixime, this is nitrofurantoin, this is cefopodaxime, this is chloramphenicol, this is ampicillin. All of these different antibiotics you have put on a single, let's say this is E. coli, and this zone of inhibition you're getting. So you know that the bacteria is scared of the antibiotic. It is staying far away from the antibiotic. It is scared. What about here? Bacteria is not scared. It is growing right up to the mouth of the antibiotic. And how do be, it is not possible to remember the zone of inhibition for every drug and bug combination, है ना? हर E. coli और हर एंटीबायोटिक का कैसे याद करेंगे? इट इज नॉट ह्यूमनली पॉसिबल फॉर अस. तो हम क्या करते हैं? वी रेफर टू CLSI चार्ट. सो CLSI is the institutional body which gives these charts every year, which we have to refer to and do. The another modification of this disk diffusion test is called as Stokes method. Stokes method. So what are we doing in Stokes method? We are simply testing the antibiotic of the patient against a known strain of a bacteria, right? So over here we can only test one or two antibiotics, and we basically compare the zone of inhibition with a control strain. Control का क्या मतलब होता है? जिसपे हमारा control है, the thing we can control, we know how this is going to behave. So if I take this single disc of nitrofurantoin and I expose it to the patient's E. coli, and then I expose it to the E. coli, which is my known strain, I know how my known control is going to, how much zone it is going to give. So I will just simply compare the zone given by my control with the zone given by the patient's strain. So that is the meaning of control comparison. We are not measuring any diameters with a scale here. Okay, so this is just a single line thing that you had to understand. Then you have something which is called as microbroth dilution. What is microbroth dilution? You have taken test tubes, and in these test tubes you have taken a fixed amount of bacteria. What is the how is the presence of bacteria shown? The presence of bacteria is shown by turbidity, है ना? So turbidity है, that means bacteria is present. The moment turbidity gets lost, you know that the bacteria has vanished. There is no turbidity inside this anymore. So that means that the bacteria has been inhibited or killed. So what have I done? I have taken a fixed amount of bacteria in all of these test tubes, and I am going to serially increase the concentration of antibiotic. Instead of saying that we are increasing the concentration of antibiotic in this direction, can I also say that I am diluting the antibiotic in this direction? Yes, this particular test tube has maximum antibiotic, 64. This one has just the double, the half of it, 32, then half of it, 16, half 8, half 4, half 2, half 1. So what is this called as? This is called as doubling dilution. Okay, so actually we do doubling dilution, but how do we read it? We actually read it in this direction, from right to left. Why? Because we want to see that what is that concentration of antibiotic which is able to kill that bacterium. Okay, so if I give you one tight slap, you might not cry. If I give you two tight slaps, you might not cry. What is that number of slaps which makes you cry? That is what we are trying to see. Okay, so what is that maximum dilution that you have achieved, which was the minimum amount of antibiotic which was required to kill this bacteria? So you see that 1 microgram was not enough to kill the bacteria, 2 microgram was not enough to kill the bacteria, 4 was not enough to kill the bacteria. Finally, at 8 microgram, the bacteria gets killed. I have a clear broth. What is this clear broth called as? This is called as MIC. So 8 microgram per ml is the MIC of this bacterium. So what is MIC? Minimum inhibitory concentration. Minimum inhibitory concentration. The minimum concentration of antibiotic required to inhibit the visible growth of my bacteria. Visible growth meaning turbidity is lost. What does that imply? That implies that when I have to test bacteriostatic antibiotics, I can use MIC. Bacteriostatic antibiotics, I can use MIC. But if I want to know about the bactericidal nature of an antibiotic, I want to know whether it is going to absolutely kill or not. How do I make sure? See this test tube which had lost turbidity and it has become clear, and I was thinking all the bacteria is killed. I took some broth from this and I put it onto an agar plate, and to my surprise I saw so many colonies growing. So I went to the next clear test tube. From that I put onto an agar plate. I saw still so many bacteria. Then I went to the next clear test tube, and now finally I don't have any bacteria growing on the solid agar plate. So what is this telling me? That now bacteria has been absolutely killed. So this here, which test tube is this? This is test tube number 32. So 32 micrograms per ml is the MBC, is the microbicidal concentration or minimum bactericidal concentration. So this is measured by the MBC, right? So don't you think that this is a very beautiful method? You can find out MIC, you can find out MBC, you can find out very, very specifically, because when you try to measure the zones of these guys and you put a scale in front of it, to me it might look like 32 centimeters, but to you it might look like 31 centimeters. So there is a lot of problem with disc diffusion. That problem gets taken care of in broth dilution, but broth dilution is cumbersome. Who's going to put so many test tubes and do so much of drama? So you have the best of the both worlds. What is that best of both worlds? It is the E-test. What is E-test doing? It is giving me the convenience of a disc diffusion, but what is it measuring? It is measuring the MIC. So MIC is measured using diffusion method. So what have I done? I have not taken a disc here. I have taken a filter paper strip. What advantage is it giving me? See, these discs, they could only load a particular amount of antibiotic in them. This is a very small disc. If I pour 6 microgram of antibiotic in this, it is only going to contain 6 microgram. If I pour 3 microgram, it is only going to contain 3 microgram. So I cannot see the gradient, that dilutions that I was creating, that increasing concentration of antibiotic that I was creating, that was not getting fulfilled with the disc diffusion. So what did I do? I took a filter paper strip on which I graded the antibiotic in increasing or decreasing concentration. So you see over here there is the maximum concentration of antibiotic. Here it is little reduce. So you see that when the antibiotic concentration was very high, the bacteria stayed very far away. As the antibiotic concentration kept on reducing, the bacteria started to come closer. And it was only at this point that after this, the bacteria was not scared anymore and it was growing right up to the face of the strip. So, yahan tak it was very much scared of the antibiotic. So, this is the minimum concentration of the antibiotic which was able to keep the bacteria away. So, the neck of this epsilon which is formed is the MIC. The neck of this epsilon gives you the MIC. So, what are we measuring? We are measuring MIC. But how are we measuring? We are measuring using a diffusion method only. We don't need no test tubes. We don't need no broth dilution. Yes, like I said, जहां पे आपका epsilon start होता है, where the epsilon starts to appear, that pinching point is what is the MIC. This pinching point is what is the MIC. Okay, I hope your question was answered, Dr. Parth. All right, chalo, let's not waste much time on this. Okay, let's now talk about microscopes. You obviously know what is this microscope. Any line diagram you get, if you find an obstruction below the condenser, so it is in fact a very useless question which has been asked previously, that what is the position of this obstruction, which is nothing but an annular ring. So where is the annular ring placed in a dark ground microscope? It is below the condenser. Okay, it is below the condenser. Actually, what am I trying to achieve with this dark ground microscope? I'm trying to achieve a hollow cone of light. What does that mean? Have you ever seen a dancer performing on stage? On the stage, there is a spotlight, okay, and there is also many other things on that stage. There is the background dancers, there is the props, there is the background team, everything. You don't end up seeing everybody. Why? Because the hollow cone of light is only shining on your dancer. Rest everything on that stage is dark. So you don't see anything except your dancer. And who are these bacteria which dance under our microscopes? These are the spirochetes. They are doing the lashing extension, the flexion, the corkscrew. So they are doing cartwheels. So these are the perfect dancers that we have to see. So for your spirochetes, you have to see under dark ground microscopy under a hollow cone of light. And how is this hollow cone achieved? By placing an annular ring just below the condenser, just above the light source, or it can be said just above the light source. ठीक है ना? तो light का source आ रहा था, we created an obstruction. Now only a hollow cone of light is coming. So only a hollow cone of light is falling onto your specimen. What do you see? How are we able to see? We are able to see because the light reflects from an object and enters our eye, है ना? If there is a very brightly lit room and there is a white box kept in that room, you cannot see it. Okay, sometimes why are you not able to see glass? Because the light passes through and through from it. It is not reflected from any object. So that is why when light reflects from an object, you only see that object. So under this dark ground microscope, just like the stage wala phenomenon of a hollow cone of light, you can only see the dancing microbe over here on this slide. Okay, so what is it used for? It is used for the spirochetes, for looking at the motility. Because what is the advantage of this? The advantage is that I don't have to use any stains. So no staining is required. I'm simply playing with the dynamics of the light and I'm able to create this beautiful contrast. I don't need to pour any stains or any toxic chemicals, because whenever I put a stain, they are dyes, they kill my bacteria, and a killed bacteria will not dance. Okay, so when I want to see the dance of the motility, I don't want to stain. So for that, I use the dark ground microscope. Another thing that I will tell you right here, that annular ring I talked about. If the question mentions, or if the diagram shows you two components, one component is that this particular microscope has an annular ring at the bottom. Okay, cool. Second component is that it has a phase plate. Phase plate. And where is the phase plate? Before the eyepiece, before the ocular lens or the eyepiece. ठीक है, तो ये क्या बन गया आपका? Whenever you see these two words, you know what is phase plate? It is changing or shifting the phase of the light, and a shift in the phase of the light is going to give you a shift in the intensity of light. So that is going to give you a contrast. What is that contrast microscope called as? Phase contrast microscopy. Phase contrast microscopy. ठीक है, फेज कॉन्ट्रास्ट माइक्रोस्कोपी में दो कंपोनेंट्स मिलेंगे, एनुलर रिंग एंड फेज रिंग. गिव मी अ क्वेश्चन, आंसर. व्हिच माइक्रोस्कोप प्रोड्यूसेस द बेस्ट कॉन्ट्रास्ट? Which microscope produces the best contrast? Tell me, whatever you want to tell me, write in the chat box. So guys, it is not electron microscope. Many students like to think that best contrast is produced by the electron microscope. No. Best contrast is given by the phase contrast microscope. Okay, by the phase contrast microscope. This also has that advantage of keeping the bacteria alive. No staining is required over here. No staining is required over here. In the electron microscopy, you get the worst quality images of your life. You know that. They are the worst quality images. So electron microscope is not the answer. It is phase contrast followed by your fluorescent microscopy. But the problem with fluorescent microscopy is also the same. You have to put a fluorescent dye. All right. Chalo. So now let's look at this fluorescent microscope. Every time you see excitation filter, emission filter, dichroic mirror, you have yourself a fluorescent microscope. Fluorescent microscope. What is the problem here? Problem is that they have asked a rather pathetic question in the past, where they erased all of these labelings and they said, what is this? Right? What is this? And then they had written, is it excitation filter? Is it emission filter? Another question they asked, what is this? This was fairly simple. This is a mirror. This is a slanting mirror which is behaving in a in a biased manner. It behaves in a biased manner. It allows one particular wavelength of light to reflect through it, but it allows one particular wavelength of light to pass right through it. Okay, so इसको तो इसने reflect करके नीचे भेज दिया, पर इस green वाले को it allowed to pass up. So what is it behaving? How is it behaving in a contradictory manner? Your dichroic mirror. Dichroic का ही मतलब है it is going to behave differently with two different colors. Dichroic, it allows dash wavelength to pass and it allows dash wavelength to reflect. So you see the UV spectrum or the less wavelength is allowed to reflect, but a higher wavelength is allowed to pass through. So higher wavelength passes through, whereas lower wavelength is allowed to reflect. Okay, and how to never confuse excitation filter and emission filter? See, what is the light source? It is the white light. Okay, white light eye, it broke into Vibgyor and now it was excited to reach the UV spectrum. So the first filter is excitation. So whenever you start reading, you are very, very excited, right? And by the time you are emitting out fumes, by the end of the marathon, you are emitting out fumes. So similarly, in this fluorescent microscope, initially it is very excited. So just find the source of light. Even if this exact image is not given, what if they move the light source over here and they completely mirror image, change the orientation of this image? Don't get confused. Every time in a microscope, the story starts from the source of light. The story starts from the source of light only, right? So here also the story will start from the source of light. No matter if the source of light is placed at the bottom of the page, left side of the page, right side of the page, or top of the page, does not matter. Just see whichever filter is coming at the beginning of the story is closer to the source of light, that is your excitation filter. And by the end of all of this drama which has happened, there is emission fumes, matlab there is emission filter which is coming out, which is only allowing certain emitted light to reach your eye or your detector. ठीक है ना, इतना ज़्यादा कंफ्यूज होने की जरूरत ही नहीं है. Now we have this guy, everybody recognizes this guy. This is a electron microscope जिसमें ये आपके viewing ports हैं. This is a screen on which you can view. From here is coming what? Here is coming an electron beam, and of course electron beam is going to travel in vacuum, because electrons, if they are made to travel in air, they are going to takraw with the particles of the air and they are going to disperse everywhere. So in order to give the electrons one particular direction, you have to create vacuum. And of course these glass ground lenses cannot condense your electrons onto a particular specimen. So you need to use electromagnetic lenses. ठीक है. So there are two types of electron microscopes. You have the SEM and the TEM. You have the SEM and the TEM. What is SEM? Scanning electron microscope. So it is going to scan the surface. So these electron beams, if this is my hand, this is a virus, electron beams are going to tuck, fall onto the surface, and like this tuck, they are going to create a 3D surface image. 3D surface image. Whereas your TEM transmission is going to pass through and through your specimen. When it passes through and through your specimen, it shows me beautiful internal structures, but not beautiful, in fact very crazy, very bad looking images, but 2D internal structures. 2D internal structures is what you're going to see on TEM. Now quickly tell me some important fluorescent dyes which are used. Oh, because the use of visualization port is because if you have ever used a microscope, you will realize that it is very difficult to control the stage while looking over there. Okay, so this is about the hand-eye coordination. When you have a visualizing port, you can easily coordinate the stage with your hands. So that is only required for the hand-eye coordination, because just imagine in an electron microscope, how much magnification are you creating? You're seeing something which is in nanometers. You're creating 10 to the power 9 magnification. So when you have a camera lens and you're really zoomed in, even if your hand shakes a little bit, even if you move the image by just a few millimeters, there's a huge shift in the visualization field. So electron microscope is a very zoomed in view. So the controlling the stage needs very fine motor skills. That is very difficult while looking over here on a screen. So that is why you have that visualization port. A rather interesting question, Dr. Parth, but yeah, that shows a curious mind. Yes, excellent, you're writing all the dyes. Auramine, rhodamine. Now you also tell me the organisms then. Auramine, rhodamine is a dye used for whom? Then you have acridine orange. You tell me, dye used for whom? Calcofluor white. Calcofluor white. Tell me guys, auramine, rhodamine is for Mycobacterium tuberculosis. Acridine orange is for malaria. And calcofluor white, fluorescent calco. Calco is supposed to stay in the chitin layer. Yes, we can, Dr. Path. We very much can. Otherwise, it's a whole useless, lost exercise. Calco means, yes, calco means chitin. So, calco is what is telling you that it stains the chitin layer of fungus. So, of course, this is what we use for fungi. Fungi to visualize the yeast cells, to visualize the hyphae, etc. Chalo ji, next. So, here we are on a gram-negative and positive cell wall. What are all the things about gram-negative positive cell wall? You know that gram-positive cell wall is quite simple, quite easy. You have a cell membrane, right? Then you have a cell wall. That's it. End of story. Cell wall is made up of a very, very thick peptidoglycan layer, nothing else. Then from the top, what is all this coming out? This is the teichoic acid or the lipoteichoic acid. What is the feature of teichoic acid and lipoteichoic acid? It helps in adhesion. A rather crazy question was asked wherein they said that which of the following structures are important for attachment or adhesion of streptococcus pyogenes to the throat in the sore throat. Okay, and everybody was looking for M protein because everybody knows strep pyogenes is equal to M protein. What ended up happening? They wanted to ask lipoteichoic acid. So what particular what is the only concept that was required by you at that point of time? That gram positive bacteria actually has lipoteichoic acid, all gram positive bacteria. So it did not matter whether they had written strep pneumoniae, strep pyogenes, staph aureus, or whatever in the question. The lipoteichoic acid is present in all gram positive bacteria, and which helps in adhesion to the surfaces. So even if they have not mentioned any special or specific protein, lipoteichoic acid was the answer. Many students got confused because they वो रट के गए हुए थे कि स्टेप बायोजीन्स मतलब कि एम प्रोटीन, स्टेप बायोजीन्स मतलब कि एम प्रोटीन. ऑप्शन में एम प्रोटीन था ही नहीं. ठीक है, व्हाट वाज गिवन इन द ऑप्शन? Lipoteichoic acid. No problem, ma'am, because lipoteichoic acid is present in all gram positive cell wall. Then you have gram negative. Gram negative are negative people. Negative people are a little complex. They have many layers to them. You don't know how and when they're going to behave, how and when they will take something on their heart and not, so what are they planning and plotting. So gram negative people have a lot of layers. So you have a cell membrane, ठीक है, cell membrane, and you have a very thin peptidoglycan. This peptidoglycan is 100 times smaller than the gram positive peptidoglycan, right? 100 times smaller. Another important thing, the cell membrane is closer to the nucleus, so I can call it the inner membrane. You see, the exact copy paste, the exact replica of this membrane is placed on the top also. So this cell membrane is copy and pasted over here. What is this copy pasted upper membrane? Can I now call it outer membrane? Yes, I can call it outer membrane. So which bacteria has outer membrane, gram positive or gram negative? Only gram negative. Which bacteria has a thick peptidoglycan? Only gram positive. Which have lipoteichoic acid? Only gram positive. Which have a thin peptidoglycan? Only gram negative. Now if this peptidoglycan is fairly thin, you see there is a lot of empty space over here and over here. What is that called as? That is called as periplasmic space. Which has a huge periplasmic space? It is the gram negative bacterium. And what is this periplasmic space going to contain? This is going to contain certain important enzymes such as your beta lactamases. Okay, and then on top of this outer membrane you have certain proteins. Can I call them as outer membrane proteins? When these outer membrane proteins, they come together, they create a channel. What is that channel called as? That is called as a porin channel. On top of this porin channel you have a lipopolysaccharide layer. You know, this lipopolysaccharide layer is made up of 3 layers. So what is the mnemonic to remember the 3 layers of lipopolysaccharide? So lipopolysaccharide can be written as LPS. So what are the 3 layers of the lipopolysaccharide layer? It is also LPS. ठीक है, LPS, where L is the innermost. What is that? Lipid A. Lipid A is the innermost, right? Then you have core polysaccharide, and then you have the side chain. You know what is the side chain? This is O specific side chain. What is this lipid A? This lipid A is nothing but the endotoxin. You know, there are 2 types of toxins produced by bacteria, the exotoxin and the endotoxin, right? Yes, this will stay. Yes, microdermapeds is there. Okay, no, that's all right, Dr. Path. It is always a good thing to ask silly questions, because how else otherwise are you going to learn? Okay. So endotoxin, I have told you there are 2 types of toxins produced by bacteria, exotoxin, endotoxin. Endotoxin ke liye kya hota hai? Exotoxin is like missiles. Okay, it is thrown exo, it is thrown out. What is an endotoxin? It is a part of the cell wall of the bacterium. So if the bacterium has to kill somebody, if the bacterium has to trigger this bomb, it has to die itself. So What happens in endotoxin? Cell lysis. It's like a suicide bomb. The bacteria has to die itself before it can kill anybody else. So that is lipid A, that is endotoxin. It is very much an integral part of the lipopolysaccharide layer, which is a very much an integral part of the cell wall of the gram-negative bacteria. Okay, of the gram-negative bacteria. ठीक है. सो एलपीएस कहाँ पे है? आउटर मेम्ब्रेन के ऊपर है. इनर मोस्ट क्या है? एलपीएस. आउटर मेम्ब्रेन में कौन से प्रोटीन्स है? आउटर मेम्ब्रेन प्रोटीन्स विच आर द पोरिन प्रोटीन्स. सारा फीचर किसका है? ग्राम नेगेटिव. जस्ट रिमेंबर, पॉजिटिव पीपल आर वेरी वेरी सिंपल पीपल. दे हैव नथिंग. दे जस्ट हैव वन पेप्टिडोग्लाइकन एंड वन लाइपोटिकोइक एसिड विच हेल्प्स इन adhesion, which here is a p-value. Okay जी, चलो जी. Now let's talk about motility. This is the most favorite topic of everybody. Everybody easily remembers. Who shows tumbling motility? What is tumbling? Tumbling is somersault. So, tumbling is cartwheels. When do we do cartwheels? When we are very very happy. Happy means hysteria. Hysteria rhymes with listeria. So listeria monocytogenes shows me the tumbling motility. And when are we happy? When it is extremely hot or when it is nice and pleasant and cold. So the bacteria is also very very happy and hysterical and listerical or listeria when it is very very nice and cold outside. So it loves cold enrichment. Cold enrichment. What is cold enrichment? That it loves to tumble at 25 degrees Celsius. Okay, so at 25 to 27 degrees Celsius, this guy is going to show me tumbling motility. Nothing at 37 degrees Celsius. So this is called as differential motility. So this brings me to another question. How many bacteria actually show you differential motility? Tell me. डिफरेंशियल मोटिलिटी का क्या मतलब है? अगर गर्मी लगेगी तो नहीं चलेंगे, ठंड लगेगी तो चलेंगे. So differential motility means that they are motile at 25 degrees Celsius and they are non-motile at 37 degrees Celsius. One is of course listeria. Who are the other two guys? You have two close cousins of Yersinia pestis. What are their names? Yersinia enterocolitica and Yersinia. Which Yersinia? Pseudotuberculosis. ठीक है. Yersinia pseudotuberculosis and Yersinia enterocolitica and Listeria monocytogenes. What are they? Differential motility. Please do not confuse it with Yersinia pestis, because Yersinia pestis is always non-motile. It does not matter what temperature you're giving it. Yersinia pestis is always non-motile. Chalo ji, darting and shooting motility. Everybody remembers Vibrio cholerae. Vibrio cholerae plus Campylobacter. Okay. Campylobacter, Vibrio cholerae, S-shaped bacteria, they show darting motility. Why did I not write Vibrio species? Because another guy of the Vibrio species, which is Vibrio parahemolyticus, shows me swarming motility. And what is the mnemonic for swarming motility? If there are any students of mine, they will write it. Otherwise, I'll let you know. Swarming motility क्या होती है बेटा? Swarming motility में आप ऐसे imagine करो कि एक बेचारा एस्ट्रोनॉट बैठा हुआ है मार्स पे और कॉर्पोरेट हो गया अब तो सब. द स्पेस स्टेशंस आर बीइंग टेकन अप बाय इलॉन मस्क एंड देयर आर ऑल द प्राइवेट कंपनीज कमिंग अप टू फॉर द स्पेस स्टेशंस, राइट? सो दे आर ऑल corporate employees at the end of the day. These astronauts, what do corporate employees do? They make PPTs. Okay, so this astronaut bichara is sitting on Mars and is making a PPT. So what is he? He is a bichara. He's quite serious. He's making a PPT. And where is he making the PPT? On Mars. Mars is the red planet. When a bacteria swarms on a blood agar plate, it looks like Mars. Okay, so swarming motility is bichara PPT on Mars. Bichara means Bacillus cereus. P stands for Proteus. Then you have Vibrio parahemolyticus. Okay, that is why I said not Vibrio species. Only and only Vibrio cholerae shows me darting or shooting, because Vibrio parahemolyticus is here showing me swarming. T stands for Clostridium tetani and Mars is Serratia marcescens. Serratia marcescens. ठीक है. So these are your swarming motility. फिर आती है तुम्हारी spiral. Oh sorry. Hmm. Is it visible? Yeah, okay, thank God for that. Chalo. Then what did I say? Corkscrew motility. Everybody knows this. What is corkscrew motility? By the spirochetes. Spirochetes. Okay. Gliding क्या होता है? What is gliding? So gliding is like gliding on snow. Just imagine a few penguins just gliding across snow, or a paragliding guy. What are they? Mycoplasma. Then you have falling leaf. Falling leaf. SA, oscillatory. Falling leaf. What is that? Giardia. Lamblia, which is a parasite. Vibrating or twitching. T for twitching, T for trichomonas. Vaginalis, which is also a parasite. Stately या slow motility किसकी होती है? It is the Clostridium. What is stately? Stately means sluggish and slow. They are very, very lazy. They don't want to move around. What is that motility? That is called as stately motility. Which guys? Clostridium. But which Clostridium? All Clostridium except tetany. Except tetany are going to show you stately motility, because tetany is busy doing what? PPT on Mars, that is swarming. Chalo ho gaya. ये रहा तुम्हारा बेचारा. Oh, I had forgotten that I had given you the image. This guy is showing you swarming. PPT on Mars. Can you see swarming? This is the swarming of the concentric circles, and this is looking like the red planet, which is the Mars. और इस Mars पे कौन बैठा है? बेचारा astronaut making a PPT. All right. So you can remember it. बैक्टीरियल ग्रोथ कर्व पे आते हैं. बहुत ही आसान है जिंदगी, बहुत ही आसान. देखो, दो चीजें याद रखनी है तुम्हें. इंसानों का जीवन कैसे चलता है? हाउ इज द लाइफ ऑफ ह्यूमंस? हाउ डज इट हैपन? फर्स्ट ऑफ ऑल, यू आर लेजी, यू आर अ बेबी, यू आर जस्ट लाइंग इन बेड, यू आर जस्ट ईटिंग एंड यू आर जस्ट गेटिंग फैट. ओके, दैट इज द लैग phase. Then starts your youth. Youth of the bacteria is like the log phase. What happens in youth? I will tell you everything what happens in youth, and then you will be able to analyze it with the features of the bacteria also. What happens in youth? When you are young, you're very, very active. You're jogging, you're active, you're running, you're jumping around. Then you're meeting people. You want to meet people. You're very interactive. You're very metabolically, biochemically active. You care about makeup, you care about how you look, you will put on a nice gram stain and go out of the house. This is all the youth of bacteria. And in the youth, you're very, very sensitive. Koi bol de a pagal, a duffer, you get hurt, like what did he say? If today in my age anybody tells me a duffer, a pagal, I will not even care. But when I was young, I used to care a lot. So you are very, very sensitive. So if I put some antibiotics on you, you are going to succumb to it. So you are very, very susceptible to antibiotics. So what all do you perform in log phase? In log phase, there is metabolic activity, biochemical activity, antibiotic susceptibility, and gram staining. Best time to do gram staining, log phase. Best time to do AST, log phase. Best time to do biochemical testing, log phase. So what is log phase? There is youth. And what happens in youth? You multiply, you reproduce. So this is the time when the bacteria is having maximum reproduction or maximum cell division. So what is the phase at the end of which there will be maximum babies? It is going to be live phase. Then stationary phase. Stationary phase is ओके लक्ष्मण, बट आपकी माइक्रोबायोलॉजी की जिंदगी आसान हो जाएगी इफ यू जस्ट स्टिक थ्रू दिस मैराथन. जस्ट बियर 4 hours of torture and your zindagi will become asan. All right. So stationary phase. So see what is happening in the bacterial growth curve. I have taken a test tube and I have put the limited amount of bacteria inside the test tube. There is going to be a point of time when that nutrition or that broth is going to, exhaust. Now what happens? Whenever in the world there are exhaustion of resources, what happens? War. Okay, so stationary phase is like the war zone. Now death has started happening in the bacterial world. Bacteria has started killing other bacteria. Death is happening. War is happening. There are two types of people in war. There is one type of people who are hiders, okay, who hide. And then the other type of people who are fighters. They fight. So the bacteria are also of two types, the hiders and the fighters. What will the hiders do? They will go and hide in bunkers. They will hide behind blankets. So sporulation happens when? Sporulation happens at the stationary phase. Then when you have to go and hide in the basement, in the bunkers, you will keep big boxes of food. Dabba भर भर के खाना ले जाओगे, right? What are that big boxes of food? Storage granules. In which phase is storage granules formed? In the stationary phase. Then you have the fighter bacteria. What will the fighters do? They will fight with missiles and bombs. So exotoxins are missiles and endotoxins are bombs. You know it. So exotoxins and endotoxins are produced in which phase? Stationary phase. This is the time when the bacteria becomes attacking, offensive. It is going to attack other bacteria. How is it going to attack? Where do antibiotics come from? They are produced by the bacteria themselves. So in which phase is the bacteria going to produce antibiotics to kill other bacteria? That is the stationary phase, है ना? तो किस phase में it is the most susceptible to antibiotics? Log phase, youth. In which phase it is the most production of antibiotic done by the bacteria? Stationary phase. There is a fine difference, okay? Then bacteria also release bacteriocins, etc. In the war, do you care about makeup and how you're looking? No. So if at this point I give the bacteria some gram stain, will it care? No. Finally, what happens after war? There is death. So of course there is death phase. What happens after war? You have mutilated bodies. So in the bacteria you see involution forms. Now the question, in which phase is capsule formed? Now most students tend to confuse capsule and spore. ठीक है, spore बनेगा stationary phase में because spore is a hiding place for the bacteria. So in the war they are going to hide. But what is a capsule? A capsule is a shiny car. A capsule is a well-tailored fitted tuxedo. When do you care about well-fitted clothes and shiny cars? In your youth. So in the log phase the capsule is going to get formed. So okay, sorry, I will just scroll down to the bottom. Yeah. So you tell me, in which phase are you going to see the maximum size of bacteria? Obviously, बैठ के खा पी के मोटा हो रहा था. There was no cell division happening here. So maximum size is achieved at the end of lag phase. When is the maximum viable count achieved? Here. At the end of log phase is the maximum viable count. ठीक है? Then one is the maximum total count, dead plus alive. That is end of stationary phase. Here you have maximum total count, dead plus alive. ठीक है? So now what all was happening in the lag phase? No division, only increase in size. What happens in log phase? पागल हो गया बैक्टीरिया. Exponential division. Divide करे ही जा रहे हैं. There is exponential cell division. There is no death happening here. Log is jog. Log is youth. What all happens in log? We have just seen. What all happens? Maximum metabolic activity. Maximum antibiotic susceptibility, maximum biochemical activity, then best time to gram stain, and you have your capsule formation. ठीक है? Then you have stationary phase. What is happening in stationary phase? New cell division is equal to cell death, right? Is equal to cell death. War has happened. Stationary is war. So the hiders will hide in bunkers. So sporulation, storage granules. Tell me, in which phase of the bacterial growth curve is the volutin granules of Corynebacterium diphtheria form? Okay, all right, we can increase. Yes, correct. अच्छा, मुझे बताओ, guys, tell me the question. In which phase is the volutin granules of Corynebacterium diphtheria formed? Volutin granules बताना. And then, of course, this is the phase in which antibiotics are produced. produced. Okay, this is the phase in which colicin is produced. This is the phase in which toxins are produced. Toxins are produced. ठीक है, and यहाँ पे क्या बनता है death phase में? Involution forms. Involution forms, right? Death is more than division here. Yes, stationary में बनेगा आपका diphtheria का phase. चलो, now let's look at the horizontal gene transfer. What is horizontal gene transfer? Two bacteria sitting next to each other, unrelated, not mother and child, they are able to transfer the genetic material from one to the other. What is that? That is horizontal gene transfer. There are three ways of doing that: transduction, conjugation, and transformation. So what is transformation? In transformation you have passive uptake of naked DNA. क्या हुआ? Passive uptake of naked DNA. Passive uptake of naked DNA is what is happening here. And what was the experiment? Whenever you hear one a capsulated strep pneumoniae, uncapsulated strep pneumoniae, killing a mouse, not killing a mouse, what is all that shebang about? That is about Griffith typing, Griffith experiment. Griffith experiment. So whenever you hear this shebang of capsulated bacteria, non-capsulated bacteria, heat-killed bacteria, inject into a mouse, mouse is dying, mouse is living, what is that? Griffith experiment. All of that was done to prove transformation. Then we have transduction. What is transduction? It is like abduction. Somebody is coming and abducting a particular DNA of a bacterium and giving to the other. What is that abduction done by? By a bacteriophage. So what is bacteriophage going to do? It is going to take up a little piece of the bacterial DNA and give it to another. What is this? This is mispacking. Okay, mispacking. So transduction can be of two types. You have generalized transduction. What happens in generalized transduction? Bacteria comes, picks up the DNA of bacteria A and gives it to bacteria B. That is generalized transduction. What is this called as? Mispacking. So you're in your hostel. Okay, you have your bed, bistar, pankha and everything in your hostel. Now something belongs to the hostel committee and something belongs to you. What belongs to you? Your books, your mattress, your bed sheets. But what does not belong to you? The bed, the table, the chair, the fan. You called the packers and movers while you were shifting home. What did they do? They mispacked. They also took out the fan and packed it in your saman. They also folded up the bed and put in your saman. They also folded the table, put in your saman. What is that? Mispacking. You are taking home something which did not belong to you. So the bacteriophage is picking up a DNA which did not belong to the bacteriophage. It is picking up the DNA of bacteria A and then transferring it to bacteria B. Simple pick and drop is happening in mispacking. What is specialized transduction? Specialized is also called as restricted transduction. What does that mean? That it is only going to pick up a restricted portion or a specialized portion of the chromosome of bacteria A. So suppose bacteria A had penicillinase producing gene. So only and only that penicillinase producing gene is going to be picked up from bacteria A and transferred into bacteria B. How did this specialized transduction happen? This happened because of missplicing. So when the DNA was supposed to be cut, it was cut at a wrong place. So by wrong cutting, you have also picked up one specialized or restricted portion of the DNA. Okay, so not going into the details of all of it, just remember the buzzwords. What is it? Generalized is mispacking. Generally you have mispacked. You have taken up the chromosome of bacteria A, given it to bacteria B. Who is doing this? Who is the packer and mover? It is the bacteriophage. What is specialized or restricted? You have taken one particular segment of the DNA and given it. Why? Because you cut it out wrong. So that is missplicing. Okay. What is conjugation? You know it. Conjugation means forming of a conjugation tube. The bacteria which has the fertility plasmid and is able to make the sex pilus is called as a F plus cell. The bacteria which does not have the fertility plasmid, hence cannot make a sex pilus, is called as an F minus cell. When an F plus cell conjugates with an F minus cell, what do you get? Both become F plus. Guys, bacteria is not stupid. Bacteria is not an philanthropist that he's going to give the chromosome, give the plasmid and sit idle by itself. No, it is like दूसरे का भला करो पर सबसे पहले अपना भला करो. Okay, so दुनिया का भला हो शुरुआत मुझसे हो, that is the concept of the bacteria. So yes, they are going to give the plasmid to other guy, but they are going to send out a copy. So it is not cut and paste, it is not cut and paste, it is copy and paste. So the bacteria are picking up a taking a plasmid, creating its copy, then giving it to the other guy. So both end up becoming F plus. Then you have something which is called as an HFR. What is an HFR? High frequency recombinant cell. It is a very, very selfish guy. What happens? This is a single bacterium only. This is the chromosome of bacteria. This is the plasmid of bacteria. The plasmid falls in love with the chromosome. The plasmid comes and attach with the chromosome. Now ये कह रहे हैं अब हम जन्म जन्म के साथी. We are now one entity. We are now one unit. We are binding to the chromosome of the bacterial cell only. So what have we become? We have become a high frequency recombinant. Why? Because anytime this guy can break off from here and go back home, anytime it can come back and join again. So this is very high frequency recombination keeps on happening, wherein a little portion of plasmid can keep binding to the chromosome and keep detaching also. So when this high frequency cell, this comes across this negative guy, this poor guy, this high frequency recombinant cell is quite selfish. It is very, very selfish. It is not going to give anything to the negative guy. So the status quo is going to maintain. HFR conjugates with F negative, you end up getting HFR and F negative only. Nothing changes. Selfish, because the size of the chromosome has become big here. Now, when this HFR was going to break off, it should have broken off in a manner where the green portion separated and the red portion separated. But of course, bacteria are quite stupid. While splicing, they make a mistake. So what mistake did he make? Instead of splicing from this point, it spliced from this point, right? So now along with the little red portion, it has taken a little green portion also. However, it is still a plasmid. So now this does not look like this guy. This does not look like this guy also. So this is a totally different guy. It is an F prime guy. And F prime behaves just like F plus. So when F prime conjugates with F minus, you end up getting both F prime. Again, a copy paste, not a cut paste. Yes, दूसरों से पहले खुद की जय कीजिए, करेक्ट है ना. चलो, so now let's quickly talk about disinfectants. This is quite easy. So you have sterilants and disinfectants. What are sterilants or sterilizing agents? These are the guys which kill the spores. So sterilant is the one which kills spores. S for sterilant, S for spores. ठीक है. There are physical and chemical sterilants. What are physical sterilants? They can be heat-based methods or non-heat-based methods. Heat-based can be dry heat, moist heat. Moist heat simply means steam, which means autoclave. So dry heat, moist heat is going to be more than 100 degrees Celsius. Whereas the non-heat-based methods are going to be filtration and radiation. ठीक है. तो ड्राई हीट में क्या मोर देन 100 which is your hot air oven, incineration, flaming, all of that. Then amongst the chemical sterilants you have ETO and plasma sterilization. ETO and plasma sterilizer. ठीक है. अब देखो बहुत आसान है लाइफ. आपको इसमें ना मेजरली क्या याद रखना है? यू हैव टू रिमेंबर द spores, the biological indicator spores. ठीक है. So what do you do? How have we written it? We have written first, we have written hot air oven, then we have written steam heat का मतलब ही है autoclave, then we have written autoclave. Over here on the chemical side we have first written ETO, है ना. First we have written ETO and then we have written plasma sterilizer. देखो ये दोनों जो आसपास लिखे हैं, the ones which are written in front of each other, they are going to have the same indicator, the same biological indicator. So hot air oven and ETO, both of them are going to have Bacillus atrophius. A for atrophius. ठीक है. And autoclave and plasma sterilizer, both of them are going to say Geo mere lal, Geobacillus stereothermophilus. Stereothermophilus. एक रह गया radiation is Bacillus pumilus. And filtration में spore की जरूरत नहीं है. This does not involve killing. In filtration there is no killing involved, there is only physical removal involved. So question, which method does not involve killing? It is filtration. So no killing is involved, so you don't need to check with spores. So you just need Brevidumonas diminuta or the red colored bacterium Serratia marcescens, because that is red colored, so we can easily find out whether filtration has happened or not. ठीक है, marcescens. Acha, what about these disinfectants? Disinfectants are the guys which do not kill the spores. Only the high level disinfectants, when used for a very high concentration for a very high amount of time, then they can convert into chemical sterilants, otherwise they will never kill the spores. ठीक है, इसमें होते हैं APH, aldehyde, paracetic acid and H2O2. Then intermediate level also has APH, alcohol, phenol and halogen, and low level has the chlorhexidine, which is nothing but the mouthwash. ठीक है, इनके कैसे तुम्हें concentrations याद करने हैं? Remember aldehydes. So in aldehyde you have something which is called as glutaraldehyde. So can I write it as gluturaldehyde? Gluturaldehyde. So 2 is standing for 2 to 2.4% for 20 minutes. ठीक है, 20 minutes. Then you have the other aldehyde, which is formaldehyde, which has been discontinued. Then you have orthophthaldehyde. इसमें क्या लगा? Hyphen. What is this hyphen telling you? That this is half a word. So half stands for 0.5%. 0.5%. ठीक है. Then you have paracetic acid. Paracetic acid is P. P means point. Point and 5. So this is 0.1% and 5 minutes. Then H2O2. H2O2. 2 plus 2 is 4. More than 4% is sporicidal. So if you increase concentration of H2O2 to more than 4%, it can also start to kill the spores. Okay, it becomes sporicidal. So just remember, gluturaldehyde 2% for 20 minutes, orthophthaldehyde 0.5%, paracetic acid 0.1% and 5 minutes, and H2O2 2 plus 2 is 4, add 4, more than 4% is sporicidal. Then you have to of course remember all of these indicators of sterilization. ठीक है ना? चलो, now let's talk about the cell wall of fungus. मैंने क्या बोला था? cell wall में क्या है? Chitin. This chitin is the layer which is stained by the lactophenol cotton blue, and this is the stain which layer which is stained by the chalcofluor white stain. Then you have the beta-glucan, which is the most important layer. Okay, so you have certain important antigens of fungi. You have certain important antigens of fungi. What are those? You have 1,3 beta-D-glucan. ठीक है, 1,3 beta-D-glucan. Then you have Galactomannan. And then you have the capsular polysaccharide. So 1,3 beta-D-glucan is shown by Candida and Pneumocystis pneumoniae, PCP. In fact, in PCP, the 1,3 beta-D-glucan is going to be very, very high. Okay, very, very raised. Galactomannan is specifically shown by Aspergillus. Specifically your invasive aspergillosis. ठीक है. Capsular polysaccharide, no points for guessing. It is Cryptococcus. Cryptococcus is the only capsulated fungus, right? Then you have mucorales. Mucorales are very battameez. They will not show anything. They will not show any of these antigens. What are these? These are the important cell wall antigens of the fungi. अगर 2 से 2 to 2.4% for 20 minutes याद हो ही रहा होता तो निमोनिक की जरूरत ही क्यों पड़ती? ओके, सो 2 से 2% तो याद हो जाएगा ना बेटा. सो 2 से 2% याद कर लो. 2 means 2% and 2 means 20 minutes. 2 and 20 from the gluturaldehyde. ओके, चलो जी. नेक्स्ट क्या है हमारा? व्हाट इज दिस इमेज? दिस इज एन LPCB इमेज. कैन यू सी द ब्यूटिफुल हेयर floating about in air? What is this? This is aspergillus. You can see the conidiophore, you can see the vesicle, you can see the biceriate, and you can see this line of conidia on top. What is this? This is aspergillus. Now, which aspergillus is this? Let me see if I have the image. Okay, yes, so we will come on to it. So what is this? This is the LPCB mount image. What is this? This is LPCB. What is LPCB? LPCB is lactophenol cotton blue. There is lactic acid, you have phenol, and then you have cotton blue. Which of the two is the stain? Cotton blue is the stain. That is why it is looking blue. Phenol is a simple disinfectant, right? It is going to inhibit other commensal bacteria and other commensal fungi. What is lactic acid? It is going to preserve the morphology. Preserve the morphology of fungus. That is lactic acid. Okay. What is this? This is SDA agar, Sabarodes dextrose agar. What is the feature? That the pH of it is acidic and it has antibiotics. What are the three antibiotics? Gentamicin, chloramphenicol, and cyclohexamide. Cyclohexamide is also called as actidione, right? Then you have a modification of this SDA. What is that called as? Emmons modification. Emmons modification of the agar. What is the Emmons modification? That the pH here is neutral. And for whom do we do it? We do it for the dermatophytes, because the dermatophytes are not able to withstand the acidic pH of normal SDA agar. So the Emmons modification of SDA is going to be for dermatophytes wherein the pH is neutral. Okay? Who is this? Everybody knows. Tinea or pityriasis versicolor, meaning variety of colors. You see certain hypopigmented patches and you also see certain hyperpigmented patches. Then what does it look like? It gives me the spaghetti and meatball appearance. Spaghetti and meatball appearance. And on the olive oil or on the oil SDA. Just a minute. On the oil overlay SDA, it is showing me the nice fried egg. Okay, oil में क्या बनाते हैं? Fried egg. So it shows me the fried egg colonies. Please do not confuse the fried egg colonies of Mycoplasma and मैलेसीजिया फर है ना? क्यों? बिकॉज नहीं मेड्यूसा यहाँ नहीं आएगा बेटा. मेड्यूसा इज बैसिलस एंथ्रासिस. ओके, सो माइक्रोस्कोपिक फ्राइड एग इज एक्चुअली माइकोप्लाज्मा एंड मैक्रोस्कोपिक बड़े बड़े फ्राइड एग इज एक्चुअली यर्सीनिया पेस्टिस एंड मैलेसीजिया फर. ठीक है, तो ये मैलेसीजिया फर में फ्राइड एग, दीज आर मैक्रोस्कोपिक, दीज आर क्वाइट बिग कॉलोनीज. ओके, whereas microscopic fried egg is mycoplasma. Spaghetti and meatball appearance is seen on the KOH mount. So this is pityriasis versicolor or tinea versicolor. You are seeing the hypo or hyperpigmented scaly lesions. Why is scaly important? Because you have to differentiate it from vitiligo. Then see, whenever in superficial mycosis I use the word tinea, I mean skin. Okay, tinea means skin. So if tinea has a variety of color of patches, it is tinea versicolor, which is by Malassezia furfur. Malassezia furfur. Okay, Malassezia furfur. Then if I'm using the word tinea nigra, then it has to be black. So you have the hyperpigmented black patches, especially on the palms and soles. So here you will only see the hyperpigmented patches, especially on the palms and soles. Okay, now wherever you hear the word black or niger in microbiology, whenever you hear the word black or niger, you need to use the word hortiae. ठीक है, और कहाँ ब्लैक दिख रहा है मुझे? यहाँ ब्लैक पिएट्रा, तो मैं यहाँ पे भी लिख देती हूँ hortiae. Okay, whenever you hear nigra or black, you use the word hortiae. So hortiae wernickeae is the one which is going to cause tinea nigra, and Piedreia hortiae is the one which is going to cause black piedra. What is piedra? Whenever I say piedra, I mean hair. So if on the hair there is a white nodule, it is white piedra. If on the hair there is a black nodule, it is black piedra. The moment you hear the word black, you think of hortiae. So if it is tinea nigra, it is black patch on the skin. If it is black piedra, it is black nodule on the hair. If it is black, it is hortiae. So tinea nigra is hortiae wernickeae, and black piedra is Piedreia hortiae. Then you have the white piedra. epiedra, it is the white nodules. So just remember, how does the lightning in the sky go? It goes like a white shiny light. Okay, so what is the lightning in the sky called as? Bijli. So the white piedra is going to shine like bijli. So you have trichosporon bijli. Trichosporon bijli. Okay, bijali is bijali. So trichosporon bijali. Now, what is sporon? Sporon means yeast. You have another guy whose name is trichophyton. Another guy whose name is trichophyton. So, what is the meaning of phyton? Phyton means hyphae. So, now we are dealing with dermatophytes. So, dermatophytes is going to be phyton, whereas the trichosporon was the spore which was making these nodules on the hair. Okay, and it was bijali, so it was white piedra. Okay, so now that brings us to dermatophytes. Now you have a very easy way to remember dermatophytes. ठीक है, dermatophytes को याद करने का बहुत आसान तरीका है. अभी तक क्या हो रहा है? अभी थोड़ा सा इमोशन बढ़ रहा है ना, अभी थोड़ा गुस्सा गुस्सा आने लगा है. So what are you having? You are having too much emotion. You're having too much emotion. So this is trichophyton. What is much? It is microsporum. What is emotion? Epidermophyton. अब देखो भाई, dermatophytes हैं तो they are going to cause infection in the keratinized layer. Wherever there is keratin, they are going to cause infection. Okay, so keratin is there in hair, on the nails, and on the skin, right? So skin, hair, and nail. So trichophyton has tri in its name. So tri means all three. So this guy is going to cause infection in skin plus hair plus nail, all three. Microsporum, how do we remember it? By much H. See, they are dermatophytes. So all the dermatophytes will definitely cause infection in the dermis. So they all three will cause infection in the skin. All right, then trichophyton had tri in its name, so it will cause in skin, hair, and nail. Much has H, so this is going to cause infection in hair. Then emotion as well as epidermophyton has N in its name, so it is going to cause infection in nail. So skin, hair, and nail, skin plus hair, and skin plus nail. This is the division that is there in trichophyton, microsporum, and epidermophyton. Now, when you look at these guys, you will see that there are some small dots which are produced, and these big guys which are produced. So these big spores are called as macroconidae, and these small dots are called as microconidae. So how to remember that which one has a lot of microconidae and which one has what type of macroconidae? So just write microconidae and macroconidae. So what happens in life when you start reading microbiology, you remember a lot of it. So initially you remember abundant micro. So in the first guy, there is going to be abundant microconidae. Then as you move forward, you tend to forget micro. So there are few microconidae in microspore. Then towards the end, all vanished, all gone. Absolutely no microconidae are seen in epidermophyton. Okay, then what about macroconidae? Macroconidae are signifying your emotions. So as you're reading forward, your emotion is rising. So in the beginning, you have thin emotions. So thin pencil shaped emotions were there. Then as you moved forward, your emotions start growing and they became like a spindle. Then towards the end, you wanted to take up a bat and hit somebody. So much emotion you are getting. So what is that? Bat shaped or club shaped huge macroconidae. So if I write that I can see no microconidae and huge bat-shaped macroconidae, you know I am talking about epidermophyton. And there is only one species of epidermophyton that you have to care about, and that is epidermophyton flocosum. Can you see the same? Yes, you can see the same. You can see absolutely no microconidae here, and you can see the big club-shaped macroconidae over here. What is this? This is epidermophyton flocosum. So what all history can you get for epidermophyton flocosum? Either a skin lesion or onychomycosis. N is onychomycosis, right? Matlab nail is onychomycosis. Then you have amongst microsporum, you have two important guys, Microsporum gypsum and Microsporum canis. I am interested in canis. Why? I will come to it in a little bit. Then you have amongst the Trichophyton, you have two important guys. Okay, you have Trichophyton ruberum and Trichophyton mentagrophytes. Mentagrophytes. Okay, see, menta is mental. Mental means loopy. Loopy means spiral. So Trichophyton mentagrophyte shows me spiral hyphae. Spiral hyphae. Okay, so let's just make a division here. Yeah, it shows me spiral hyphae. Then you have ruberum. Ruberum means red. So it shows me the red pigment. There are only two fungi that you have to remember which produce the red pigment. One is Trichophyton ruberum and one is the one जिसमें हमने मार के लाल कर दिया था, that was Marnefi, Talaromyces or Penicillium marnefi. ठीक है. Then amongst ruberum and mentagrophytes there is another differentiation. There is something which is called as hair perforation test. What is hair perforation test? You take a naive piece of hair, meaning you take a newborn baby and you take a piece of hair of a newborn baby. And then on that newborn baby's hair, you take a piece of the moldy growth. These are all molds, yeah? Dermatophytes are not yeasts. These are all true molds. Please don't make that mistake. So these true molds you pick up and you put on this hair. Now these molds are going to eat up the hair. Right? How are they going to eat up the hair? Like dogs. Yes. So dogs की तरह they will eat up. ऐसे पूरा perforate कर देंगे hair shaft को. They will eat it up like dogs. So when I am saying dogs, I can say canis. Yes. And when I say dogs, can I say men? Yes. So when I say dogs, I can say men. So the mnemonic is men can. What is men can? Men stands for Trichophyton mentagrophytes. And can stands for Microsporum canis. Microsporum canis. So if you have to differentiate between ruberum and mentagrophytes, in mentagrophytes you will see spiral hyphae and you will see hair perforation test is positive. Whereas in ruberum you will see a red pigment and you will see hair perforation test is negative. Hair perforation test is negative. And you know how does ruberum look like? This is a hypha and on top of this microconidia are sitting like a bird on fence. So Trichophyton ruberum shows you the bird on fence appearance. Bird on fence appearance. So who has loopy or spiral hyphae? The mental mentagrophytes. Who has bird on fence? Remember a beautiful red bird. So this is Trichophyton ruberum. Microsporum canis is also hair perforation test positive. Because when I say dogs, I can say men. So men can, mentagrophytes and canis. Flocosum is totally different. Okay, over here you can see the spiral hyphae, the spindle-shaped macroconidia of Microsporum canis. Over here you can see the spiral hyphae and the small little microconidia and the pencil-shaped macroconidia of Trichophyton mentagrophytes. Right? All dermatophytes are urease positive. Okay. So you have a mnemonic for urease positive fungi. Okay. Chalo. We will write it over here. You have a mnemonic for urease positive fungi. Urease positive ka matlab UP. So what is the mnemonic? UP ka Deputy Chief Minister. UP ka Deputy Chief Minister. So this is urease positive. And what is DCM? DCM. D is dermatophytes. What is C? Cryptococcus. What is M? This guy, Malassezia furfur. Malassezia furfur. All three are urease positive. So when I say all dermatophytes are urease positive or not, so out of T mentagrophytes, Microsporum canis Epidermophyton flocosum. Who is urease positive? All three, because dermatophytes are urease positive. Cryptococcus, urease positive. The one which causes tinea versicolor, which is Malassezia furfur, is urease positive. So UP card Deputy Chief Minister is urease positive. Then whenever you see copper penny bodies, and what is the history you're going to get? Trauma. So whenever there is a history of trauma or thorn prick, what do you get? You get three possibilities. Okay, either the answer is mycetoma or eumycetoma, which is Madura foot, or the answer is chromoblastomycosis. Chromoblastomycosis. Or the answer is sporotrichosis, which is Sporothrix schenki. Okay, these are the only three possibilities, and this in fact was a straightforward MCQ asked in INI-SET. Straightforward. These were the exact three options, and the fourth option was something else which you had to mark as an exception. Okay, mycetoma, chromoblastomycosis, and sporotrichosis. Male students feeling sad? Okay, fine. See, not all men. Okay. Chalo ji. So now we're talking about copper penny bodies. These brown little balls which are looking like copper penny. Name itself is telling you that I'm colored, so I am chromo. Okay. I am a ball shaped structure, so I am blasto. And I am a fungus, so I am myces. So I am chromoblastomyces. Okay. So chromoblastomyces shows me the copper penny bodies, which are also called as the medullary bodies, which are also called as the sclerotic bodies. Medullary bodies, copper penny bodies, sclerotic bodies, everything shown by chromoblastomycosis. And the lesion that you get is called as warty lesions or verrucous plaques. Verrucous plaques or warty lesions are shown by chromoblastomycosis. Mycetoma and chroma confuses. Yes. So when you see verrucous plaques or warty lesions, you have chromoblastomycosis. What will you not get here? You will not get no granules or discharging sinuses. Because ये granules or discharging sinuses का ठेका किसने लिया हुआ है? Who has taken the tender of granules and discharging sinuses? It is mycetoma. Where is mycetoma? Okay. Maybe we don't have an image. No problem. So we will write it. If you get the question specifically mentions granules or discharging sinuses, then you're dealing with mycetoma. Mycetoma medullaris. Right? What is that you will see? Tumor like lesions which have discharging sinuses. And what is sinuses discharging granules? In mother foot have black granules. There is fungal called as Budo. Okay, and who's the guy? Mycetoma. Granules. Right. Madura. But no is there. So no yellow ices and the one is mycosis. Sporotrichosis. You got it. He's a diagnosis. And that's going to be the east as in the blue. Tissues. At the and have around such shaped. So it's okay. Cigar shaped is an asteroid base. It's a 100 degrees. If a patient is a rose gardener, the patient is a gardener and the rose gardener is. And the patient is an ulcerated nodules. Lymphocytic scars. What are these ulcerated nodules? Are long defects. Right. Now, this is rose gardener. This will roses or flowers. This is in easy. This is called floral ligation. Floral ligation. Okay, all the problem voice video next time. Is it all right? Take this in mind. It's in mind. Okay, guys, I think we are live now. Everything seems fine now. Guys, please let me know if I'm visible, audible. I can see that I seem to be visible and audible. So, should we take up sporotrichosis again? Yes, yes, there was a glitch. There was a glitch. I have noticed. We fixed it, it seems. Right, guys? Yes, all right. So let's quickly. So I think this is where we had done it up till Madura foot, like I was telling you. So when it is Madura, it is you have this please let me know. Sure. So I'll continue. Like acetoma of a tumor granules. The granules are true from Mirella Mumps. Okay. Then if you have a rose gardener, so the history of somebody from Madhya Pradesh, from orchards, a thornic, and a thornic have a long lymphatic no exception. That is both his cherries. Hi guys, can you hear me now? Yeah, back. Guys, please let me know if we are back. All right, guys, so we are back. I hope all of you are also back. Yes, so that's the thing about technology. The more advanced we get, the more glitches we have to face. Nevertheless, we are back, and how? Back and stronger. So, many of you had said that. So, please pass on the message to your friends also that we are back and we are going to continue the session. Right, so this is where we had stopped. We had stopped at mycetoma. Madura foot is something that had started glitching, and many of you could not see anything over there. So when we talk about Madura foot or mycetoma, then the thing, specific history that you're going to get is, of course, thorn prick, right? So this is what we were talking about. If you get any history of trauma or any history of thorn prick, you have three possibilities. One is mycetoma, one is chromoblastomycosis, and then one is sporotrichosis, right? So we have seen what is chromoblastomycosis going to look like verrucous plaques or warty lesions, and there will be no granules or discharging sinuses. And upon microscopy, what will you see? You will see chromoblast. What is a chromoblast? A colored round body. So you are going to see brown colored copper penny bodies, also called as medullary bodies, also called as sclerotic bodies. Okay. Then how do you know whether it is Madura foot or mycetoma? You will have a classical history of discharging black granules from a tumor-like mass on the skin. Okay, so there is a tumor-like lesion called as tumefaction, plus discharging sinuses, plus granules. This is a rule, this is a triad. And if the granules are black in color, that is a true fungal infection or eumycetoma caused by Medullella mycetomatis. Okay, if it is yellow, white or red granules, then it is any bacterial cause, which is actinomadura. Now bacteria can be actinomyces, it can be nocardia. So if it is yellow and sun-like sulfur granules, then it is actinomyces. Okay, all right, guys. Yes, so we are back with micro and we are doing micro. So please join in, everybody. Right. Chal. So now we move on. Then the next thing, the third DD of a thorn prick or trauma is Sporothrix schenki. And you know that Sporothrix schenki is a dimorphic fungus, meaning in the tissues at 37 degrees Celsius, it is going to be in the yeast form. And at 25 degrees Celsius or in LPCB or culture, it is going to show me the moldy forms, the mold form. And this is also called as the rose gardener's disease. So if you ever get a history of a rose gardener, person from Himachal Pradesh or Kashmir or like that, and then they say that there was a thorn prick and patients particularly presented with ulcerative nodules along the lymphatics. Okay, this is the only possible presentation. Please do not think of sporotrichosis with any other clinical finding or thing that they have given. Classically, always ulcerative nodules along the lymphatics. Then in that you see at the tissue at 37 degrees Celsius, you see the yeast forms, which is a central cigar-like body surrounded by a splendor Hopley phenomenon. What is that called as? Asteroid body, because it is looking like a star. So you have cigar plus asteroid bodies at 37 degrees Celsius in the tissues. When you take that tissue and you put it onto a culture media, which is SDA, obviously SDA is culture media for all fungi, then at SDA agar, when you make an LPCB mount of it, you will see floret-like conidiations or flower-like conidiations, because it is rose gardener's disease, so it is showing you the floret-like conidiations. All right, guys, is that clear? Right. Then we have Cryptococcus. Cryptococcus is the only crypto fungus. Crypto means hidden, so it is hidden beneath a capsule. You obviously know Cryptococcus is a cause of chronic meningitis. Chronic meningitis. Now, there are two species of Cryptococcus. Okay, one is Cryptococcus neoformans, and the other guy is Cryptococcus gattai. Now, what is the difference? Neoformans causes this chronic meningitis in immunocompromised people, immunocompromised host, whereas gattai is a go-getter. Gattai is a go-getter. So it is going to go and get the immunocompetent people as well. So this is going to cause an immunocompetent as well. And what is the selective agar for gattai? It is canavanine, CBG. ओके, CBG. अच्छा, न्यूफॉर्मेंस के लिए क्या फीचर है आपका? ऑफ कोर्स यू हैव दिस इज कॉल्ड नेगेटिव स्टेन. नेगेटिव स्टेन ऑल्सो कॉल्ड एज इंडिया इंक स्टेन और इंडिया इंक और योर नाइग्रोसिन स्टेन. Nigrosin stain. These are the special stains for it. What is the special culture media? Special culture media, it is bird seed niger agar. Yes, bird seed niger agar. What is bird seed niger? On this, the Cryptococcus is going to produce caffeic acid, and caffeic acid is brown in color, so it is going to produce brown colored colonies. Brown colored colonies on my this bird seed niger agar. How to remember? Bird. So Cryptococcus is spread through pigeon droppings, right? So bird, pigeon, they are also called as the rats of the sky because they spread nasty diseases such as cryptococcosis. So C for cryptococcus, C for capsule, C for chronic meningitis. And I always tell my students what, that the brain is inside a capsule. What is the capsule of the brain? The skull, right? So similarly, the agents which cause meningitis, whether bacterial or fungal, they are also inside a skull. They are also inside a capsule. So the capsulated fungus is obviously going to cause meningitis because the brain is also inside a capsule. Similarly, you have Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitidis. All three are capsulated and all three cause meningitis. So just remember, the meningitis brain is in a capsule, so the organisms which cause meningitis are also inside a capsule. All right. Chalo, so you have the bird seed niger agar, the caffeic acid, and the most important thing that you have to do in the detection of cryptococcus is to detect the capsular antigen. Capsular antigen. So in how many ways can you detect the capsular antigen? You can do the immunochromatographic testing and you can do a rapid card test. You can do the negative staining or the India ink test, wherein you see this unstained halo around cryptococcus, right? And yeah, there is one very important stain which is mucicarmin stain. Mucicarmin stain. Why? Because the mucicarmin is going to stain the carminogenic cell wall. Carminogenic cell wall. So cryptococcus cell wall is carminogenic, right? And that is why it is going to take up the mucicarmin. All right. Now let's talk about some true molds. What are these true molds? Aspergillus and mucor. Okay? Aspergillus and mucor are the true molds. What is the major difference? You see, both of them are hyaline hyphae. What is the meaning of hyaline? That they are colorless hyphae. Because colored hyphae are called as diatomaceous fungi or your phaeoid fungi, right? What is the difference between the two? If you look at this one, you can see the beautiful septations over here. You can see the equivalent branches. Equivalent मतलब जितना ये breadth है उतना ही ये है उतना ही ये है. There are equal breadths which are there. So this is dichotomous branching and this angle is acute angle, right? So your aspergillus has what? Aspergillus has acute angle, dichotomous branching, hyaline hyphae, right? Acute angle, septate, hyaline hyphae, right? That is aspergillus. What about mucorales? Mucorales have these 90-degree angle branches and they have these unparalleled walls, and even the branches are not dichotomous. It does not care that one branch is very, very thin and one branch is very, very thick. So these are non-dichotomous. So what about your mucorales? Mucorales include species such as Rhizopus species, Mucor species. Mucorales is the family, okay? It also contains hyaline hyphae. But what are the hyphae? They are ribbon-like, broad, aseptate hyphae, hyaline hyphae, right? And what is the branching? It is right angle branching or obtuse angle branching. Okay, Now, this is a photo. This is H&E staining of a tissue. What can you see? You can see the beautiful ribbon-like hypha, no septations, and even obtuse ankle branching, and no dichotomous feature also here, right? So this is your mucorales. Chalo. Next क्या है तुम्हारा? What is the difference in the LPCB mount? So tell me, is this a mucorale or is this an aspergillus? How to find out? You see this conidiophore, and then you see this nice vesicle. What is a vesicle? It's like a khopadi. It's like a head. And now what is the type of hair that is present on the head? So from this vesicle, you have these phyllodes or finger-like projections. And from these finger-like projections, you have chains of conidae coming above. Okay. So that is a typical feature of aspergillus. Your mucorale, on the other hand, is going to have a sporangium, round sporangium and columella, and sometimes when this sporangium collapses, it can look like a closed umbrella also, right? But what is very, very important is this rhizoids. What are rhizoids? These are root-like structures which are present at the bottom of these mucorales. So the guy which has the rhizoids is called as rhizopus, and the guy which does not have the rhizoids is called as mucor. Okay. So this is the difference, major difference in the LPCB mount of your aspergillus. So what all are you going to see in aspergillus? You're going to see a vesicle, and on top of the vesicle you're going to see the phyllodes. Now these phyllodes can be of two types. Okay, either it could be a biseriate guy. What is biseriate? That there are two rows, and it could be uniseriate, which means just one row of phyllodes, right? So now who is who? How to find out? If on the LPCB mount you have uniseriate phyllodes, then you have only one guy. Who is that? Aspergillus fumigatus. Now, how do we remember this? See, whenever it is uniseriate and conidae coming out, it is like a sparse growth of hair. है ना, कम बाल हैं इसके सिर पे. If it is biseriate, it has lots of hair on its head. So your flavorful flavors with the hair, kind of waving about in air, that is flavus. And then you have Aspergillus niger. What is niger? It is the black colored fungus with an afro type of a haircut. So over there you have a huge head full of hair. But this Aspergillus fumigatus is like a kind of Aspergillus fumigatus. And what is this black colored one? This is Aspergillus niger. No problem. Now another thing you have to remember about fumigatus, that on LPCB mount it shows me the pulled hair appearance. Now how to remember this? When you are smoking cigarettes, right, in the smoky green flame color, when you're smoking cigarettes and your mom catches you, what is she going to do? She's going to pull your hair and give you two tight slaps. So what are you going to look like? You're going to look like Aspergillus fumigatus. Just remember, if you smoke, you will get your hair appearance. And similarly, you just have to remember that is right? If it is mucorales, then there will be no rhizoids. Umbrella-like columella. Okay, so this guy is going to be like an umbrella, like this. Okay, so an umbrella-like columella, or no rhizoids, that becomes mucor species. If rhizoids are present, that becomes the rhizopus species. All mucorales are called as lid lifters or tube fillers. You see, this is the SDA agar in a Petri plate, and this is the SDA agar in a test tube. So you see how this Aspergillus is growing nicely, silently. You see, this is the slant of the agar, and Aspergillus is growing here. It has no business filling the tube up like that. But this guy has filled up the tube entirely. So whenever you hear the words tube fillers or lid lifters, that is, they are very rapidly growing, fast growing guys. Okay, they grow like fluffy cotton candies. Is there a lag still? Guys, let me know if there is a lag still. Okay, what to do now? Guys, is it audible? Guys, PDF you will get at the end of the lecture. You will get an annotated PDF at the end of the marathon. Is it okay now? Is it fine? Let me know. Yes, it's fine. All right. So when you see tube fillers or lid lifters, right? So what is the property of lid lifting? That when the this fungus is growing like cotton candy in a culture plate, it takes up the lid of the petri dish and it flows it away, throws away the lid. That is a lid lifter. So what is this telling you? That these are rapid growers, and they give the cotton candy fluffy growth. Cotton candy fluffy growth. Okay, fluffy growth. What will it cause? It causes rhino or rhino orbital cerebral rhinocerebro-orbital mucormycosis. What is that called as? Why is it called like this? Why is it called like rhinocerebro-orbital mucormycosis? Because it is locally invasive. It is locally very invasive. So much, and it is angio-invasive. So it is going to follow the path of the blood vessels. Wherever the blood vessel is going, this guy is going to go along with them. So it is going to enter through the nose, cause entire destruction of the nose, go to the orbit, destroy the orbit, go to the brain, start destroying the brain. So it is locally invasive, causes massive, necrotic eschar. Necrotic eschar. Right? So if whenever in the question you have the buzzword of necrotic eschar on the face, this becomes very important clue for rhinocerebro-orbital mucormycosis. Another clue which is going to be there is diabetes mellitus, DKA, corticosteroid therapy. Corticosteroid therapy. Okay, all of these are going to be your other important features. That is why during COVID times you were getting so many patients of rhinocerebro-orbital mucormycosis, because everybody was taking corticosteroids left, right, and center. Okay, there is a lag between video and voice. Please let me know if that is the case. वीडियो बहुत पीछे चल रहा है. Video is not in sync. Okay. Okay, guys. Let me figure it out. तो स्टीमिंग स्टॉप करनी पड़ेगी. Guys, you let me know if there is any improvement. Let me know if there's any improvement, or it's the same thing, voice and video lagging. Okay. Students are saying कि वीडियो रुक गई है लेकिन एक्सप्लेनेशन चले जा रही है मेरी. So that's a definite lag. अभी ठीक है? It seems same और सही है? So as I'm moving the hands, I'm speaking, guys. Okay, it's all right. अब सही है, right? Should I continue? Good, chalo, excellent, great. So next, what is this? The moment you look at this, you know this is a single yeast cell, and from this yeast cell wall, this little protrusion or tube is coming out. What are we looking at? We are looking at GTT. What is GTT? It is germ tube test. Germ tube test. What is germ tube test? Also called as Reynolds broad phenomenon. This is shown by whom? This is shown by Candida. Now, two species of Candida. So this is a test to find out the species, to speciate the Candida. Okay. So you have Candida, and what are the common features of all Candida? The moment you hear creamy pasty white colonies. Yes, creamy pasty white colonies. And then they are saying that okay, it is a oral thrush, it is a vaginitis, all of the Candida common clinical features you can identify by this point of time. And they are saying that on gram stain we are seeing budding yeast cell. So what is budding yeast cell written as? It is written as BYC, and this BYC is showing them pseudo hyphae. So based on this much, you know that this is Candida. Now, how to speciate the Candida? One, you do the germ tube test, and two, you do something which is called as the Dalmau plate culture. Dalmau plate culture. Dalmau plate culture, or it is also called as cornmeal agar. CMA, cornmeal agar, right? So what is germ tube test? This is shown by Candida albicans, either by Candida albicans or Candida dubliniensis. Candida dubliniensis. Now, how to identify between Candida albicans and dubliniensis? I take both of these Candida and I put them on a cornmeal agar. What is the property of cornmeal agar? It is nutritionally deficient. It is nutritionally deficient. So now what happens when there is no nutrition? Every microorganism creates spores. What are these refractile shiny spores of Candida called as? They are called as chlamydospores. Chlamydospores. So how do we differentiate? These chlamydospores are terminal. They are terminal, right? You can see that this is a pseudo hyphae. ये सब pseudo hyphae आ रहा है, और इस pseudo hyphae के end पे, at the terminal ends of the pseudo hyphae, you can see these refractile chlamydospores. So yes, these chlamydospores are terminal, but being terminal, if they are present singly or maximum in pair, or they are present in bunches. Okay, so if it is singly or in pair, then single means one, which is the first letter albicans. So this is Candida albicans. First letter is A. Then if it is in bunches, then B is followed by C and D. Okay, so it is A, B, C and D. So Candida dubliniensis. Dubliniensis is going to show you chlamydospores in bunches. Okay, B, C, D. BCT. चलो, फिर ये क्या है तुम्हारा? सबको पता है देख के ही. This is the crushed ping pong ball appearance. If you take a ping pong ball and you crush it from the top, this is what it is going to look like. So crushed ping pong ball is shown by your PCP. Who is PCP? Pneumocystis, pneumonia causing pneumocystis xerovici. Xerovici, right? Pneumocystis xerovici. Pneumocystis carinii is the old name. Okay, it's the old name. Now, this what was the another feature of PCP? Remember that the 1,3 beta-D-glucan is going to be very, 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 very high in the patients of PCP. This is of course going to cause disease in immunocompromised people. This is a very much a AIDS defining pneumonia. AIDS defining pneumonia. And what is the special feature of this pneumonia? Again, PCP. What is PCP? Plasma cell pneumonia. Plasma cell pneumonia, crushed ping pong ball appearance, pneumocystis jirovecii. These are all features of PCP. What is the CD4 count required for this? The CD4 count has to be less than 200. Please remember this. Okay, less than 200 CD4 count is the risk factor for, or is the significant factor associated with PCP pneumonia. Okay, and you see the crushed ping pong ball appearance. Who can tell me what stain is this? Whenever you see black colored anything, you know that which metal, which compound gets oxidized in air? It is silver. So it is gomori methamine silver stain. Always one of the stains used for fungus is GMS, and GMS gives the black color on the slides. Okay, so on the GMS stain, you are seeing the crushed ping pong ball appearance. What is the treatment of choice here? Treatment of choice is C for cotrimazole. Cotrimazole. Okay, cotri is the treatment of choice. What will you see on chest X-ray? On chest X-ray, you will get viral pneumonia-like picture. You will get bilateral infiltrates, ground glass opacities. So ground glass opacities bilaterally are seen either in viral pneumonias or in PCP. All right. Then you have another GMS. This is also GMS. This is also GMS. And guys, this is also GMS. So please remember, in GMS, the background being green is not important. The fungus being black is important. So over here, it is a brown background, but still the fungus is black. So this is all GMS staining. What can you see here? You can see the broad-based budding. You can see the B, right? BBB, broad-based budding, and this is shown by blastomyces. Blastomyces. Okay, blastomyces is important cause of what? North American blastomycosis. North American blastomycosis. It has the BAD1 antigen which causes the disease. Okay, so what is this called as? North American disease of blastomycosis. What are you seeing? Broad-based budding. You're seeing the broad-based budding in the yeast cell. Okay, it is called as blastomyces dermatidis. Then who is this? This is a sailor's wheel, mariner's wheel, pilot's wheel, or a Mickey Mouse appearance. So what is this? I always tell a story. What is the story? That you met a hot boy, he took you to your cave which was up north, then he brought a parachute. And parachute means paracoccidioidomycosis, right? And parachute से कहाँ ले गया वो आपको? He took you in his parachute to Brazil. Where is Brazil? In South America. So paracoccidioidomycosis is by Paracoccidioides brasilensis. And when he took you in his parachute, so obviously he was a what? How will you go to Brazil? He was a pilot, he was a sailor, he was a mariner. So all of these sailor's wheel, mariner's wheel, pilot wheel appearance, and later on you realize that he was an idiot boy, so he was a Mickey Mouse. A Mickey Mouse appearance. Okay, so all of these are paracoccidioides south, which is also called as South American blastomycosis or South American disease. So yes, the word is blastomycosis here, but the disease is paracoccidioidomycosis. Correct, paracoccidioidomycosis, right? And if it was coccidioides immitis, then it would have been a big spherule with little sporangia inside. What is that? Coccidioidomycosis. Okay, and what do you see in them? You see the ghost cells. So what did the hot boy do? He dropped you off with his parachute in Brazil, and then he ghosted you. Yes, ghost cells, or he deserted you. So desert valley rheumatism. Okay, all of this is coccidioidomycosis. Sure. Now, let's come on to bacterial food poisoning. Food poisoning, bacterial food poisoning, there are only two important guys. One are the low incubation period or less incubation period guys. What are these less incubation period guys? That means within 6 hours of ingestion, you're going to have the symptoms of food poisoning. Two important guys. Which guys? Of course, Staph aureus and Bacillus cereus. We have just seen one mnemonic pertaining to Bacillus cereus. What was it? It was the Bichara PPT on Mars. So Bacillus cereus shows me swarming motility, and Staph aureus is a gram positive cocci, you know it. So Staph aureus is by ingestion of milk, cream, or cheese products. So milk products or cream ingestion is going to give me Staph aureus food poisoning, classically associated with nausea, vomiting much more as compared to diarrhea. Yes, diarrhea will be much less, but nausea, vomiting is going to be much more because it is going to stimulate the vagus nerve stimulation. And what is the toxin? It is enterotoxin, which is a type of an exotoxin F, and this enterotoxin is a super antigen. Yes, super antigen. What do super antigens do? They non-specifically stimulate the V beta region of the T cell receptors. I hope you remember this. Yes. And then you have Bacillus cereus. This is called as reheated fried rice syndrome. Reheated fried rice syndrome, right? Because when we reheat the rice at 40 degrees Celsius, the spores, they germinate and they lead to release of this enterotoxin. What is common about this also releases the enterotoxin, right? What is common about these two? These two enterotoxins are your preformed toxins. That is what is common and that is what is important. And that is why the incubation period is low, because these are preformed toxins. They're already formed inside the food item. Yes. So that is why these toxins, they're already present inside the food item. So along with the food item, you're not essentially ingesting the bacteria. You're essentially ingesting this toxin. So now that whole period which was required for the bacteria to go and infect your cell and then start releasing toxin, that period is overcome. That is why within 6 hours, within 1 to 3 hours of ingesting the food item, because you have directly ingested a preformed toxin itself, the toxin will straight away go and attack the vagus nerve or attack the enterocytes and attack the cells of your body. So that whole process of bacteria infecting them. Hi, Kanchan. Hello. Hey guys, we are back. We are back. We are back. Are we back? Guys, can you hear me, see me fine? I guess we are back now. Guys, all good? Is it all good? Yes, chalo, thank God. So we are back again. So no matter how hard the cosmic connections are trying today is to not let us complete microbiology, but we are not going to budge. We are going to finish it off, right? Yes, yes, we will be doing toxins. ओके 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 फूड पॉइजनिंग ठीक है चलो देखो मैंने क्या बोला कि व्हेन आई से बैक्टीरियल फूड पॉइजनिंग देन यू हैव टू remember the most important, which is the short incubation period food poisonings, which is the symptoms coming within 6 hours of ingestion of food. So you have two major DDs over there. One is Staph aureus and one is Bacillus cereus. Okay, Staph aureus, you will have a history of ingestion of milk or cream, and over here this is called as reheated fried rice syndrome. Both of them produce enterotoxins. The enterotoxin of Staph aureus attacks the vagus nerve. That is why you have much more nausea, vomiting as compared to diarrhea. And the enterotoxin of Staph aureus is a super antigen. Both of these toxins are preformed toxins. That is why, because they're preformed toxins, the bacteria has already released them into the food item. Okay, so what are you doing essentially when you're eating the food item? You're essentially ingesting the toxin itself. Instead of ingesting the bacteria and the bacteria going and infecting your cell and then releasing the toxin from the cell, all of that gets bypassed and immediately the toxin itself is ingested. So the toxin goes and starts attacking. That is why it is very, very short incubation periods. Okay, Bacillus cereus also produces some diarrheagenic toxins. Okay, so this toxin here is the emetic type toxin. Emetic type toxin. Why? Because this one is going to lead to nausea, vomiting. And then you have Bacillus cereus. What is Bacillus cereus? Then you have the diarrheagenic toxin of Bacillus cereus, right? This is the diarrheagenic toxin. So what is diarrheagenic toxin going to do? Diarrheagenic toxin is going to lead to much more diarrheas. This is going to lead to 16 to 18 hours diarrheas. And what is this associated with? This is associated with chicken, poultry, etc. Right? Chicken, poultry, etc. Okay, so these are certain important food poisoning agents. Then another 16 to 18 hours incubation period, guy, is your Clostridium perfringens. So please remember, Clostridium perfringens not only causes gas gangrene, it also causes your food poisoning, which is also from chicken and poultry ingestion, and 16 to 18 hours diarrhea much more than nausea, vomiting. Okay, diarrhea much more than nausea or vomiting. Okay, now you have this guy. Identify this guy right from the plate. You have golden yellow colored pinhead colonies with a clear zone of beta hemolysis surrounding them. Okay, what is this? This is of course Staphylococcus aureus. Staphylococcus aureus. What is the selective media? So this is the beta hemolysis on blood agar. No problem. What is this? This is the yellow colonies on a pink colored agar. What is this? This is your SMA. SMA. What is SMA? What is SMA? Sorry, SMA or MSA, you can call it anything. This is basically milk. Oh God. This is, yes, Staph aureus, correct. Yeah, this is salt milk. Oh God, this is also glitching now for some reason. Agar. Okay, SMA. Now, what is another important selective media for Staph aureus? Ludlums. Ludlums media, right? What is very, very important to know about Staph aureus? It is MRSA. What is MRSA? Methicillin resistant Staph aureus. And how do we detect it? We detect it by cefoxitin disc diffusion. Cefoxitin disc diffusion. If it is susceptible to cefoxitin, it is MSSA. If it is resistant to cefoxitin, then it is MRSA, which is methicillin resistant Staph aureus, which is because of the change in the change in the penicillin binding protein, where PBP is converted into PBP2A, right? That is MRSA. There can also be VRSA, which is vancomycin resistant. Now, what is it the most important common causes of? MRSA is the most common cause of your acute bacterial endocarditis. What is happening today? Acute bacterial endocarditis. Yes, acute bacterial endocarditis, which is also called as abe. So I always tell that you have to remember abe and sabe, right? So sabe kya hota hai? Abe is very aggressive. It's an abusive word. It's an explosive, aggressive word. So when acute bacterial endocarditis is caused, it is caused by a highly virulent MRSA or Staph aureus, right? Most common cause. If you have sabe, which is sabe ke dil ka veer, meaning Streptococcus viridans, causes subacute bacterial endocarditis. What are the two important clinically differentiating histories that you will get? In Sabe, you will get a history of dental plaques, or you will get a history of brushing the teeth, or you will get a history of damaged valves or IV drug abuser, etc. But in acute bacterial endocarditis, which is Staph aureus, you will get a rapidly progressing endocarditis with no other risk factors. A normal, common, healthy native valve endocarditis. Okay, so acute bacterial endocarditis or native valve bacterial endocarditis is caused by Staph aureus, right? Native valve is involved. Then it is the most common cause of your osteomyelitis. Osteomyelitis. Then you have any abscess. What abscess? There can be breast abscess, psoas abscess, any kind of a localized abscess is caused by Staph aureus. Okay, na? So you will get beta hemolysis, you will get salt meal agar, you will get Ludlam's medium. And of course, if it is Staphylococcus species, it has to be catalase positive and coagulase positive. Coagulase positive. Yes, chalo ji. ये क्या दिख रहा है आपको? What can you see over here? You can see a camp test. Christy Atkin Munch Peterson. Camp test. What happens in camp test? In the middle, you will put a streak of Staph aureus. So can you see this beta hemolytic Staph aureus in the middle? Right, and then you have put one streak here, one streak here. Both of these guys are also beta hemolytic, so these are the beta hemolytic streptococci. You see, on one there is an enhanced arrowhead zone of beta hemolysis. So this was the beta hemolysis of Staph aureus, this was the beta hemolysis of some beta hemolytic streptococci, and suddenly you can see that this beta hemolysis getting enhanced in an arrowhead zone. Over here also there was the beta hemolysis of Staph aureus, and this was the beta hemolysis of some beta hemolytic streptococcus, but you see no enhancement of zone. So this enhancement of zone, arrowhead enhancement of zone, is called as camp positivity. So camp test positive is the one in which there is enhanced, and kaisa enhanced? Arrowhead enhanced zone of beta hemolysis. So which streptococcus? Yes, correct. Which is which streptococcus? What is our mnemonic? The mnemonic is boot camp. Boot camp, meaning group B streptococcus is camp positive. Okay, camp positive. So who is this guy? This guy here is your group B strep, and this guy here is your group A strep. What are the names of these? Group B streptococcus is, B is not B or A is not A. So streptococcus A galacti. A galacti. You know, life would have been much more easier if group A was called as A galacti, but life is not fair, right? So A galacti is group A. Group B. A galacti is group B. Okay. And what is group A strep? Group A strep is Streptococcus pyogenes. What do they cause? They cause B for babies. B is for babies. So group B Streptococcus causes neonatal meningitis. Neonatal meningitis or neonatal sepsis or purpureal sepsis. Whereas what does group A cause? Group A causes gas in the throat, meaning sore throat or pharyngitis. Yes, and it causes non-suppurative complications. What are the non-suppurative or non-pus producing complications? You have post-streptococcal glomerulonephritis, you have acute rheumatic fever, then you have pandas. What is pandas? What is pandas? Pediatric autoimmune neuropsychiatric disorder associated with streptococcus. Yes, these are everything which is caused by group A streptococcus or strep pyogenes. How do you differentiate? Of course, all streptococci are going to be catalase negative. Catalase negative, right? So when you hear the word catalase negative, when you hear camp negative, but you hear bacitracin sensitive, that is your group A streptococcus. So you can see the beta hemolytic streptococci, you can see that this bacitracin disc, there is no zone of inhibition, right? Over here again you see the same thing. So if you put a bacitracin disc and you have to check the sensitivity. What is the mnemonic? Abs. What does abs stand for? Group A is bacitracin sensitive. Okay, group A is bacitracin sensitive. This is abs. So that is how you differentiate between group A and group B. Right now, can you easily write between group A and group B? I mean, you don't have to write, but you have to differentiate. So the two major differentiating factors is, this is strep pyogenes, that is strep agalacti. This one causes sore throat. This one causes B for babies or neonatal meningitis or neonatal sepsis. Over here you have boot camp. So the camp test is actually positive in group B, and you have abs. So bacitracin sensitivity is seen in group A. So this is resistant and this is negative, right? So this is how you differentiate between the two. As a rule, they are going to be catalase negative, right? So when I say streptococci, you have the beta hemolytic strep, which is the group A strep and the group B strep, right? Then you have the alpha hemolytic guys. Amongst the alpha hemolytic guys, if you have GPC in chain, you're dealing with Streptococcus viridans. And what was Streptococcus viridans causing? Subacute bacterial endocarditis, right? There will be a history of dental plaques, history of tooth brushing, etc. And this causes damaged valve endocarditis, damaged valve bacterial endocarditis, right? If it is GPC in pair and it is very much capsulated, मतलब एक blanket के अंदर Netflix and chill, one pair of GPCs is doing Netflix and chill under a blanket. What is that? That is Streptococcus pneumoniae or pneumococcus, right? Streptococcus pneumoniae or pneumococcus. Now you tell me one question. One question is right here. Then you know that native valve endocarditis is Staphylococcus aureus. Damaged or subacute bacterial endocarditis is Streptococcus viridans. Who causes prosthetic valve endocarditis? Come on, chat box. Prosthetic valve endocarditis. Come on, prosthetic valve endocarditis. Who will answer? Nobody? Prosthetic valve endocarditis. See, prosthetic valve endocarditis will happen when the surgeon is opening up the patient and they are putting a prosthetic valve. At the time of opening up the patient, they have not done proper skin asepsis. So the skin flora itself has gone inside and infected the prosthetic valve. Who is a skin commensal? Staphylococcus epidermidis. It is present on the epidermis. So Staphylococcus epidermidis is your guy, which is responsible for prosthetic valve endocarditis. ठीक है, not aureus, epidermidis, correct है ना? Correct. चलो, now you have Streptococcus pneumoniae. Streptococcus pneumoniae is also called as pneumococcus. You know it is capsulated, right? So what are all the features of Streptococcus pneumoniae? That since it is Streptococcus, it is going to be catalase negative, right? Since it is Strep pneumoniae, it will cause meningitis. So it is going to be capsulated. What is the capsule made up of? Everybody's capsule is made up of polysaccharide, except two guys, and we will meet those guys in a little bit. Okay, then So basically, I'll just tell you here, who are the two guys who don't have a sugar capsule? The guys who need a lot of protein. So who needs a protein? A big guy. Who's the biggest bacteria? Bacillus anthracis, big A. So big A and his yar, which is Yersinia pestis, they both need a protein capsule, but the polysaccharide capsule is present in all. So all capsulated organisms have polysaccharide capsule except big A and his yar, which is Bacillus anthracis and Yersinia pestis, right? So of course the capsule of strep pneumoniae is also a polysaccharide capsule, right? Then you have bile solubility. Bile solubility. So basically, if I take a test tube and I take a broth containing strep pneumoniae and I add some bile salts to it, this guy here, there was turbidity. Suddenly you will see all turbidity is lost. So this loss of turbidity is telling me that the colonies have dissolved themselves, right? So this auto dissolution is called as autolysis. So the autolysin enzyme, which is produced by strep pneumoniae, gets activated in the presence of bile salts. So bile solubility test is positive for strep pneumoniae and no other organism. What is another thing? This is a disc of optochin, right? So it is sensitive. You see this zone of inhibition. You can see these green colored alpha hemolytic colonies, and around this disc you can see the zone of inhibition. So what is this? This is optochin sensitivity. Optochin sensitive. Now it produces very characteristic colonies which are called as draftsman colonies or carom coin colonies. Why are they produced? Because of this autolysin only. Because this autolysin will dissolve the top portion of the colony. So the colony will have a central dip. This is a draftsman colony or carom coin colonies. What does strep pneumoniae cause? What is it a significant cause of? So you know your pneumococcus causes mops. What is mops? So you can get pneumonia from mops. Just remember, you can get pneumonia from mops. वो जो झाड़ू पोछा वाला mops होता है. M is meningitis. O is otitis media. Then of course pneumonia. The name itself says pneumonia, and you have sinusitis. So strep pneumoniae causes mops. Another important test which can be performed is called as the Quellung reaction. Quellung reaction. What is Quellung reaction? Quellung reaction is swelling reaction. So Quellung reaction will show you swelling. So this is a gram-positive cocaine capsule. Now you add an anti-capsular anti-serum. Now this capsule is going to swell up. So Quellung reaction is equal to swelling or swelling reaction on adding anti-serum. Right? Correct. Chalo. So pneumococcus causes mops. Pneumococcus is streptococcus. So it is catalase negative. It is capsulated. It is bile soluble, optochin sensitive, and shows me the draftsman colonies as well as the Quellung reaction. What is happening here? You see these are all neutrophils. You can see the multiple lobe neutrophils. Inside these neutrophils, can you appreciate the small little red colored dots? What are these? These red colored dots are your Neisseria. What is Neisseria? What is Neisseria or Neisseria? It's a gram-negative diplococcus. Gram-negative cocci present in pairs, right? Gram-negative diplococcus. There are two types of Neisseria. Neisseria meningitidis and Neisseria gonorrhoeae. Tell me, which one is going to be capsulated? Of course, the one which is going to cause infection in the khopri, right? So meningitis is the one which is going to cause infection in the khopri. So this is the one which is going to have capsule, right? Capsule. Whereas this guy is non-capsulated, right? Then M for meningitis, M for maltose. So this is going to be maltose fermenter, whereas this is going to be a non-fermenter for maltose. Okay. Meningitis obviously causes meningitis, and gonorrhea causes gonorrhea, right? Which is the most important virulence factor. Which of them is the kidney shaped? Gonorrhea is what is kidney shaped, right? Whereas meningitis is just simple diplococci with a capsule all around. Okay, this guy is kidney shaped, right? What are the complications of Neisseria gonorrhoeae or gonorrhea called as water can perineum? Water can perineum. So if males are infected, it is going to involve the whole of perineum, and there will be small little holes, and from that the pus will shed out. So it will look like a water can which is used to water the plants. Now it has these small round thing and all of these holes from which holes the water comes out. So this is what the perineum of the guy is also looking like a water can. Then so this happens in the males. What happens in the females? It's Fitz-Hugh-Curtis syndrome. Fitz-Hugh-Curtis syndrome, it means nothing. It also means now the whole pelvis and perineum is involved inside the females, right? Now, what is the complication of meningitis by Neisseria meningitis called as? It is Waterhouse Friderichian syndrome. Please don't confuse water can perineum and Waterhouse Friderichian syndrome. Okay, na? So now what is the feature of this Waterhouse Friderichian syndrome? You are going to have, gangrenous purpuric rash. There is going to be gangrenous purpuric rash. That is why it is also called as meningococcemia. Now the bacteria is in the blood, meningococcemia, right? This is very, very fatal. This is very highly dangerous. And whenever you have any meningitis or any history, and they are saying that there is a purpuric rash or there is a petechial rash or there is any kind of hemorrhage involved, you know that they're talking about Neisseria. Okay, in the previous FMG questions, they had given one autopsy of a brain and said that this person was having recurrent meningitis, and on the brain autopsy, we can see many hemorrhagic rashes or petechiae. That also becomes Neisseria. So whether it is any kind of meningitis, any kind of gonorrhea, and association is with a petechial rash or purpuric rash, you know we are talking about Neisseria. Okay, it causes a bad kind of a meningitis. So what is the treatment of choice for both of them? The treatment of choice always is either third generation cephalosporins or ceftriaxone, right? Ceftriaxone. Either they have written ceftriaxone there or they have written third generation cephalosporins. It means the same thing. Okay, ceftriaxone or third generation cephalosporins is the treatment of choice. Then you come to phage coded toxins. Phage coded toxins. So what does this mean? So every bacteria has the ability to produce toxins. How does one bacteria transfer the ability to produce toxin to other bacteria? Through plasmid transfer. Okay, but some of these bacteria, they get infected with phages, and these phages have the ability to produce a toxin. Now this phage has given its own chromosome to the bacteria, and now bacteria has the ability to produce toxin. What is this called as? This is called as lysogenic conversion. Guys, please don't confuse this. Okay, many of you tend to think that the generalized transduction is the reason for phage coded toxin. No, in any kind of transduction, first let me write lysogenic conversion. In any kind of transduction, the phage is not giving its own DNA to the bacteria. Phage is bringing the DNA of another bacteria and giving it to this bacteria. It can bring a whole DNA in generalized transduction. It can bring a small piece of DNA in specialized transduction. But what happens in lysogenic conversion? The phage is converting the bacteria. So phage has the ability to produce toxin. It is converting a bacteria that now the bacteria can produce the toxin. So what are the examples of these phage coded toxins? A, B, C, D, E. Right? A, B, C, D, E. What is A? Actually, A is the worst out of all of this mnemonic. Streptococcal pyrogenic exotoxin A and C. Streptococcal pyrogenic exotoxin A and C, right? This is produced by a group A Streptococcus. What does this cause? This causes scarlet fever. Scarlet fever. Okay. Then you have B for botulinum toxin. Botulinum toxin. Then you have cholera toxin. Cholera toxin. Then you have diphtheria toxin. Diphtheria toxin. And E is what? Enterohemorrhagic E. coli O157H7, which produces the verocytotoxin. Verocytotoxin. Okay. Or VCTX. Verocytotoxin, which leads to hemolytic uremic syndrome. Hemolytic uremic syndrome. Okay, HUS, correct. right? Now, what is this then? What is this? Can you see the Albert stain and can you see the metachromatic granules? And over here on the KTB agar, can you see the black colored colonies? Of course, we are dealing with Corynebacterium diphtheria. We are dealing with Corynebacterium. Corynebacterium diphtheria. Okay, so how to remember all the features of Corynebacterium diphtheria? Again, you start writing A, B, C, D. A, B, C, D, E, F, G. A, B, C, D, E, F, G. What does A stand for? A is Albert stain. So what is the stain that we are using? It is Albert stain. Albert stain. What is B? B tells you bull's neck appearance. The bull's neck appearance of the patient. Then C is the cuneiform pattern arrangement or the coryne. This is the cuneiform pattern arrangement or the Chinese letter pattern arrangement or the V-shaped pattern arrangement of the bacteria. What is D telling me? D is telling me that it produces diphtheria toxin, which is a phage-coated toxin and it's a toxin-mediated disease. So what is the mechanism of action of diphtheria toxin? It is EF. What is EF? It inhibits elongation factor 2. Inhibition of elongation factor 2 is what is EF. Okay, what is G? G stands for granules. So what granules is it showing me? Volutin granules, Babes Ernst granules, or your metachromatic granules. Okay, metachromatic granules. What is the agar which is used? KTBA, potassium tellurite blood agar, which is the selective medium. Selective medium. And you have LSS, Loeffler's serum slope, which is the enriched medium. Enriched medium used for Corynebacterium diphtheria. LSS, Loeffler's serum slope. All right. So quickly tell me all of these toxins and their mechanism of action. So we have just seen diphtheria toxin is produced by C. diphtheria. Yes, what is the mechanism of action? It is inhibition of elongation factor 2. Who will tell me what is the mechanism of detection of this toxin? It is called as Alex gel precipitation test. Alex gel precipitation test. All right, which is done for Corynebacterium diphtheria to detect this diphtheria toxin. Okay, now other guys, exotoxin A. This exotoxin A is produced by Pseudomonas aeruginosa. Pseudomonas aeruginosa. And what is the mechanism of action? Same, inhibition of elongation factor 2. What is happening as a result of this? As a result, there is inhibition of protein synthesis by this bacterium, right? By the cells of our body. So the inhibition of protein synthesis. Now, tetanus. Tetanus toxin. What is the mechanism of action of tetanus toxin? It leads to tetany. Tetany means contraction. So it leads to a continuous state of spasm. So it is going to inhibit the, so of course it is produced by Clostridium tetany. And what is Clostridium tetany going to do with this toxin? It is going to inhibit the GABA glycine, which is the inhibitory neurotransmitter. Basically, there is excitatory stimulation by ACH. The muscle goes into contraction. Then GABA glycine comes, says stop contracting and relax. Now this stop the contracting and relaxation signal has been stopped by Clostridium tetany. So there is always a continuous or constant spasm or tonicity. Tonicity, that is what is tetanus, right? So botulinum toxin is complete opposite. It leads to flaccid baby syndrome or floppy baby syndrome. So there is flaccid paralysis. So for that, the Clostridium botulinum will produce botulinum toxin, which will inhibit acetylcholine at the neuromuscular junction. So there will be a floppy child syndrome or flaccid paralysis. Right here, there is spastic paralysis in tetany and there is flaccid paralysis in your botulinum. Okay. LTST क्या है? LTST are both released by E. coli. Which E. coli? Enterotoxigenic E. coli. So T for this causes traveler's diarrhea. Traveler's diarrhea. What is the mechanism of action? So labile toxin leads to increased CAMP. And stable toxin leads to increased CGMP, which is the receptor on which both of these are binding. Both of these are binding to the GM1 ganglioside receptors on the enterocytes or inside the cells of the intestine. Right? Then what is STX? STX is Shiga-like toxin. Shiga-like toxin, also called as verocytotoxin, produced by enterohemorrhagic E. coli O157H7. What does it do? It inhibits the 60S ribosome subunit. Inhibition of 60S ribosome is also going to lead to inhibition of protein synthesis, right? This will also inhibit protein synthesis. Cholera toxin. What does cholera toxin do? COT acts on the ZOT. Yes, zonula occludens toxin. Now, it also increases the CAMP just like the labile toxin, and it also acts on the GM1 ganglioside receptors just like the labile toxin. So, the mechanism of action of cholera toxin is exactly the same as that of the enterotoxigenic E. coli, which causes traveler's diarrhea, but it's just that the cholera toxin is a much more potent toxin. So the effect that it produces is much more worse and much more enhanced effect, right? Then you have alpha toxin. Who produces alpha toxin? Alpha toxin is produced by your Clostridium perfringens. Right? And Clostridium perfringens produces something with the alpha toxin, which is lecithinase production. Lecithinase production. So how do we test it? We test it by the Nagler reaction. By the Nagler reaction. All right. So this is all the important mechanisms of actions and all the important toxins. Ye kaun hai? Who is he? This is your continuous grain sign. This is of course Clostridium tetani causing tetanus. What is this sign called as? Rhizus sardonicus. Yes, Rhizus sardonicus is a complication of tetanus, and along with that there is opisthotonus, which is the bow-like opposite kind of a direction of the person who's gotten infected, right? So Clostridium tetani is your guy who's causing this disease. It is of course an obligate anaerobe. What does it look like? It shows me the drumstick appearance. Drumstick appearance. Why does it show me this drumstick appearance? Because it has this bulging terminal round spore. So this is the bacteria, and on the end of the bacteria there is a bulging spore which is totally round, right? So all the Clostridium they produce, B for bulging spores, right? So it has a bulging, T for terminal, and it is round. And these are endospores because they are formed inside the body of the bacillus. So bulging terminal round endospores giving me the drumstick appearance. Rhizus sardonicus and opisthotonus. All right, this is Clostridium tetani. Now what else does Clostridium tetani show me biochemically? It for all anaerobes, what is the culture media? RCM, Robertson cooked meat broth. On the Robertson cooked meat broth, it turns the Robertson cooked meat broth black. So because of turning black, it is giving me the proteolytic reaction. Proteolytic reaction. Okay, then they ask you about the basics of vaccination. So the human anti-tetanus immunoglobulin or the human immunoglobulin against the tetanus is only and only given in case of deep contaminated wound, and deep contaminated wound plus the history of vaccination is more than 5 years old. Okay, so if you have taken the vaccine in the past 5 years, there is no need to give any immunoglobulin. Okay, but if the vaccination is more than 5 years old, especially if more than 10 years old, then definitely you need to take immunoglobulin in case there is a deep contaminated dirty wound. Okay, contaminated and dirty wound is what is the most essential. Then you have this. What can you see here? You can see that this is an agar plate which has been divided into two halves. On this one half you can see opacity, on this one half you can see opacity not present. So here opacity is there, here there is no opacity. You know which one of the two is a positive test? This absence of opacity is called as the positive test. Okay, how? Why? Because this is something what we call as the Naegler reaction, and we have just seen that Naegler reaction is a reaction done to see the presence of toxin produced by toxin of Clostridium perfringens to detect the toxin of Clostridium perfringens, right? Naegler reaction. So what do? We divide the plate into two halves. We on the one half we have cultured Clostridium perfringens. Clostridium perfringens shows me the opacity. Why? Because Clostridium perfringens releases alpha toxin on egg yolk agar. What is this agar we are using? Egg yolk agar. Clostridium perfringens on this egg yolk, yes, not angular reaction, Naegler reaction, right? On this egg yolk agar, it produces opacity. Opacity is produced by it. Okay. Now, what will I do? I will add some antitoxin. On the other side of the plate, I will add antitoxin. So over here, I have added the antitoxin. And which antitoxin have I added? I have added the anti-alpha antitoxin. Which antitoxin? Anti-alpha antitoxin. So now this alpha toxin will not be produced, so this opacity will be lost. Okay, so there is lost opacity. So this lost opacity is telling me that yes, in fact there was a toxin produced which was leading to opacity, and now that I have added antitoxin, the toxin is not able to work and opacity is not going to be there. That is why this no opacity side where I have added the antitoxin is actually the side of positive reaction of Naegler reaction. ठीक है, ये क्या है आपका? It's like a storm is brewing inside a test tube. This is called as stormy clot reaction. Stormy clot reaction, or also called as litmus milk test. Litmus milk test. Everything is shown by Clostridium perfringens itself. Okay, Clostridium perfringens. Now we talk about Bacillus anthracis. Who is Bacillus anthracis? He is called as Big A. So Big A and his yar. Big A and his yar are gym buddies. So if they are gym buddies, they have protein capsules. Protein capsules. Okay. So who are these? Bacillus anthracis and Yersinia pestis. These are the only two guys which have a protein capsule. What is the protein capsule made up of? Poly-D-glutamate. What is it made up of? Poly-D-glutamate. Poly-D-glutamate. All right. So, what does Bacillus anthracis cause? It causes anthrax. There are two types of anthrax. One is called as the wool-sorter's disease. What is wool-sorter's disease? It is pulmonary anthrax. Pulmonary anthrax. And then you have Hyde-Porter's disease. Hyde-Porter's disease. What is Hyde-Porter's disease? It is cutaneous anthrax. Cutaneous anthrax. Okay. So now pulmonary anthrax or wool-sorter's disease, this is also a bioterrorism category A agent. Okay. Bioterrorism category A is pulmonary anthrax or wool-sorter's disease, right? What are you going to see in Hyde-Porter's or cutaneous? You're going to see an eschar. What is an eschar? An eschar is a malignant pustule. Malignant pustule. Malignant pustule. What is happening? right? Malignant pustule. So you have edematous black colored necrotic eschar surrounded by a red erythematous base, right? So the next question that they ask you, what are this was asked in INICT. You have PX01 and PX02. What are these? These are some important plasmids produced by Bacillus anthracis, right? Plasmids. And what are they coding for? So PX01 is coding for the toxin, and PX02 is coding for the capsule. What is the toxin made up of? Toxin has got three components. It has a binding unit, right? It has a lethal factor, and it has something called as the edema factor. So lethal factor is going to cause cytotoxicity, edema factor is going to cause edema, and binding factor is going to bind to the cells. Okay, so along तीनों मिलके they are going to lead to formation of malignant pustule or eschar, which is Bacillus anthracis. What will you have? You will have a history of cattle exposure, somehow. Either he's a farmer, either he's a cattle rearer, or he's a milkman. Any of these kind of cattle exposures can be this. So sometimes they don't give you the specific wool sorter or hide porter words. Okay, sometimes they just give you history of cattle exposure, and then they ask you, yes, binding unit, lethal unit, edematous unit, or edema factor. Excellent. What is happening here? What can you see? Is this TCBS or is this CLED? This is actually CLED. What is CLED? Cysteine lactose electrolyte deficient agar. This is specifically used for detection of uropathogens or urinary pathogens. So everything grows very nicely on this guy. Even your GPCs, your GNBs, your Candida, everything grows very nicely on your clade agar. That is why it is specifically used for urine. So what is it? On this, the yellow colonies are actually your lactose fermenters, and the green or the pale colonies are your non-lactose fermenters, because lactose has been added to make it differential. So what is the your indicator used here? It is bromothymol blue, or bromothymol green. Yes, because on acidic pH, they turn yellow. So if it is a lactose fermenter, they will turn yellow on clade agar, right? What is the advantage? That this is economical and all the uropathogens can grow. So what is the concept which is used? It is called as the CAS concept. This CAS concept is reserved for urine cultures. Over here, you have to not just find out whether bacteria is present in the urine. You have to find out whether that bacteriuria is in fact significant. So whether it is significant bacteriuria. And what is significant bacteriuria? More than 10 raised to power 5 CFU per ml of bacteria present. Right? What is the only exception where you get rid of the CAS concept, where you don't believe in CAS concept? It is pregnancy. Yes, pregnancy, infancy, and in case MTB is suspected, because MTB is anyways not going to grow on your clade agar. Right? Then you have Enterobacteriaceae or the Enterobacteriales family. You have these certain set of biochemicals in front of you. Indole MRVP citrate, right? And I will also write urease over here, right? Urease is the pink colored agar. So if it is pink, it is positive. If it is yellow, it is negative. So you can see pink is positive and red is positive in all except citrate, in which blue is positive, right? In which blue is positive. So how to remember? Pink is positive and pink and red are similar looking, so they are negative. So in urease also positive is pink and negative is yellow. Okay, so how to remember? Just write two guys here. One is E. coli, one is Klebsiella pneumoniae, and then you write IMVICU. IMVQ. Okay, IMVQ. How to remember? E. coli is plus plus minus minus minus, and Klebsiella is minus minus plus plus plus. So Klebsiella is going to be urease positive, Klebsiella is going to be citrate positive, where E. coli is going to be indole positive, citrate negative, and urease negative. And this is going to be indole negative. Okay, this is the classical points about it. Then you have another guy whose name is Klebsiella oxytoca. ऑक्सीटोका ने क्या किया? इंडोल को टोका, इसने इंडोल को टोक दिया. सो ओनली इंडोल इज पॉजिटिव हियर. Rest everything looks exactly like Klebsiella pneumoniae. Okay, so the rest everything looks exactly like Klebsiella pneumoniae, just that indole is positive with Klebsiella oxytoca. Right? Then you have the diarrheagenic E. coli. कौन कौन से होते हैं? Enterotoxigenic E. coli, enteropathogenic E. coli, enteroaggregative E. coli, diffusely adherent E. coli, enterohemorrhagic E. coli, and enteroinvasive E. coli, right? So P stands for pediatric. So this is going to lead to pediatric diarrhea or infantile diarrhea, and it does not cause any destructive lesions. The bacteria simply sits on top of those the microvilli, and it causes effacement, attachment and effacement kind of lesion. Okay, whereas ETEC, T for traveler's diarrhea, it will be because of either the labile toxin or the stable toxin produced by the bacterium, right? This causes traveler's diarrhea. Then you have EAEC, is your enteroaggregative. Here you will have stack of bricks like arrangement of E. coli on top of the brush border epithelium. In diffusely adherent, it is going to diffusely adhere to the brush border epithelium. What about enterohemorrhagic E. coli? Enterohemorrhagic E. coli is O157H7. O157H7. It causes hemolytic uremic syndrome as a side effect, as a prolonged complication, right? But what does it cause acutely? Acutely, it causes dysentery, which is blood in stool. Enteroinvasive is also like Shigella, like dysentery. This guy also causes dysentery, right? What is the toxin of it? Verocytotoxin or Shiga-like toxin, the mechanism of which we have seen already, right? Okay, LT increases CAMP and ST increases CGMP. I hope you know this. Okay, done. Now you have this. Again, this looks like CLED, but this is not CLED. What is it? This is TCBS. What is TCBS? Thiosulfate, citrate, bile salt, sucrose, agar. Thiosulfate, citrate, bile salt, sucrose, agar. Can you see the yellow colonies? They are specifically produced by Vibrio cholerae, whereas the green cholerae are produced by Vibrio parahemolyticus. So sucrose is fermented by Vibrio cholerae. That is why you see the yellow colonies of Vibrio cholerae on TCBS. TCBS is the selective medium. It's the selective agar or the selective medium for Vibrio cholerae. For Vibrio cholerae. All right. चलो ये देखो, अब you amongst Vibrio cholerae you have two types of biotypes. You have biotypes and your serotypes. Biotypes are two, classical and LTOR. Biotypes. So how to remember LTOR? Just remember Thor. Okay, LTOR is equal to Thor. So who is Thor? Thor is very, very, highly strong. He's very resistant and he's very strong. So what is Thor or LTOR going to do? He's going to cause a lot of destruction. So you show him to the sheep blood agar, it is going to destroy them by beta hemolysis. You show them to chick erythrocytes, he's going to destroy and agglutinate them. You show him polymyxin B, he's not going to be scared, he's going to be resistant. You show him another bacteriophage, he's going to be totally resistant to that bacteriophage. You show him VP test, he's going to come out positive. You show him CAMP test, he's going to come out positive, right? So everything is positive and resistant when we are talking about Thor or LTOR. right? It will destroy the RBCs, it will cause beta hemolysis, it is going to not be affected by a phage, it is going to not be affected by polymyxin B, camp VP, everything is going to be positive. The only thing that it is susceptible to is to a phage जिसके नाम में ही है L-tor phage. So the property of this phage is that it eats L-tor. So other than this L-tor phage, everything else is going to be positive and resistant when we are talking about the L-tor biotype. And everything else going to be negative and susceptible for classical, except of course the L-tor phage, which is not be able to eat up the classical biotype of Vibrio cholerae, right? When we talk about toxins, so the classical one came first, so it's cholera toxin 1, and this is CTX2, which are the genes for production of cholera toxin of L-tor and classical biotypes. Okay? Yes, correct. Thor, right? L-tor is Thor. ये हो गया. Now what about some halophilic vibrios? You know, Vibrio cholerae shows you darting motility. Vibrio cholerae is not halophilic. I mean, it can only tolerate up to 6% of NaCl. But your halophilic vibrios, they are salt loving vibrios. And where is salty water seen? It is seen in the seas, which is the most beautiful ocean or sea vacation spot in India, Andaman. So what is our mnemonic? People vacation in Andaman. People vacation in Andaman, right? So P stands for Vibrio parahemolyticus. Vibrio parahemolyticus. V stands for Vibrio vulnificus. And A stands for Vibrio alginolyticus. Now, why have we remembered in this particular order only? Because this is the increasing order of salt tolerance. This is going to be 7% NaCl tolerance, 8% NaCl tolerance, and 10% NaCl tolerance. So the most salt tolerant out of all is Andaman or alginolyticus, which is going to tolerate 10% of NaCl. What does Vibrio parahemolyticus cause? It causes seafood dysentery. So there is going to be a history of ingestion of seafood or oysters, and this is going to lead to dysentery. Now you remember, it was a part of the mnemonic of Bichara PPT on Mars. So this guy is going to show me swarming motility, right? Then it is going to show me green colonies on TCBS agar. We have just seen it is going to show me green colonies on TCBS agar, on TCBS. Green colonies on TCBS. Vulnificus is called as the wound maker. Wound maker. So you see the very bad wounds, skin and soft tissue infections from exposure to salty and seawater. So wound maker, you went ocean dipping, you went sea dipping, and you got very bad skin and soft tissue wounds. That is Vibrio vulnificus. Alginolyticus, on the other hand, which is the most salt tolerant, it causes your swimmer's ear, right? It causes swimmer's ear, and it also causes conjunctivitis, right? Then you have some phenomenon which is called as Kanagawa phenomenon. Kanagawa phenomenon on Vagatsuma agar. Right? Kanagawa phenomenon on Vagatsuma agar. What is this? This is nothing but Kanagawa phenomenon means beta hemolysis. And Vagatsuma agar means alkaline blood agar. Right? So this particular bacteria shows beta hemolysis on an alkaline blood agar. Who is he? He is Vibrio parahemolyticus. Right? Vibrio parahemolyticus. Pen is also crashing now. See what all is going to happen today. This marathon is like a pure test from God today, right? So only the ones who are the strongest are going to stay back and they are going to continue. Now the pen has started crashing. Before this, the iPad was crashing, then the laptop crashed, then the internet crashed. No problem. But we are not the ones who are going to crash. Okay, chal. So now let's talk about spirochetes. Who are spirochetes? These are your spiral bacteria, the thin spiral bacteria which are spirochetes. All right. Just a minute, guys. Yeah, okay. Spirochetes, you have three important spirochetes. Who are they? They are called as boring triplets. Oh God. Boring triplets, right? Boring means Borrelia. Yeah. Boring means Borrelia. Okay. And triplets means treponema. Treponema. And lets means leptospira. Leptospira. All right. So, Borrelia can be of two types. One is Borrelia recurrentis. And you know, recurrentis means to recur again and again. So that is Borrelia recurrentis causes relapsing fever. Relapsing fever. And relapsing fever can be endemic relapsing fever or epidemic relapsing fever. Right? So how to remember endemic and epidemic relapsing fever? And so every time you hear the word epidemic, you know you're talking about louse. Lice is always associated with the word epidemic. So epidemic typhus, epidemic relapsing fever, it all pertains to louse. So over here, this is caused by louse is the vector, and for endemic relapsing fever, it is the tick. Tick is the vector. All right? Yes. Then from Borrelia, you also have Borrelia burgdorferi. Burgdorferi. So BB. From BB, you remember Justin Bieber. Justin Bieber had gotten the Lyme disease. Lyme disease. Okay. What all do you see in Lyme disease? This is also by exodistic. Exodistic. So how do we remember? With exodistic, he got real sick. Okay. So from exodistic, he got real sick. And what are the features of Lyme disease? Just imagine that somebody is squeezing a lime. So when they squeeze the lime, this spiral Borrelia is coming out and it is hitting different targets. So when you squeeze the lime goes into your eye, on your face. It causes facial palsy, right? It goes and hits your bones, leading to chronic arthritis, and that comes and hits your skin like a bullseye. So that is bullseye rash. Bullseye rash. Okay, then you have another one which is Borrelia vincenti, which causes Vincent's angina. Okay, then you have third one which is Borrelia vincenti, which causes Vincent's angina, which is actually polymicrobial. There are many anaerobes also involved. That is Vincent's angina, right? Then you have this case. A 28-year-old man presents with painless ulcer on his genitalia. There is no history of trauma. On examination, ulcer has indurated edges and a clean base. What is this? This is a hard chancre, right? Their regional lymphadenopathy is present, but it is non-tender. What is the most likely diagnosis? Yes, this is not chancroid, because chancroid bleeds on touch. And that is not hard and indurated. That is a soft chancre. That is a soft chancre. Okay. Herpes genitalis, no. There are multiple vesicular painful ulcers. It is, of course, primary syphilis. It is not lymphogranuloma venereum, because in lymphogranuloma venereum, you see the groove sign. Groove sign. So, who causes primary syphilis? Treponema pallidum. Treponema pallidum causes primary syphilis. So that brings us to genital ulcers. You have to classify your genital ulcers into two ways. Either the ulcer itself is painless, right? Or the ulcer itself is painful. So you're talking about the painless or painful nature of the ulcer. If the ulcer is painless, then you have to look at the lymph nodes. Either the lymph nodes are also painless, or the lymph nodes are very much painful. Okay, either lymph nodes are also painless or the lymph nodes are painful. So if they are painless lymph nodes, then there are two possibilities, meaning dono hi painless hai. So one is the prototype disease, which is syphilis, in which you have the primary, secondary, tertiary syphilis, and you have the hard indurated chancre. The second is, सी दोनों ही नहीं है ना, देयर आर दोनों नहीं, ना तो अल्सर है ना तो लिम्फ नोड है, इट इज दोनों नहीं. सो दोनों नहीं टेल्स यू दोनों वनी. दोनों नहीं टेल्स यू दोनों वनी. Donovani. What is Donovani telling me? That this is donovanosis. Donovanosis. And who causes donovanosis? It is Klebsiella granulomatis. Granulomatis causes donovanosis, in which you see the Donovan bodies. Donovan bodies. What are Donovan bodies? These are the safety pin or bipolar staining appearance wala intracellular bodies inside the macrophages, and it has the sound of a D. So what is D telling me? That this is going to bleed. Bleed means that this is beefy. This is beefy red ulcer. Beefy red ulcer. Okay, this is donovanosis. Then you have painful lymph node and painless ulcer. मतलब ulcer पे कोई focus ही नहीं है. The entire focus has been shifted to the lymph node. So when the lymph node is the most important feature, it is lymphogranuloma venereum. Lymphogranuloma venereum. How many letters are there? 3. L1, L2, L3. This is caused by chlamydia, trachomatis, L1, L2, L3. Okay, and G for groove sign. Groove sign is what you see over here. Groove sign. Okay. Then you have the painful ulcers. Now the painful ulcer can be further into two divisions. Either painful ulcer and plus painful lymph node. Even the, if the ulcer is painful, then even the lymph node is going to be painful. The lymph node is also going to be painful, right? So if it is a painful lymph node and a painful ulcer, either it can be unilateral or it can be bilateral. Either it can be unilateral or it can be bilateral. If it is unilateral, it is one-sided. So everything which will have the O sound, chancroid will be unilateral bubo. There is going to be unilateral bubo, which is the painful lymph node. Okay, and there is also the D sound. So this is going to bleed on touch. And what all D sound? Haemophilus ducrei. Haemophilus ducrei. Then you have bilateral. So you have two words, HSV2, which is genital herpes. Genital herpes. There is going to be multiple painful, vesicular, bilateral ulcers. All right, that is genital ulcers. Now you have certain other species of treponema which cause important skin and soft tissue infections or systemic infections which are not venereal diseases. So you have certain non-venereal treponems. Non-venereal treponems. What are they? You have treponema pallidum, which causes syphilis. Then you have treponema pallidum endemicum. Name itself is telling you that this is going to cause endemic syphilis. Yeah. Then you have treponema keratium. So what do you have to remember? Now, by this time, so much of glitches happening and you've dedicated the whole day to microbiology today and still nothing seems to get done. You will need a crate full of pints. Yes. So treponema keratium is going to cause pinta. Pinta. So just remember, a carrot of pints is Treponema keratium is causing pinta. Then you have the other remaining guy, which is Treponema pertuni that causes yaws. Yaws. Okay, important ones done. What is this? Can you appreciate the fried egg colonies? And these are microscopic fried egg colonies. So they are mycoplasma. Mycoplasma, which shows fried egg colonies, and they are microscopic. How small are they? They are 300 micrometers is the size of the colonies. Okay, is the size of the colonies. So mycoplasma causes what? Mycoplasma causes atypical pneumonia. Atypical pneumonia. So what is atypical pneumonia? It is walking talking pneumonia. Actually, you have a patient coming to your OPD walking talking normally, and you get a radiography done for him, you get an x-ray done for him, you realize that okay, this guy is walking talking, but the radiological imaging is far worse than the patient symptom. So you are going to get very very angry कि यार ये क्या बात हुई, तुम तो चलते फिरते आए हो, but you have got pneumonia. So what will you do for this patient? You will do a medico-legal case. You will do an MLC. What is MLC? MLC is for atypical pneumonia. That is Mycoplasma pneumoniae, Legionella pneumophila, and you remember Legionella pneumophila, Legionella, AC vents, outbreaks. So whenever you hear AC vents, outbreaks, that is Legionella pneumophila. And then you have Chlamydia pneumoniae, right? What do you have to remember? Mycoplasma, this shows this kind of colonies on the PPLO agar, pleuropneumonia like organism, or the PPLO agar showing me the fried egg colonies called as mycoplasma. Then Legionella pneumophila also grows on a black colored agar which is called as BCYE, buffered charcoal yeast extract agar. It grows on the BCYE agar, and it shows me a very specific phenomenon in the pathogenesis which is called as coiling phagocytosis. Coiling phagocytosis. So normally how does phagocytosis happen? The bacteria comes and the phagocyte just engulfs it like this. Okay, but what does Legionella do? He's quite a twisted guy. He takes up the antigen and coils around and phagocytosis. So this type of coiling is called as coiling phagocytosis. Okay, ये देखो क्या कह रहा है. 10-year-old child from Dehradun with a 5-day history of fever, abdominal pain and vomiting. On examination you are seeing S-char. Lab investigations with thrombocytopenia, elevated liver enzymes. What is it? It is, come on, tell me. What is this? You can see an S-char and you can see Dehradun. So Dehradun is telling me about scrubby vegetations. Scrubby, yes, this is scrub typhus. Scrub typhus is caused by Orientia shushugamushi. Orientia shushugamushi. Okay, so what about the other guys? We have to now talk about the Rickettsial railway station, right? What happens on this Rickettsial railway station? All the Rickettsial group is here. So you have two trains, a typhus train and a spotted fever express. Of course, on the spotted fever express, you have Rocky Mountain spotted fever causing Rocky. Yes, and Rocky is spread through Rickettsia rickettsii. So what is our mnemonic to remember this? The mnemonic is Rick. Okay. Rick mountain tick. Rick mountain tick. So what do I remember from this? I remember that Rocky Mountain spotted fever is tick-borne. Tick-borne. There is the derma center tick, and it is caused by Rickettsia rickettsii. Rickettsia rickettsii causes it, and it in this you are going to see the rashes on palms and sole. There is going to be rashes on palm and soles. Yes, rashes on palm and soles. Okay. Now, along with him, who has gotten on? This Indian prince. He's an Indian crown prince. So, Conori, Rickettsia conori causes Indian tick typhus. So, Indian tick typhus is obviously also going to be tick transmitted. So, you have two spotted fever expresses, which is Rocky Mountain spotted fever and Indian tick typhus. So, Rick mountain tick is showing you the Rickettsia rickettsii causing Rocky Mountain spotted fever, which is tick-borne. Along with that, there's the Indian crown prince. So Rickettsia conori causing Indian tick typhus, which is also tick transmitted. Another special feature of this Indian tick typhus is that even in this also you are going to see the black colored eschar. So eschar can be seen in scrub typhus, eschar can be seen in cutaneous anthrax, and eschar can be seen in Indian tick typhus. All right. Then you have the typhus strain. इस typhus strain पे देखो कौन कौन है. There is this soldier. This soldier is pro-war. Pro-war. So it is Rickettsia provazicae. Rickettsia provazicae. And he has louse. Whenever we talk about soldiers, whenever we talk about outbreaks, whenever we talk about epidemics, whenever we talk about soldiers living inside trenches, we always talk about lice, because lice happens in overcrowded spaces, right? So this louse is causing the Rickettsia, is carrying the Rickettsia provazicae, which is causing the epidemic typhus, because wherever there is louse, there is epidemic. Okay, and this soldier is a pro-war soldier. He's a pro-war soldier. So Rickettsia provazicae causes epidemic typhus. Along with him, who has gotten it? This rat. You see, this rat is totally infested with fleas. Still, he's smiling. So he's a tough rat, right? He's quite a tough rat. So tough reminds me typhi. And of course, the only other one is endemic typhus. So that is why both of them have gotten on the same train. So I can remember I'm talking about the typhus group, because it's the typhus train. So you have two types of typhus, epidemic typhus and endemic typhus. Epidemic will be caused by louse. Who has the louse? The soldiers, and soldiers are pro-war. So it Rickettsia provazicae. Along with that soldier, the mouse or the rat had got on. Rat was infested with rat flea. So he was quite tough. So Rickettsia typhi causes endemic typhus, which is spread by the rat flea. All right, so now you can easily remember which are the tick-borne rickettsial fevers. It is on the spotted fever express, RMSF and ITT. RMSF is Rick mountain tick, so tick-borne Rocky Mountain spotted fever causing rashes on the palms and sole caused by Rickettsia rickettsii. Then the crown prince is Indian, so Indian tick typhus also spread by the tick by Rickettsia conori or crown princess conori, and it causes eschar. Then on the typhus strain, you have epidemic typhus and endemic typhus. Whenever I say epidemic, I say lice. When I say lice, I say soldiers. Soldiers are pro-war. So epidemic typhus is louse-borne, spread by Rickettsia provazicae. And the other guy was a tough rat with the rat flea. So rat flea-borne Rickettsia typhi causes endemic typhus. Okay, ये हो गया पूरा. This is all the rickettsial group. Then you have the scrub typhus. Scrub typhus by Orientia shushugamushi is also called as chigerosis. Chigerosis. Why? Because it is spread through the bite of chigger mite. Chigger mite. So what is chigger mite? It's the badmash baby of the mite. Okay, it's the larva. It's not the adult mite who bites. It's the chigger who bites. So you need scrubby vegetations. The presence of scrubby vegetations is very, very important. And what is the classical triad of this? You get maculopapular rashes, maculopapular rash, plus you get an eschar, plus you get the lymphadenopathy, right? That is scrub typhus or chigerosis caused by Orientia shushugamushi. Now you have a test which is called as the Weil-Felix reaction. Weil-Felix reaction. Yes. So Weil-Felix reaction is shown by the rickettsial group. It's a heterophile antigen test. Heterophile antigen test, right? This heterophile antigen test is shown by whom? So it is basically the antigens of rickettsial group and antigens of Proteus. They cross react. Okay. So there is a mnemonic which is okay, Mira, scrub. So there is a girl, her name is Meera, and you're telling her, okay, Meera, now you scrub. Okay, so you know that ox K produced by Proteus mirabilis is given positive by scrub typhus. Okay, so OX-K antigen produced by Proteus mirabilis is given positive by scrub typhus, right? So when I talk about Weil-Felix reaction, there are the OX-K antigen, OX2 antigen, and OX19 antigen. OX-K is produced by Proteus mirabilis, and these two are produced by Proteus vulgaris, right? This is given by scrub typhus. What about these two? So OX2 plus OX19 both are given by Rocky Mountain spotted fever, but OX19 is given by rest all. ठीक है ना? So this becomes easy, right? Now along with this you have another thing which is the rickettsial pox. Rickettsial pox. This looks very much like the chicken pox. Who gets chicken pox? Children. What do children watch? They watch Mickey Mouse. So from Mickey Mouse you can remember Mighty Mouse. So this is mite-borne. Rickettsial pox is mite-borne and it is caused by Rickettsia acari. Okay. So all of the rickettsial group is done. All of, your scrub typhus is done. Now what is there? Now we come on to parasitology, right? You're done with the major chunk, you're done with general, you're done with the bacteria, you're done with, mycology. Now we're on to parasitology and virology. Okay, so yes, we lost a lot of time today and we were supposed to do 4 hours of microbiology, but it seems that we are just going on and on with microbiology today. But I think let's have faith, let's be strong, and let's finish it off, because then otherwise you will never be able to find another day to revise micro. Okay, so chalo, let's begin and let's finish it off. A 35-year-old male presents with abdominal pain, bloody diarrhea, and weight loss for 2 weeks. On stool microscopy, there is trophozoites with the ingested RBCs. Immediately you know that we are talking about amoebic dysentery with the trophozoite with the ingested RBC, right? Which of the following statements about all causative organism is true? It produces cysts that are quadrinucleated and infectious. Yes. These quadrinucleated cysts are the mature cysts which are infectious for Entamoeba histolytica. It is a flagellated protozoan? No, it is an amoeba. It does not invade the tissues. No, it invades and it causes amoebic liver abscess. It causes the flask shaped ulcers, and you know amoebic liver abscess is associated with the anchovy sauce pus or the anchovy sauce pus. It only causes intestinal disease, no extraintestinal. We have just seen the extraintestinal manifestation is the amoebic liver abscess. Yes, amoebic liver abscess. So you can see here, this is the pseudopod, this is the nucleus with the central karyosome, and this is the mature quadrinucleate cyst, right? And you can see the, yes, of course, the central karyosome inside the nucleus. This is the trophozoite of Entamoeba histolytica. Entamoeba histolytica is causing flask-shaped ulcers in the intestine. The amoebic liver abscess gives me the anchovy sauce pus or the anchovy sauce pus appearance. How do I do a lab diagnosis? For diagnosis, I can do the triage test. The triage panel test, which is a stool antigen, test, which detects three things. It detects Entamoeba histolytica, it detects Giardia, and it detects Cryptosporidium. Right, Cryptosporidium. So, yeah, all right. So you have a 20-year-old male presents with history of swimming in freshwater lake. Swimming in freshwater lake, then he develops high-grade fever, headache, photophobia, neck stiffness. Of course, we're talking about amoebic. Beta PDF you will get at the end of the class. You will get the annotated PDF at the end of the class, right? So, yeah, we are talking about free-living amoeba which cause meningitis. So, now it is written, a few days later, he develops high-grade fever, headache, photophobia, next. Despite treatment, he rapidly deteriorates and dies. What are we talking about? We're talking about brain-eating amoeba, Naegleria fowleri, which is spread through the nose. So, you are swimming in water through the nose. The trophozoite breaks the cribriform plate and enters the brain, causing primary amoebic encephalitis, which is highly fatal. Primary amoebic meningoencephalitis, which is PAM, which is highly fatal. Within 12 days, it causes the death of the patient. Okay, which of the following statements is true? It causes chronic meningoencephalitis in immunocompromised, yes, but not chronic. Acute meningoencephalitis in immunocompromised. It is a free-living amoeba that enters through the gastrointestinal tract. No, it enters through the nose, Then you have it causes primary amoebic meningoencephalitis by entering through the nasal mucosa. Correct, right? Then ciliated protozoa, no, it is not ciliated. It is amoeboid. It is also amoeboid. But the trophozoite stage of Naegleria fowleri can change between a flagellate form and an amoeboid form. So the trophozoite of Naegleria fowleri is amoeboid as well as flagellate. So it keeps changing between the two. But the infective stage is only and only the amoeboid stage, which enters through the nose and causes the infection. The flagellate stage does not cause the infection. The cyst also does not cause the infection. Okay. Then you have another. So you can see this is the cyst form, this is the flagellate form, this amoeboid form, this is the flagellate form. In nature, this flagellate and amoeboid, they keep changing, and then this amoeboid can change into cyst and then vice versa, right? So this keeps happening inside the nature. Who is he? This guy is showing spines in the trophozoite and the wrinkled membrane in the cyst. This is your acanthamoeba. The name is saying amoeba, meaning amoeba with the spines or spicules in its trophozoite, right? It causes two things. It causes granulomatous amoebic encephalitis. Granulomatous amoebic encephalitis is chronic meningoencephalitis in immunocompromised. So this statement which was given here, this statement is actually true for your acanthamoeba, okay? And then it causes another very important thing, which is contact lens keratitis. Contact lens keratitis. Okay, contact lens keratitis. The moment you hear contact lens keratitis, you either think of acanthamoeba or you think of Pseudomonas. Only two things you think of in contact lens keratitis. Okay, 45-year-old पिग फार्मर, पिग आते ही दिमाग में आना चाहिए बैलेंटिडियासिस, एब्डोमिनल पेन एंड ब्लडी डायरिया, डाइसेंट्री भी हो गया, स्टूल माइक्रोस्कोपी में लार्ज सीलिएटेड ट्रोफोजॉइड, the only ciliated trophozoite infecting man is the one with the cilia, cilia means ball means Balantidium coli, with the kidney shaped macronucleus. Everything has been given in the question itself. There is a kidney shaped macronucleus and a small dot shaped micronucleus followed by the body and the cilia. This is what is Balantidium coli. The pigs are the reservoirs of this and this causes your dysentery or bloody diarrhea. ठीक है ना? ये कौन है? This is the old uncle with the two nucleus and the suckers and the four pairs of flagella and the pear shaped trophozoite and the cyst with the four nuclei and the central exostyle dividing into two. This is Giardia lamblia. What does Giardia lamblia cause? Giardia lamblia causes flatulence or steatoria. What is steatoria? Steatoria is fatty stools. Fatty stools. Why? Because it is leading to fat malabsorption. This fat malabsorption is happening where? In the small intestine. In the small intestine, especially where? In the duodenum. In the duodenum. So which is the most important specimen that you have to collect for giardiasis? It is the duodenal aspirate. Duodenal aspirate. And how do we collect it? We collect it by the string test or the entero test. Okay, this is the string test or the entero test. String test or the entero test. All right. Then giardia, yes, it causes fatty stools or steatoria, and it is in the duodenal aspirate taken from the stool test or the entero test is actually the best test that you can perform. Then you know, of course, there is also the triage panel testing which can be done for giardia, intimeba histolytica and also your cryptosporidium. Okay. Then who is this? This is another trophozoite with the four anterior flagella and the one long undulating membrane behind. You know, so I had a friend, she had such a long hair that when she used to sleep at night, she would get tugged all in her hair itself. Okay, so same happens with this. T for Trichomonas vaginalis. Okay, T for Trichomonas vaginalis. Oh God, what has happened? So this has only a trophozoite stage. There is no cyst stage, right? It has only trophozoite stage. There is no cyst stage seen. And there is this undulating membrane or the fifth flagellum which wraps around the body of this guy. This is the only cause of parasitic sexually transmitted disease, right? Only this one. This causes your trichomoniasis, which is vaginal infection. And what do you see in this? You see no whiff test, no Amsel's criteria fulfilled. Okay, you see motile trophozoites. And what do this show? The twitching motility. The twitching motility is seen. All right, and of course you remember it causes strawberry cervix, which is also called as colpitis macularis. Colpitis macularis. Okay, which is the strawberry cervix, and you will classically see the greenish frothy discharge. Greenish frothy discharge which comes out from this, and you use Diamond's media, right? All of this you know. Then you have this guy. Who is he? This is also an undulating membrane. You can also see an undulating membrane in this. But this is not the anterior flagella. This is not the pear-shaped body. This is the S-shaped body. What is this? This is trichomoniasis. This is trypanosomiasis. Trypanosomiasis. What does trypanosomiasis cause? It causes sleeping sickness. Sleeping sickness. Sleeping sickness can be of two types. There is American sleeping sickness and there is African sleeping sickness. Okay. Yeah. There is American sleeping sickness and there is African sleeping sickness. Right? What is American sleeping sickness? It is American trypanosomiasis. How do we remember? This American guy, Tom Cruise. Tom Cruise has fallen in love with a red-haired girl called Romana. And where are they falling in love? In their Mercedes-Benz C-Class. So what all do you see in American trypanosomiasis? You see Trypanosoma cruzi or Tom Cruise is the causative agent for it. Who is this? This is Romana. Romana has red hair. So redovid bug. Redovid bug, which is also called as the kissing bug since they are falling in love, or the triatomine bug, right? Then what is her name? Romana. So it shows the Romana's sign. What is Romana's sign? This periorbital swelling which is seen is called as Romana's sign. Where are they falling in love? They are falling in love in Mercedes-Benz C-Class. So C is Chagas disease. Chagas disease. And from Benz you can remember that benzimidazole is the treatment of choice. And since she has fallen in love with Tom Cruise, she's going to have a big heart. So dilated cardiomyopathy or megacolon, megaesophagus. What is happening? All of these are the features of your American trypanosomiasis. Okay? So you have dilated cardiomyopathy. Dilated cardiomyopathy, right? Then what about African sleeping sickness? You have sleeping sickness. So the agent is Sessefly, Sessefly or Glossina species. Glossina species. You see Bruce Lee. So this is caused by Trypanosoma brucei. Now Trypanosoma brucei can be Trypanosoma brucei gambiense or it can be Trypanosoma brucei rhodonense. Right? So how to remember? Which one is West African sleeping sickness? Which one is East African sleeping sickness? So what does Bruce Lee ride? Bruce Lee rides the G-Wagon. The G-Wagon. So Gambiense is West African sleeping sickness. Okay. So Gambiense will be West African sleeping sickness and Rhodonense will be East African sleeping sickness. And what is seen here? So where is Bruce Lee? In winter. So it shows the winter bottom sign. What is winter bottom sign? It is the post-cervical lymphadenopathy. Okay, so just remember Bruce Lee rides his G-Wagon in the winters. So winter bottom sign and West African sleeping sickness, Trypanosoma brucei gambiense. Right? And Tom Cruise falls in love with the red-haired Romana in a Mercedes-Benz C-Class. ये क्या हो रहा है? Black discoloration and hepatosplenomegaly. The moment you see this, you know we are talking about leishmaniasis. And what can you see? The dot and a dash sign. Dot and dash, with the dot being the kinetoplast and the dash being the nucleus. The kinetoplast and the nucleus. What is this? This is the A mastigote sitting inside a macrophage. This is LD bodies or Leishman-Donovan bodies. What are LD bodies? They are nothing but the A mastigote. inside a macrophage. Yes, because this is obligate intracellular and this is called as Kala Azar, which is caused by Leishmania डोनोवनी. लीशमेनिया डोनोवनी. ठीक है, लीशमेनियासिस. काला आजार के क्या क्या फीचर्स हैं? व्हाट इज द पेंटैड ऑफ काला आजार? यू हैव द काला विच इज द ब्लैक डिस्कलरेशन ऑफ द स्किन, you have the fever which is the azar, then you have the hepatosplenomegaly which is the most important thing, hepatosplenomegaly. ठीक है, leishmania donovani causes visceral leishmaniasis. This visceral leishmaniasis is what we call as kala azar. What happens when you have visceral leishmaniasis? You treat with antimony. You treated with antimony and there was sun exposure and the patient developed nodules on the face. That is post Kala Azar dermal leishmaniasis. This is a complication of leishmaniasis. As a result, the treatment of choice nowadays is liposomal amphotericin B. Liposomal amphotericin B is the treatment of choice these days. Who are these guys? The acid fast round structures, the small round ball of cryptosporidium, the big round ball of cyclospora, and the big oval of cystoisospora. Then the cryptosporidium is single walled, that is why it is called as naked sporozoids. These are called as naked sporozoids because a single cyst wall is there, and this is double walled and there are 2 into 2 4 sporozoids inside, and over here it is 4 plus 4 8 sporozoids inside. Okay, so there are 8 sporozoids here, 4 sporozoids here, 4 naked sporozoids over here, right? These 4 naked sporozoids are cryptosporidium, this is cyclospora, this is cystoisospora. Okay, and the sizes are about 4 to 5 micrometers. Then double it, 8 to 10 micrometers. Double it, 20 to 30 micrometers. What is the breadth? Just half of it, 10 to 15 micrometers, right? They are all acid fast. For all of them we perform Zn staining, but you see all of these are pink, but over here this is pink, but this is not pink, this is not pink, this is not pink. So cyclospora is characteristically non-uniformly acid fast. Non-uniformly acid fast. It is non-uniformly pink. But your cryptosporidium is crypto, means it is hidden. So all the three are going to show autofluorescence, but cryptosporidium is not going to show autofluorescence. So the thing with cryptosporidium is that it does not show no autofluorescence. However, what does it show? It shows auto-infection. Auto-infection is seen, and this leads to outbreaks also. So you can see the differences over here of all the three. What is the infective form of all? See, infection will only happen if you ingest a sporulated oocyst. So the infective form of all is sporulated oocyst. But the moment it is excreted out in the feces, the cryptosporidium is sporulated, whereas these two are unsporulated. They are going to become sporulated in the soil. So what do you think can cause an outbreak? Cryptosporidium causes an outbreak through recreational waters, through recreational water, meaning swimming, etc. Why? Because this guy is resistant to chlorination. Resistant to chlorination. ठीक है, oocyst shape is of course round in these and oval in that. It has 4 sporozoids. It has 2 into 2. It has 2 plus 4, 4 plus 4, which is 8. So here there are total 8. Here there are total 4. Here there are total 4, which are also naked. As a result of this nakedness, the auto-infection is possible in cryptosporidium. ठीक है, this is uniformly acid fast, variable or non-uniformly acid fast. Here there is no autofluorescence and here there is autofluorescence. The sporulation of course happens inside the host. That is why a sporulated oocyst is being excreted in the feces. Over here, the sporulation occurs in the soil or in the environment. This cryptocurrency is by the nitazoxanide, and these two are cotrimoxazole, right? You know the differences of all of these. Now, toxoplasma. Toxoplasmosis is caused by Toxoplasma gondii. Toxoplasma gondii, right? He causes toxoplasmosis. One very important feature of toxoplasmosis is that cat is the definitive host, and it causes congenital toxoplasmosis because it's a part of the torch infections. It is a part of the torch infections, right? So toxoplasma causes congenital toxoplasmosis. So how do we remember everything about congenital toxoplasmosis? We just remember a small little baby cat. Okay, we just remember a baby cat. So this is a big head of a baby cat, this is a small body of a baby cat, this is the tiny little, and you see, this is the cat. Now you do one thing, you give the cat many spots on the head, and you give her red eyes. So what are all the features of congenital toxoplasmosis? Because it's a cat, you know that the definitive host of this disease is the cat. Okay, is the cat. And you see the red eyes, so you know it is chorioretinitis. Oh God. Chorioretinitis. Then the spotted head is telling you intracerebral calcifications, intracranial calcifications, and the big head of the baby is telling you it is hydrocephalus. Hydrocephalus. These are all the features of congenital toxoplasmosis, right? Now, toxoplasmosis is caused by cat, so the mainstay of diagnosis is also CAT scan or CT scan. CAT scan or CT scan is the mainstay of diagnosis. Who is he? This is Plasmodium falciparum. Why? Because you can see the multiple rings inside a single RBC. You can see the double dotted rings in the multiple dots in a single ring. And you can see this acole form, which is the applique form, which goes to the corner and sticks. What is this? This is the banana shaped gametocyte of Plasmodium falciparum, right? Plasmodium falciparum causes malignant malaria, which is the most dangerous type of malaria. And why does this malignant malaria happen? Because of cytoadherence. Over here you see big oval RBCs with a single ring stage inside. This is Plasmodium ovale or Plasmodium vivax, and this here is the schizont of Plasmodium vivax, and this is the band form of the trophozoite of Plasmodium trophozoite of Plasmodium malariae, right? So these are all the features of Plasmodium. So how do we do a diagnosis of Plasmodium? You know that falciparum is going to cause your malignant malaria, so that is going to cause cerebral malaria, black water fever, algid malaria. Why does this happen? This happens because of the feature which is called as cytoadherence and rosetting. So what happens in Plasmodium falciparum? The parasite infects the RBC, makes the RBC sticky. Now these sticky RBCs are binding to the blood vessels, binding to other RBCs inside the brain. So it has gone and hidden inside the brain. Now you cannot find the parasite out in the blood. You cannot find the parasite, because it is sticking inside the RBCs, that inside the blood vessel of the brain. That is called as cytoadherence, and this cytoadherence happens because of the PCAM1 receptors, and this happens because of the PFEMP, Plasmodium falciparum endothelial membrane protein. This is what makes the RBCs into sticky knobs on the RBC. Okay, so that is why the RBC is sticking everywhere, and it is causing malignant malaria or cerebral malaria. ठीक है. Now, this what is the way of diagnosis of malaria? You know, you do a peripheral blood smear, and on the peripheral blood smear you see all the ring stages, the Schizon stage, etc., which we have just seen. Okay, then you can do a quantitative Buffy coat. In the quantitative Buffy coat, you look at the Buffy coat under the fluorescent microscope and you quantify the parasite which is there. Then you can do something which is called as immunochromatographic test or rapid card test. In this, if you detect aldolase or if you detect LDH, then this means all species of Plasmodium. But if you detect HRP2 plus specifically Plasmodium falciparum LDH, this shows Plasmodium falciparum positivity. So HRP2 is the specific antigen for, yes, PCAM molecules, correct. HRP2 is the specific antigen for detection of Plasmodium falciparum. Please remember that. Okay. Then who is he? He is the cross, Maltese cross sign. What is this? Maltese cross sign. Okay, and with X you can remember exodistic. Yes, this X is kind of also looking like K. So just remember tick, exodistic. What is this? Babesia microti. Babesia microti causing babesiosis. This is a close cousin of Plasmodium, right? So even in Plasmodium, even in Babesia, even in Toxoplasma, human is what? Human is the intermediate host. Human is the intermediate host. And who is the definitive host? It is the exodistic, which is the definitive host in case of babesiosis. Okay, babesiosis. Right. So we are almost about to finish off. So let's hang in there. Okay, let's look at these eggs. What are these? These are the eggs of cestodes. So cestodes have these hexacanth embryo inside with the polar thickenings and the polar filaments, and it is a non-bile stained egg. Of course, what are we dealing with? We are dealing with H. nana. H. nana. What are the features of H. nana? H. nana says no. No means no intermediate host. So this is also called as the dwarf tapeworm. The dwarf tapeworm with the no intermediate host is H. nana. Then you have this guy. What is this? Operculum. Operculum means dhakkan. So who is the dhakkan or the lid operculated egg? Is the D for Diphyllobothrium latum. He is the dhakkan of the cestode family. Why? Because he has di means two intermediate host, and this is the way that trematodes behave. All right. Then who is this? This is the hexacanth embryo with the radial striations. So here we are dealing with Taenia species, and you know Taenia species has the bile stained eggs. The bile stained eggs. All right. Then hydatid cyst. Hydatid cyst is the disease caused by Echinococcus granulosus. Echinococcus granulosus. Who is he? He is the dog tapeworm. Dog tapeworm. Who is the intermediate host? Intermediate host is human. And who is the definitive host? Dog. How do you get it? Ingestion of the eggs of the parasite. And you get the hydatid cyst disease. What is the treatment of choice? Pear is the treatment of choice for hydatid cyst disease. Okay, Echinococcus granulosus. Trematodes. Trematodes का क्या feature होता है? All trematodes have some common features. They are all called as the flukes. Flukes. So you have the liver fluke, lung fluke, right? Blood fluke and intestinal fluke. All of them have common features. That in all of them, the mode of infection is going to be ingestion. And what are we going to ingest? We are going to ingest the Metacercaria larva. The Metacercaria larva. Alright guys, so since the pen is glitching really, I'm going to write less and speak more. So what I want you to do right now is to get up and pay attention to what I'm speaking, because I am going to speak a lot more than I'm going to write right now. So there is going to be a verbal diarrhea. However, not much will be written because this pen and this thing is really glitching and it's really slowing us down. So Metacercaria larva is the infective stage of all trematodes. It is through ingestion of either fresh aquatic plants or fish. Okay, so if it is the fresh aquatic plants, it has the sound of F. So every parasite with the sound of F, such as Fasciola hepatica, Fasciolopsis busci, they are all going to be F, or fresh aquatic plant. Rest all Clonorchis, Opisthochis are going to be transmitted through the fish. Okay, it has 5 larval stages. The mnemonic is my silly red cat meows. Myracidium, Sporocyst, Rhedia, Cercaria, Metacercaria. This Metacercaria is the one which we are ingesting and getting infected. So either we ingest Metacercaria larva with the help of fresh aquatic plants such as in Fasciolopsis busci, which causes intestinal fluke, and Fasciola hepatica, which causes the liver fluke, or we ingest this Metacercaria larva in the form of a fish, which is then which is by the ingestion of Clonorchis, Opisthochis, which is Clonorchis opisthochis, which cause bile duct cholangiocarcinoma. They are the malignant guys. All right, then there are the 5 larval stages in all trematodes, and all trematodes are monoecious, meaning sexes are not separate and they all look like flukes or they have a leaf like body. And most importantly, they lay down the operculated eggs, which is the dhakkan wala eggs or the lid eggs. Who are the exceptions of this flukes? It is the schistosomes. They are the exceptional guys of the trematode family. All exceptions of the schistosomes can be written with the word schistosome. So let's write. Okay, schistosomes. So you have the mode of infection is actually skin penetration by the Cercaria larva. There are 2 sexes. It is snail only, the spined eggs and the slit-like body. So the mode of infection of all trematodes was what? It was ingestion of Metacercaria larva. Here it is skin penetration by the Cercaria larva. Okay, all of the trematodes were monoecious. Here the sexes are 2. There are 2 separate sexes. Now, if there are 2 separate sexes, where is the female going to live? The female is going to live inside the slit-like body of the male. While they were all looking like flukes or had the leaf-like body, here there is the slit-like body. While they all had operculated eggs, this guy has spine in the eggs. It is non-operculated eggs. While all of them had 2 intermediate hosts, right? 2 intermediate hosts, where the one intermediate host, the first intermediate host was snail, and the second intermediate host was either a fresh aquatic plant or a fish, depending upon which parasite. Over here it says snail only. So for schistosomes, there is only one intermediate host, which is snail. All right. So then you look at the different guys. Schistosoma mansoni, Hematobium, and Japonica. So Mansonica mnemonic hai tedi mansi. So it has the tedi or the lateral spine. Right? You know, schistosoma hematobium causes bladder carcinoma or urinary bladder cancer. So if you draw a bladder, you draw the urethra like this, right? Similarly, you have the egg with the terminal spine. All right. Then you have the lateral knob in Japan, because Japanese people are cute as a button. So button or knob is seen in schistosoma japonicum, right? So like I have said, guys, I'm going to speak a lot more, write a lot less, because of the glitches that we've been facing since in 11 a.m. today. It's been 6 hours of fighting the glitches, but we have stayed and continued, and we are going to finish off and revise entire microbiology. So more than me, huge, like very strong you guys are. So good job, guys. Right. Next is what? This is a bile stained egg with the undulating membrane and the undifferentiated ovum. And here you can see the clear cut embryo with the crescent shapes and the undulating membrane. So what is this? This is the unfertilized egg of Ascaris, and this is the fertilized egg of Ascaris. Yes, this is the unfertilized egg of Ascaris, and this is the fertilized egg of Ascaris. Both of them are bile stained eggs. All right. Now, which one of these floats in the salt solution? You know that feather floats, right? So fertilized egg of Ascaris will float in saturated salt solution. Trichuris will float in saturated salt solution. Hookworm will float in saturated salt solution, and Enterobius vermicularis, which is the pinworm, will float in saturated salt solution. All right. Then what is this? This is a non-bile stained egg with the blastomeres, and this is your Hookworm egg, hookworm egg. There are two hookworms, Nicator americanus and Ankylostoma duodenale. So Ankylostoma is telling you that it is everything about the mouth, and Nicator is telling you that it is going to suck your blood like nectar. So both these hookworms, what do they cause? They cause iron deficiency anemias because they suck out all the blood. They have well formed mouth parts, Ankylostoma, through which they are going to cut the plate. They have cutting plates or well formed teeth through which they are going to suck out all the blood from your intestine. Then you have this guy with the two mucus plugs and the barrel shaped egg. Who is he? He is Trichuris trichiura. Trichuris trichiura is known for causing dysentery, is known for causing rectal prolapse, and is known for causing your coconut cake rectum. Coconut cake rectum. Okay. So what is so how to remember? This is a bomb shaped egg. So this is going to burst in your intestine, leading to blood loss, dysentery, leading to rectal prolapse, and a coconut cake rectum. All right. And of course, Trichuris trichiura is a soil transmitted helminth. The eggs are going to float in the saturated salt solution. The larva and no egg. This is of course the strong independent female, Strongyloids stercoralis, which enters through skin penetration, and this is ovoviviparous. Ovoviviparous. Okay, and who is this? Plano-convex, the T-shaped egg with the tadpole-shaped larva. This is of course your pinworm. Pinworm, which is Enterobius vermicularis, right? The D-shaped plano-convex egg. हो गया. ये कौन है? This is the round worm. This is of course Ascaris lumbricoides, wherein you can see the male and the female. You can see the well-developed mouth parts of the hookworm here. This is the whip-shaped. So what is whipworm? Whipworm is nothing but your Trichuris trichiura, and there's so many whipworm is giving you the coconut cake rectum appearance. The coconut cake rectum appearance. All right. What is this? Elephantiasis. Elephantiasis is caused by lymphatic filariasis. You have two important lymphatic filariasis, Wuchereria bancrofti and Brugia malai. Please remember that Brugia malai is more common in India. In India, right? India say you can remember Manmohan Singh and Sonia Gandhi. So Manmohan and Sonia, this is Mansonia is the mosquito which spreads Brugia malai. Please remember this. Okay, Brugia malai is through Mansonia. So India, so remember Manmohan and Sonia. Okay, now Wuchereria bancrofti, Brugia malai, loa LBW. These are the guys which have a sheath. So you look at the microfilaria of these filarial worms, and you see that Wuchereria bancrofti sounds like bankruptcy. So there are no nuclei in the tail tip. Brugia malai is bi. So bi means two. So there are two nuclei in the tail tip. Whereas loa has a line of nuclei in the tail tip. Okay. So these are sheathed microfilaria with no nuclei, two nuclei, and line of nuclei. Whereas Onchocerca volvulus, all of these microfilaria are seen in the blood, whereas in Onchocerca the microfilaria is seen in the skin. So when I say Onchocerca volvulus, I have to focus on O. So when I am saying O, I am saying 0. 0 means 0 nuclei and 0 sheath. Okay, and O, if I draw O like this looks like a leopard skin. So there is the leopard skin appearance. If I draw this O and I hang it down, it becomes hanging groin. Right? If I make this O and I put a cross, this is blindness. River blindness. That is also onchocerciasis. Now we have always seen the leopard sign on a black colored skin. So this is caused by the black fly or the Simulian fly. The only thing is that these microfilaria are going to be seen in skin snip smears. Skin snip smear is going to show me the microfilaria with no nuclei and no sheath. All right, onchocerciasis. Then loa is a African eye worm. And तुम मेरी आँखों के राजा हो, you are the deer of my eyes. It is caused by deer fly, because you are deer of my eyes, so deer fly. All right, so this is loiasis by the deer fly. Okay, deer fly. DNA viruses are happy viruses. Y looks like 4. So there are 4 P's. Human herpes virus, Hepadina family, Adeno, Papova, Parvo, and Pox. Yes, Pox and fudge. Right? What is PADNA? This is hepatitis B. The only hepatitis virus which is DNA and has partial DNA, PNA-DNA. All these three are non-enveloped. All of the happy viruses are enveloped viruses, whereas adeno, papova, parvo are non-enveloped. All happy viruses are double-stranded DNA viruses. पर वो तो सिंगल स्ट्रैंडेड डीएनए वायरस है. All right, excellent. What is happening here? You see the multiple vesicles. This is the multiple vesicular rashes seen in a cluster. Of course, we are talking about herpes, which is caused by HSV1 and HSV2. You know that HSV2 is below the waist and HSV1 is above the waist. So this will cause genital herpes, and this will cause the herpes labialis or gingivostomatitis, right? So this is through the oral secretions and this is through the genital secretions. Who causes mollaret meningitis? It is HSV2. And what is mollaret meningitis? Recurrent lymphocytic meningitis. So there is going to be recurrent meningitis and there is going to be lymphocytosis, and you get a 10 to 15-year-old child with the recurrent lymphocytic meningitis, that is mollaret meningitis. Over here you see herpetic vitello, which happens in the dentist. You see herpes gladiatorum, which happens in your sumo wrestlers. Then herpes labialis, which happens on the top of the lips, right? So that is herpes HSV1. Then you have mumps. Mumps is this bull neck appearance. It is the parotitis. Then this parotitis can go and cause orchitis. This can go and cause epididymoorchitis or epididymitis, oophouritis, meningitis. Basically, it has started from the lymphoid organs and it is going to spread to all of the body, right? So measles, mumps, and rubella, these are the 3 important vaccines: measles, mumps, and rubella, where mumps strain is the Gerald-Lynn strain. Is the Gerald-Lynn strain. Rubella is the RA273 strain. And measles has Moratan, Edmonston, Zagreb, and Schwartz strain. All right, fine. Then amongst the paramyxoviridae, we have to remember this mnemonic, PMMRM. What is PMMRM? It is para-influenza, right? There is mumps, measles, not rubella, it is RSV, and it is human metapneumovirus, right? What do they keep saying? They keep saying ha and na. हाँ एंड ना. सो हीमग्लूटिनिन एंड न्यूरामिनिडेज. ठीक है, तो क्या होता है? दे से ओके वी नीड बोथ हाँ एंड ना. मीजल्स हैज वन लेस मीन्स हाँ है और ना नहीं है. सो हाँ इज देयर but na is not there. Okay, then RSV says both negative, HMPV says both negative. So if they talk about hemagglutinin or neuraminidase proteins, then this is the way to remember it. And all paramyxoviridae have the F protein or the fusion protein present in all of them, right? So when we talk about viral exanthems, especially of childhood, we have to talk about measles, scarlet fever, rubella, erythema infectiosum, which is parvovirus B19, then roseola, which is the herpes virus 6 and 7, and varicella zoster virus, which is chickenpox virus, which is nothing but human herpes virus 3, and hand, foot, mouth disease, which is the Coxsackie virus A, especially Coxsackie A16 and A6, right? So you remember the shoe brand A6, so just remember A6 and A16, all right? So measles is the head to toe rash, wherein you see maculopapular rashes going from head to toe. And how do they go? The way you put behind your hair. So post auricular, nape of neck, and then the trunk. So just put your hair behind like this, and that is how the rash, maculopapular rash of measles goes. Then you see the Koplik spots, which is the buccal cavity white colored raised spots with the pink or erythematous base against the second molar. You have high fever and the 3Cs plus fever, which is cough, coryza, and conjunctivitis, right? So this is the features of measles. So like I said, rash begins at the hairline and spreads inferiorly. This is the way, this is the head to toe kind of a rash. This is measles. So what is the way that measles rash spreads? So on the 10th day you have, on the 12th day, and on the 14th day. So 10th day is fever plus 3Cs, 12th day is the Koplik spots, and 14th day is the rash. Okay, this is the way, this is the timeline in which your measles rash spreads. Then scarlet fever is Streptococcus pyogenes, which is group A Streptococcus. It is by streptococcal pyrogenic exotoxin A and C. There is the sandpaper kind of a rash. And how to remember everything about scarlet fever? You remember what was the toxin? SPE, right? So there is the sandpaper rash, there is the strawberry tongue, there is the Pastia's lines. Okay, so you have Pastia's lines, strawberry tongue, and sandpaper rash, which is seen in scarlet fever, right? Then there is also circumoral pallor, etc. एरिथीमा इन्फेक्शियोसम इज बाय parvovirus. Parvo तो single stranded DNA virus है. It causes the slap cheek appearance inside the baby. So what does it do? It slaps the babies and breaks the bones of adults. So it will slap the babies and will cause arthritis in the adults. Now it attacks the P for parvovirus, P for the P antigen on the erythrocyte precursor cell. So it is going to kill the immature RBCs. So of course there is going to be a plastic crisis, right? Roseola infantum caused by human herpes 6 and 7. You have the rosy pink baby and the maculopapular uvular rashes. Okay, inside the uvula you have the spots. What is that? Nagayama spots. Nagayama spots. All right. Then varicella zoster virus, which is human herpes 3. It causes vesicles and the multiple crops of rashes, right? So at the same time, there is pustule, papule, scab, everything is present at the same time. It begins on the face and spreads centripetally. So it goes from center to outside, okay, and spreads to the flexor surfaces. So always to remember the rash of chickenpox, you do this dance, right? So this is the face and trunk, and then it spreads to the flexor surfaces to the arms, right? Then you have the hand, foot, mouth disease, which is by Coxsackie A6, A16, enterovirus 71, right? There is the hand, foot, and mouth disease. Vesicles are seen on the hand, foot, and the mouth, okay? Then amongst arboviruses, arbo means arthropod borne viruses. You have the choga or the toga viridae, which is the chikungunya. In chikungunya, you see the brownie nose sign. Then you have the flaviviridae. Flavi means yellow. What is your mnemonic? Yellow vest kid is DJ. So what are all the flaviviruses? Yellow fever, West Nile encephalitis, Kaisunur forest disease, Zika virus, and dengue. Dengue as well as Japanese encephalitis. Okay, all right. And you have these silver colored bullets. What is that? Chandipura. Chandipura, right? So just imagine that these silver colored bullets are lying in some sand. So this is caused by sand fly. Sand fly. And when I write tick, K is Kaisanur forest disease and C is CCHF. Right? Rest all are in this list is caused by mosquito. Yellow fever, Aedes mosquito. West Nile encephalitis, mosquito. Zika virus, mosquito. Dengue, mosquito. Japanese encephalitis, mosquito. Chikungunya, mosquito. And which mosquito are we talking about? The Aedes aegypti or the Aedes albopictus, right? It is Aedes through and through. So for Japanese encephalitis, what all are your buzzwords? It is Gorakhpur encephalitis. So if it is Gorakhpur, UP, West Bengal in the question, you know they're talking about Japanese encephalitis. There are major features of encephalitis. So you will see the brain parenchymal involvement. The vector over here is the mosquito, especially the Aedes. Is it Aedes or Culex? It is Japanese. So Japanese are cute. So cute tells me Culex. Cute tells me Culex mosquito is the vector for Japanese encephalitis. All right. Then you have the pigs. What are pigs here? Pigs are the amplifier hosts. And what are these water birds or herons? They are the reservoirs. What do I see in Japanese encephalitis? I see the iceberg phenomenon. That is, the subclinical cases are much more than the clinical cases, and it causes epidemics. Epidemic encephalitis, especially in the Gorakhpur belt, right? Epidemic encephalitis, okay, and it is endemic to Gorakhpur, UP and West Bengal, right? What is the vaccine strain? SA1414-2 strain. All right, SA1414-2 strain. Now let's look at the inclusion bodies. What is this? This is the owl eye inclusion. This is shown by cytomegalovirus. What is this? This is the Henderson-Patterson bodies, HP bodies, shown by the molluscum contagiosum virus, which is a pox virus, and it leads to the umbilicated dome-shaped ulcers or the pustules, RC. All right, lesions. What is this? This is the eosinophilic intracytoplasmic Negri bodies, which is shown by rabies, right? So when you talk about inclusion bodies can be both intracytoplasmic as well as intranuclear. So mnemonic is chief minister of both the states. So you have chief minister matlab CMV and measles. CMV is showing you owl eye and measles is showing you Warthin-Finkeldy giant cell. So W can be written as M can be written as W. So Warthin-Finkeldy giant cells. If it is eosinophilic, that means it is cytoplasmic. Then of course you have the Negri bodies for the rabies, and then all the poxes, like big boxes, they will live in the cytoplasm. So intracytoplasmic inclusion bodies of pox. So you have smallpox. Smallpox has two strains, vaccinia and variola. Vaccinia is the one used for vaccines, and variola is the one which causes the pox disease. So the pox disease will show the Paston bodies, and the vaccine was given in getting inspired from a cow. So cow is Guarneri bodies. Okay. Then you have the intranuclear, in which you have Cowdery type A and Cowdery type B, right? Cowdery type A is Hebap. A stands for Cowdery type A, B stands for Cowdery type B. This is herpes simplex, this is yellow fever, this is adeno, and this is polio virus. Okay, herpes simplex shows me the Lipschultz bodies or the multinucleated giant cell. Yellow fever shows me the Torres bodies, right? Done and done. Right. So, when it comes to the serology of hepatitis B, you know that hepatitis B is the first antigen to rise is the hepatitis B surface antigen, right? And the first antibody to rise is the anti-HBC antibody, right? Which is the one marker which is always high. It is the anti-hepatitis B core antibodies, which are always high, so they are also present in the window period, in the window period for hepatitis B virus, right? You know that hepatitis B and C are both of your transfusion transmitted hepatitis viruses. And when I talk about envelope, you don't give envelope to the first and the last, but you give envelope to all three in the middle. The D is the dependent or the defective virus, which is dependent on hepatitis B for its surface antigen. Hepatitis D can cause co-infection or superinfection. If HBC IgM is present, it is co-infection. If anti-HBC IgG is present, it is superinfection. All right. Okay. So now we talk about a person living with HIV, and this is a newly born patient, 3-month-old baby. So what are you going to do for diagnosis? For less than 18-month-old babies, what do? You do the dried blood spot, which is called as early infant diagnosis. And in this, you look at the HIV RNA PCR. HIV RNA PCR. All right. And otherwise, what can you do for HIV diagnosis? For HIV, you have the fourth generation ELISA. Fourth generation ELISA. And what are you using in this? You are using anti-envelope antibodies. So anti-GP160, anti-GP120, anti-GP41 plus anti-P24 as well as the P24 antigen. This P24 antigen detection is what has helped us overcome the window period of HIV virus. Then you can do the Western blot, which is confirmatory, and you can also do the PCRs for HIV virus, right? So what is this telling you? Loss of appetite, oral thrush. What is the testing strategy for any HIV positive person? You do antigen antibody combination immunoassay, which is called as the fourth generation ELISA, which we have just talked about. All right. Now, if you will tell me, GP120 is the binding antigen. Binding antigen. It binds to the CD4 receptors. What are the co-receptors to which it binds? It is chai cigarette 5 rupees, CCR5 and CXCR4, where CCR5 is the bigger number, it is present on the macrophages, and CXCR4 is present on the T cells. All right. And GP41 is what is called as the fusion protein. Fusion protein, which fuses with the cell of the host. All right. So yes, we will be posting the PDF very soon, and that's all, guys. All the very best. I mean, apologies are in order. Really sorry for all the glitches and all the bad experience because of the poor handwriting and all, but I have tried to speak a lot more and try to give you all the information in this concise amount of time. Congratulations to you for spending the whole day for microbiology.
Good evening, students. Good evening, Parth. So, NEET PG is just few days away, right? The clock is ticking, and we are going to do a marathon run of pediatric section now. So, gear your snacks, your water, whatever drink you want to have to feel energized. And without further ado, we are going to start this marathon session of pediatrics, right? So we are going to be very clear, crisp. We won't go deep down into solving complicated concepts. We'll go one-liners, what the examiners are going to ask, what are the frequent tested concepts. We'll just rush through them, right? Okay, so if the audio and the visual is fine, just give me a thumbs up and we'll just start then. Audio is fine, right? Okay, so let's start. Okay, to the very first concept, let's start with general pediatrics. So, in general pediatrics, the most important segment, these are the four curves. What are these curves? These are nothing but human growth curves, right? These are human growth curves. Human growth curves. These human growth curves are also known as Scammon curves, right? So I have four curves in front of me, curve A, B, C, and D, right? So one by one, let's see what all these curves are telling us. Firstly, let's look at the curve A. Curve A says that growth of this tissue happens maximum, that is 90% by 2 years of age, and thereafter there is little to no growth. So what is this tissue? What is this tissue? What is this tissue? The slide is not visible. I think the screen is visible now. Okay. So we were talking about human growth curves. I have these four curves in front of me. Curve A, B, C, and D. We were talking about curve A. Look at the curve A. Maximum growth is happening in the first 2 years of life. 90% of the growth has happened during the first 2 years, and thereafter there is little to no growth, right? Remaining 10% is happening thereafter. There is a plateau that is achieved later. What is this? What is this tissue? This is nothing but your neural growth or brain growth. राइट? डू यू रिमेंबर द हॉर्लिक्स एड? दे से दैट बच्चे के दिमाग का विकास सबसे ज़्यादा साल पहले 2 सालों में होता है, राइट? दे आर राइट. 90% ऑफ द ग्रोथ हैपन्स इन द फर्स्ट 2 years ऑफ लाइफ. सो दिस इज न्यूरल ग्रोथ और ब्रेन ग्रोथ, right? Now look at curve B. What is happening in curve B? Initially, this tissue is growing very rapidly. Then there is a plateau. Then again there is this spurt. So what is this? This is an S-shaped curve. This is an S-shaped curve, also known as sigmoid curve. Sigmoid curve, right? So remember this S-shaped or sigmoid curve. Where do you see this S-shaped or sigmoid curve? We see this in S for sigmoid, S for somatic. So your somatic growth is depicted by this sigmoid shape curve. Now let's see the curve C. Here, there is no growth till 10 to 12 years of age, and as soon as puberty hits, there is a spurt. So, this is nothing but gonadal growth, right? As soon as the child achieves the puberty, there is growth, gonadal growth or sexual growth. And lastly, you have this curve where you see this tenting, this peak in 4 to 8 years of age. Before that also there is no growth. After that also there is a regression, no growth. So this is nothing but lymphoid growth. In the age group 4 to 8 years of age, the lymphoid tissue multiplies rapidly. There is growth of the lymphoid tissue. That's why if you ever have encountered 4 to 8 years of age group child, you can easily palpate their lymph nodes. Their thymus is at the maximum size, right? So this is lymphoid growth. So let's revise again these 4 curves. If you see initially rapid growth, then plateau, this is neural or brain growth. Whenever you see this S-shaped curve or sigmoid shaped curve, this is sigmoid, this is somatic growth. Whenever you see peak or tenting after 10 to 12 years of age, this is sexual or gonadal growth. And wherever you see this peak at the age, in between the age group 4 to 8 years, this is lymphoid growth. So these are human growth curve, also known as Scammon curves. They depict that different tissues grow at different rates, and there is different pattern of each and every of these tissues, right? After growth curve, now let's move to नॉर्मल ग्रोथ पैटर्न. अगर आपको नॉर्मल पता होगा तभी तो आप एबनॉर्मल बता पाएंगे, राइट? सो लेट्स वन बाय वन डिस्कस दीज नॉर्मल एंड एबनॉर्मल ग्रोथ पैटर्न्स. So firstly, what is the average birth weight of a child, a term child in India? What is the average birth weight? Can anyone tell? So the recent Ghai textbook says that the average birth weight of a term child in India is 3 kgs. It's 3 kgs. So पार्थ इज आस्किंग सिग्मॉइड कर्व में ऐसे दो दो बार पीक्स क्यों हैं? देखो इनिशियली क्या होता है, बहुत रैपिडली ग्रोथ होती है. वेट 5 मंथ्स में डबल हो जाता है, फिर ट्रिपल हो जाता है, राइट? डबल हो जाता है, फिर ट्रिपल हो जाता है, फिर 4 टाइम्स हो जाता है, फिर उसके बाद एक प्लेट्यू आता है. फिर उसके बाद जब दोबारा से प्यूबर्टी आएगी, दोबारा से हिट आएगा, दोबारा से हिट आएगा, राइट? सो दैट्स वाई आपको ऐसा दिखेगा, 2 पीक्स दिखेंगी, राइट? क्लियर हो गया 2 पीक्स क्यों है? इनिशियली बहुत रैपिडली बच्चे बच्चे का वेट बढ़ता है, हाइट बढ़ती है, और फिर बाद में जब प्यूबर्टी आती है तब भी बढ़ जाता है, राइट? सो 3 kg इज द एवरेज बर्थ वेट ऑफ अ चाइल्ड इन इंडिया. अब क्या होता है उसके बाद? इनिशियली क्या वेट गेन होता है इमिजिएटली या लॉस होता है? यू आर राइट धनुष, इनिशियली देर इज वेट लॉस, फिजियोलॉजिकल वेट लॉस. ये सबका वेट लॉस होगा. 10% वेट लॉस होता है द फर्स्ट 7 days ऑफ लाइफ. और क्या होता है इस वेट लॉस में? देर इज लॉस ऑफ एक्स्ट्रा सेलुलर फ्लूइड, लॉस ऑफ एक्स्ट्रा सेलुलर फ्लूइड. ये वेट दोबारा से रीगेन हो जाता है बाय डे 10 ऑफ लाइफ, रीगेन बाय डे 10 ऑफ लाइफ, राइट? फिर उसके बाद गेन होता है. अब हमारे पास एक रेडी रेकनर है टू एस्टिमेट कि कितना वेट बढ़ता है, ये एग्जामिनर बहुत पूछता है, डबल कब होगा, ट्रिपल कब होगा, 4 टाइम्स कब होगा. सो इमेजिन अगर बर्थ वेट हम x लें तो it becomes 2x. That means it becomes double at 5 months of age. It becomes triple at 3 years of age, at 1 year of age. It becomes 4 times at 2 years, 5 times at 3 years, 6 times at 5 years, and 7 times at 7 years of age. Right? धनुष इज राइट. जो टर्म बेबीज हैं वो बाय डे 10 रिगेन कर लेते हैं और प्री टर्म लूज भी ज़्यादा करते हैं. They can lose also up to 14% and they regain by, sorry, they regain by day 14 of life. Right? So normal physiological weight loss 10% होता है in terms which is regained by day 10. Preterm 15% तक लॉस कर सकते हैं and that is regained by day 14. Right? Okay. So this is about the normal weight pattern. Yes, this is essentially the loss of extracellular fluid. तो ये सब बच्चों में ही होना है, सब बच्चों में ही होना है. बच्चे को अगर एडिक्वेट ब्रेस्ट मिल्क मिल रहा है तो ये पैटर्न एकदम सही रहता है. अगर ब्रेस्ट मिल्क कम मिल रहा है, बच्चा डिहाइड्रेटेड है, तो प्रॉब्लम होगी. राइट. सो नाउ लेट्स आफ्टर वेट, नाउ लेट्स मूव टू द लेंथ. सो जैसे हमने एवरेज बर्थ वेट पढ़ा था, वैसे एवरेज लेंथ कितनी होती है एक टर्म बेबी की? द एवरेज लेंथ एट बर्थ ऑफ अ टर्म बेबी इज कंसीडर्ड 50 सेंटीमीटर्स. 50 सेंटीमीटर्स. फिर 1 ईयर ऑफ लाइफ में देर इज गेन ऑफ 25 सेंटीमीटर. सो बाय द एंड ऑफ 1 ईयर कितना हो जाएगा बच्चे की लेंथ? 75. 50 प्लस 25, 75. राइट. फिर देन इन द नेक्स्ट 1 ईयर देर इज गेन ऑफ अबाउट 10 to 15 सेंटीमीटर्स. सो बाय द एंड ऑफ 2 ईयर्स कितना हो जाएगा? 85 to 90 सेंटीमीटर्स हो जाएगी बच्चे की लेंथ. राइट. एंड फाइनली उसके बाद इन द नेक्स्ट 1 ईयर बहुत कम गेन होता है. उसके बाद देर इज जस्ट 6 सेंटीमीटर पर ईयर टिल द चाइल्ड रीचेस प्यूबर्टी. सो मतलब 3 ईयर्स ऑफ एज में 96 सेंटीमीटर हो जाएगी. और जैसे ही प्यूबर्टी आएगी देर विल बी स्पर्ट, देर विल बी स्पर्ट. सो उसके बाद जब प्यूबर्टी आएगी तब देर विल बी स्पर्ट. कितना स्पर्ट आता है? मेल्स में it can go up till 10 centimeter per year, 10 centimeter per year. और females में 8 centimeter per year, 8 centimeter per year. Right? So this is how the increase in the length or height happens. So अब एग्जामिनर ये वन लाइनर्स पूछता है जो आपको एकदम रटे हुए होने चाहिए विद टिप्स. राइट. तो सबसे पहले वेट हमने पढ़ा डबल कब होता है? एट 5 months ऑफ एज. एंड ट्रिपल कब होता है? एट 1 ईयर. और यही देखिए हम हाइट या लेंथ का, वो कब डबल होती है? दैट डबल्स बाय 4 ईयर्स ऑफ एज. 4 ईयर्स ऑफ एज है ना, 4 ईयर्स के अराउंड ही होगी. अगर आप दोबारा से इसमें 6 ऐड कर दोगे तो 4 ईयर पे बच्चे की कितनी हो जाएगी? 102 सेंटीमीटर, विच इज क्लोज टू डबल ऑफ द length, average length. Right. So it doubles by 4 years and triples by 12 years of age. Triples by 12 years of age. फिर ये वन ऑफ द अनदर फेवरेट क्वेश्चन ऑफ द एग्जामिनर है कि चाइल्ड इज हाफ ऑफ द अडल्ट हाइट एट व्हाट एज. सो नॉर्मली अडल्ट हाइट कितनी मानते हैं हम? विद 160 सेंटीमीटर्स मानते हैं. तो उसका हाफ कितना हुआ? 80 सेंटीमीटर. 80 सेंटीमीटर कब होगा बच्चा? क्लोज टू 2 years of age. So the appropriate answer here is 18 months to 24 months. 18 to 24 months. If you have to choose one, choose 18 months. Right. So around this time the child is half of the adult height. So these are the important pointers with respect to length. Now let's move to the next growth parameter, that is head circumference. राइट. सो एवरेज बर्थ वेट हमने पढ़ लिया, एवरेज बर्थ लेंथ हमने पढ़ ली. व्हाट इज द एवरेज हेड सर्कमफ्रेंस एट बर्थ? सो हमारे पास रेंज है 33 to 35 सेंटीमीटर. 33 to 35 सेंटीमीटर. और कैसे बढ़ती है ये हेड सर्कमफ्रेंस? बहुत जल्दी जल्दी बढ़ती है. इन द फर्स्ट 3 months ऑफ लाइफ, 0 to 3 months में देर इज गेन इन 2 सेंटीमीटर पर मंथ. 2 सेंटीमीटर पर मंथ. फिर द नेक्स्ट 6 months में, दैट इज 3 months to 6 months, देर the gain is 1 centimeter per month. In the next 6 months, it's 0.5 centimeter per month, right? So this is how the gain happens. So in the first 3 months, it's 2 centimeter per month. After that, 1 centimeter per month. After that, 0.5 centimeter per month. तो अगर आप total gain देखो तो कितना हो रहा है? 2 centimeter per month, मतलब 2 into 3, that is 6 centimeter. Again, 1 into 3, that is 3 centimeter. फिर नेक्स्ट 6 मंथ्स में 0.5 इंटू 6, दैट इज अगेन 3 सेंटीमीटर. तो फर्स्ट ईयर ऑफ लाइफ में 6 + 3 + 3, दैट इज 12 सेंटीमीटर गेन हुआ है आपका हेड सर्कमफ्रेंस में. 12 सेंटीमीटर गेन हुआ. तो अगर आप 34 ले लो, बर्थ सर्कमफ्रेंस अगर बर्थ पे मैं 34 ले लूँ तो 1 ईयर पे कितनी हो जाएगी? 12 ऐड कर देंगे, दैट बिकम्स 46 सेंटीमीटर. 46 सेंटीमीटर, right? This head circumference, if you compare at the time of adult head circumference, so birth पे at head circumference 60% of the adult head circumference. और हमने पढ़ा था 90% तो growth by 2 years of age हो जाती है. So 90% of the adult head circumference is achieved by 2 years of age. Remember वो हमने ग्राफ पढ़ा था कि 2 years पे ही maximum 90% अचीव कर लेता है बच्चा. So the correct answer here is going to be 2 years. So ये दो important one-liners हुए कि head circumference is 60% of the adult head circumference at the time of birth, and it reaches 90% of the adult head circumference by 2 years of life. राइट. सो हमने ब्रॉडली जो नॉर्मल ग्रोथ पैरामीटर्स हैं वो तीनों पढ़ लिए हैं, बर्थ वेट भी, लेंथ और हाइट भी, एंड फाइनली हेड सर्कमफ्रेंस भी. अब पीडियाट्रिक्स में कुछ कुछ डेफिनेशंस हैं. वी हैव डिफाइंड सम पीरियड्स ऑफ ग्रोथ. ये पीरियड्स ऑफ ग्रोथ हमने क्यों डिफाइन किया? बिकॉज एक अडोलसेंट बच्चा इन्फेंट से बहुत अलग है, एक इन्फेंट टॉडलर से बहुत अलग है, टॉडलर स्कूल गोइंग एज से बहुत अलग है. इसलिए हमने एक स्पेसिफिक periods of growth define किए हुए हैं, because there is ever changing physiology as we grow. ठीक है, तो broadly 3 parts में हमने divide किया हुआ है इन periods of growth को: antenatal time, postnatal time and adolescent time. Antenatal means before birth, postnatal means after the birth, and then we have adolescent age group. तो पहले antenatal period देखते हैं. Antenatal, firstly what happens? There is fertilization of the ovum और zygote बनता है. So when do you label this product of conception as fertilized ovum or zygote? In the first 2 weeks, 0 to 2 weeks, you are going to label this product of conception as zygote. After 2 weeks till 8 weeks, we label this as embryo. After that, 9 week onwards till birth, we will call this as fetus. तो कैसे याद करना है? बहुत सिंपल सा है, दिमाग नहीं लगाना, एग्जाम में कंफ्यूज नहीं होना. देखो जाइगोट में कैसे लिखते हैं? जाइगोट जेड ऐसे लिखते हैं, ये 2 जैसा है ना. तो ये रिमेंबर 2 वीक्स तक है जाइगोट. फिर एम्ब्रियो, ई फॉर एम्ब्रियो, ई फॉर 8 वीक्स तक है फिर एम्ब्रियो. ठीक है, फिर आता है फीटस, एफ की अगर आप मिरर इमेज करोगे ऐसे बनेगा, उसको ज्वाइन कर दो, मतलब 9 वीक ऑनवर्ड्स आपका फीटस. ठीक है, तो ये सिंपल ट्रिक है टू रिमेंबर जस्ट इन द एग्जाम कि कौन सा पीरियड किसके लिए है. अब एंटीनेटल के बाद हम मूव करते हैं टू द पोस्टनेटल पीरियड. पोस्टनेटल पीरियड को जाने से पहले एक बहुत इंपॉर्टेंट पीरियड है, दैट इज पेरीनेटल पीरियड. पेरीनेटल पीरियड में कुछ पार्ट आता है हमारे एंटीनेटल पीरियड का, कुछ पार्ट आता है हमारे पोस्टनेटल पीरियड का. पेरीनेटल पीरियड स्टार्ट होता है आपका 22 वीक्स ऑफ जेस्टेशन से टिल 7 डेज ऑफ लाइफ, तो इसमें दोनों कंपोनेंट आ गए मेरे, एंटीनेटल लाइफ भी आ गई एंड पोस्टनेटल लाइफ भी आ गई. दिस इज एन इम्पॉर्टेंट पीरियड फॉर योर एग्जाम. अब मूव करते हैं पोस्टनेटल पीरियड पे. एज सुन एज द चाइल्ड इज बॉर्न, वी कॉल दिस चाइल्ड एज अ नियोनेट. कब तक नियोनेट बोलते हैं बेबी को? 28 डेज ऑफ लाइफ तक, राइट. सो द फर्स्ट 28 डेज ऑफ लाइफ वी कॉल चाइल्ड एज नियोनेट. इस नियोनेटल पीरियड को भी डिवाइड किया हुआ है इनटू अर्ली नियोनेटल पीरियड एंड लेट नियोनेटल पीरियड. अर्ली नियोनेटल पीरियड इज द फर्स्ट 7 डेज ऑफ लाइफ, एंड आफ्टर दैट you have late neonatal period. Within 1 year जो भी बच्चा होता है उसको हम बोलते हैं इन्फेंट. देखो, तो अगर मैं स्टेटमेंट बोलूँ एवरी नियोनेट इज एन इन्फेंट, ट्रू, बिकॉज वो लेस देन 1 ईयर है. लेकिन अगर मैं बोलूँ एवरी इन्फैंट इज अ न्यूनेट, दैट इज फॉल्स, दैट इज फॉल्स, राइट. सो इन्फैंट जब भी टर्म बोल रहे हैं, इट डिनोट्स लेस देन 1 ईयर. Neonate is less than 28 days. Then we have something that is known as post neonate. Post neonate वो period हो जाएगा, ये वो infants हैं जो neonate नहीं है, right. So these are starting from 28 days of life till 1 year. Till 1 year. उनको नाम दे दिया हमने post neonate. These are the infants who are not neonates. Then after infancy comes toddler. T for toddler, T for 3. So 1 to 3 years of life is your toddler. After that comes preschool, 3 to 6. Then comes school going. S for school, S for 6. 6 to 12 is school going child. School going child. Right? After that comes adolescent period. Adolescent period. So adolescent period is 10 to 19 years of age पहले. अब recent घई बोलती है 10 to 20 years. 10 to 20 years. Right? इसको further divide किया हुआ है early, mid and late adolescent में. So the early adolescent period is 10 to 13 years. Then we have mid adolescent 14 to 16 years. And then 17 to 20 years is the late adolescent period. Late adolescent period. पार्थ डिफरेंट नहीं है. देखो इन्फैंट एक अम्ब्रेला टर्म है. अम्ब्रेला टर्म है मतलब 1 साल से जो छोटे बच्चे हैं उनको मैं बोलूँगी इन्फैंट. इसमें सब बच्चे आ जाएंगे, न्यूनेट भी आ जाएंगे, पोस्ट न्यूनेट भी आ जाएंगे. जस्ट उनको डिफ्रेंशिएट करने के लिए न्यूनेट को हम बोलेंगे फर्स्ट 28 डेज. बचे हुए जो बच्चे हैं वो पोस्ट न्यूनेट हैं सारे ही ये इन्फैंट है ना. न्यूनेट, एवरी न्यूनेट इज ऑल्सो एन इन्फैंट, एवरी पोस्ट न्यूनेट इज ऑल्सो एन इन्फैंट. जस्ट वो पीरियड ऑफ ग्रुप्स को डिफाइन किया हुआ है. दीज आर जस्ट डेफिनेशंस यू नीड टू रिमेंबर. इन्फैंट एक अम्ब्रेला टर्म है फॉर ऑल द पीपल लेस देन 1 year of age, राइट. एक ही बात है, अम्ब्रेला टर्म है ना, इन्फैंट के अंडर ही आ रहे हैं ये सब. न्यूनेट भी इन्फैंट है, पोस्ट न्यूनेट भी इन्फैंट है. Okay, so we are done with periods of growth. Now let's move to assessment of growth. Assessment of growth parameters. So हमने कुछ parameters तो पढ़ लिए हैं. कुछ parameters हमने क्या क्या पढ़े? हमने height पढ़ी, length, weight पढ़ा, हमने head circumference पढ़ी. अगर मैं आपको बोलूँ एक न्यूबॉर्न है जिसका weight is 3 kgs, that's fine. लेकिन अगर मैं बोलूँ एक 1 year old है जिसका वेट इज 3 kg, दैट इज नॉट फाइन. मतलब वेट एक ऐसा पैरामीटर है दैट इज डिपेंडेंट अपॉन एज. Similarly goes for length. If I say this is a newborn and the length is 50 centimeter, everything seems fine. But if I say it is a 1 year old and the length is 50 centimeter, that doesn't fit, right? So again, length is a age dependent parameter. So the 3 age dependent parameters we have studied, we have weight, we have length or height, and we have head circumference. All of these are age dependent parameters. Then we have age independent parameters. What are these age independent parameters? It includes mid upper arm circumference and skin fold thickness. Skin fold thickness. Right? So let's discuss now what all instruments we use, some of the images that are frequently asked in the exam. So firstly, let's go for the weight. Can you see? We are measuring the weight of this child. There is a polybag thela type situation. We have a weighing scale over here and a hook, an S-shaped hook to hang it. You will see this in most of the Anganwadis, right? So what is this? What is this instrument called as? So just look at the hook. This is an S-shaped hook, right? So ring a bell. This is spring balance, spring balance, spring balance, right? And then we have a device like this where we have a plate or a tray in which we make the infant lie down, and this scale over here shows me the weight. This is beam scale. This is beam scale. These are the older instruments that we use. Now we have sophisticated electronic weighing machines too, right, which look like the beam scale only. So these are the two important instruments for your exam. Now let's move to length. So there are two terms written over here, length and height. So how they are different? How do you measure length? You make the child lie down and then measure from head till the foot. This is length. And how do you measure height? You make the child stand like this and then measure from head to foot. So why we are taking length in some children? Why we are taking height in some children? So till 2 years of age, you are going to take length. And how do you measure length? By this device. What is this device? This is infantometer. Infantometer. So you can see in infantometer there are two ends. One is the head end, that is fixed. That is fixed. And you have this foot end which is mobile. Foot end which is mobile. So the examiner is holding an infant, and ideally you need two persons to record the length. One is going to hold the infant and other one is going to move or push this foot end, right? We straighten the legs of the baby and then measure the length. Why we are doing this till 2 years? At 1 year of age, a child can stand, right? If you look at the developmental milestones, by the end of 1 year, a child can stand without support. So why we are not making the child stand up and take the height? Why we are taking the length in 2 years of age, till 2 years of age? Why not 1 year? And also device also has a confusing name, infantometer, and you can think that I can use it just for the infants. So remember, till 2 years of age, there is physiological bowing of legs. Physiological bowing of legs. So because of this physiological bowing of legs, I need to make the child lie down and straighten the legs and then take length. Because अगर खड़े होके करेंगे तो because of that bowing, I can get less height, right? After 2 years, you're going to take the standing height of the child. And what is this device that we use to take the standing height? What is this device? This device is stadiometer. 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 Right? Stadiometer. So these are the two devices that you use for length and height. Now let's move to the age independent parameter, that is mid upper arm circumference. So how do you take this mid upper arm circumference? We take two bony points. So this is, if this is the hand of the child, we take two bony points. Above we take the acromion process and below the olecranon process, and at the midpoint of the line joining these two, we take this mid upper arm circumference. Why this is age independent? Because in the age group 6 months to 5 years of age, what happens? Have you ever seen an infant, a chubby infant, right? Most of the children before the age of 1, you will see they're very chubby looking. Why? Because they have a lot of fat, and eventually what happens, this fat is replaced by muscle. Fat decreases and the muscle mass increases. So the overall circumference of the arm of the child stays constant. That's why it is age independent in this age group, right? Initially there is going to be fat and then the fat reduces and the muscle mass increases. This is reversal of adiposity. So this happens in this age group. That's why mid upper arm circumference is an age independent parameter. And we use this mid upper arm circumference as a screening tool for malnutrition. Can you see this colorful tape over here? What is this colorful tape? Path is right. This is Shakir's tape, right? Shakir's tape. We use this Shakir's tape for screening, for community screening of malnutrition. Community screen of malnutrition. So can you see there are three zones? We have this red zone, then yellow zone, and then green zone. So what is this red zone? This red zone is less than 11.5 centimeters, and that denotes that the child has severe malnutrition. Severe malnutrition. Then you have yellow zone. This yellow zone depicts 11.5 to 12.5 centimeter. It denotes that the child has malnutrition. And after that you have this green zone. This green zone is above 12.5 centimeter. It denotes that the child is not malnourished, no malnutrition, right? So this is how you interpret this Sharkey's tape and how we utilize mid-upper arm circumference for the screening of malnutrition. Right? After that, let's go towards the second age independent parameter, that is skin fold thickness. What are we checking in skin fold thickness? We are essentially checking the thickness of the skin fold. Whenever we are checking the thickness of the skin fold, we are actually checking the subcutaneous fat. So again, this is going to be independent upon of the age. Can you see this instrument over here? We use this instrument, we make this pinch of the skin through this instrument and measure through this instrument what is the thickness. You must have seen this device in your physics lab also. This is nothing but Harpendon's caliper. Harpendon's caliper. Right? We use this Harpendon's caliper for measuring the skin fold thickness. If the skin fold thickness comes out to be less than 6 mm, this denotes that the child has severe malnutrition. And for normal, what is the normal? More than 10 mm is the normal. The most common site where you check the skin fold thickness is your triceps fold. Triceps fold. And apart from that, you can check on the other sites like subscapular region, intrascapular region, biceps, right? Subscapular, interscapular. The most common site, just remember, it's the triceps. The triceps. You check at the triceps, the skin fold thickness, right? So with that, we covered the important parameters for assessment of growth. Now let's move towards another important topic of general pediatrics, that is puberty and adolescence. So in puberty and adolescence, the examiner is fond of asking 2-3 things only, and we are going to discuss that. The very first thing, the sequence of puberty. So the very easy mnemonic for both males and females is TPM. T females, initially there is thelarche, that means enlargement of your mammary glands, thelarche. Then comes P, that is pubarche, increase in the pubic hair. And lastly there is menarche, that is onset of menses, menarche, right? In males, T here is for testicular enlargement, testicular enlargement. Then P is again for pubarche, pubarche. Also penile growth. Lastly, M is manly. That means there will be appearance of the secondary sexual characters like beard, your voice changes, all of those things that we associated with manly, right? So beard appearance, voice changes, all of these will come last, right? With this sequence, now let's move to some of the one-liners that the examiner asks. So firstly, what is the first biochemical evidence of puberty? So remember GnRH pulse. GnRH pulse is the first biochemical evidence. But if they ask what is the best marker of puberty, then LH is the best marker of puberty. If LH is not given, then choose FSH. FSH, right? For assessment and staging of puberty, we have something that is known as the Tanner staging. This is also known as SMR, that is sexual maturity rating. Sexual maturity rating. The examiner asks on which stage, which Tanner stage there is growth spurt in females and males. So remember, in females the growth spurt happens earlier. In stage 3 growth spurt happens. In males it happens later. In stage 4 growth spurt happens in males, right? So with that knowledge, just revise your Tanner staging for males and females. So this is the Tanner staging. Let's first look at the male staging. Very easy to remember. In stage 1, everything is going to be pre-adolescent, infantile like, right? So no features of sexual maturity would be there. So pre-pubertal would be stage 1, and stage 5 will always be adult like. That means the full growth of your gonads and the pubic hair will be on the medial thigh as well, right? So this is the adult feature. In both males and females, this will stay constant. So you have now learned two stages for sure. One is always going to be prepubertal, and 5 is always going to be adult like, and the pubic hair will also be on the medial thigh, right? So first stage is prepubertal. In males, remember, prepubertal means the testicular volume will be less than 4 ml. Testicular volume is will less than 4 ml. Once it crosses the 4 ml, that means there is onset of puberty. Now the growth has started, right? So in stage 2, what is going to happen? There is increase in the testicular volume, more than 4 ml. And also the pubic hair will come now, but the pubic hair are very sparse. Can you see small pubic hair? So very sparse pubic hair are going to be there. In stage 3, now the pubic hair will grow in size. They will coarsen and curl. So there will be coarsening and curling of the pubic hair, and also there is increase in the penile length, right? So firstly the penile length increases, and finally in stage 4 the penile girth will also increase. There will be increase in the penile girth or width. And as we discussed in stage 4, the growth spurt also happens. And finally in stage 5 there will be a adult like feature. That means hair will be on medial thigh as well. Medial thigh as well. A very important feature that separates your stage 4 and stage 5 is stage 4s can sometimes look exactly like adult, but the only difference would be hair won't be on medial thigh. In stage 5, hair will be on the medial thigh. And finally, in stage 5, the adult testicular volume, that is 25 ml, is achieved. 25 ml is achieved. So these are the important points that are asked in the exam, right? Get another point regarding the puberty in males. They ask, when does scrotal reddening happens and when does scrotal darkening happens? So scrotal reddening happens in stage 2 and darkening happens in stage 4, right? So remember these points. Scrotal reddening is stage 2 and darkening is stage 4, right? Important points. Now let's move towards this female tanner staging. So in female, again the same rule is going to apply. First stage will be prepubertal and the final stage would be adult like, where you are going to see that the pubic hair are also on the medial side of the thigh. So prepubertal, and finally in stage 5, adult like, where the hair will be on the medial thigh as well, right? Medial thigh as well. Now let's move to stage 2. In stage 2, what happens? In chest, there will be appearance of the breast bud, and again there will be sparse pubic hair. In stage 3, now the breast bud will also increase and pubic hair will coarsen and curl. And as we have discussed, that in stage 3, growth spurt happens. In stage 4, pubic hair would be adult like, except they will be on the medial thigh. And now in stage 4, the contour separation happens of the breast and secondary mound formation is seen. You understand secondary mound formation? This is the breast tissue, areola and the nipple. This is the secondary mound formation that happens. Okay, so this is how you learn about the tanner staging and the important points for your exam. Now that we have covered the general pediatrics normal things, we have covered how the normal puberty happens, what are the stages, what is length, weight, head circumference and all of these things. Now let's move towards the abnormal pattern. So we are going to discuss the very important and very favorite topic of examiners, that is short stature. So what is short stature? Short stature. So when are you going to label that this child has short stature? Whenever the height for age and sex is less than -2 standard deviation or less than third percentile, this child has short stature, right? So with short stature, very important concept comes target height. What is target height? I am short because my parents are short, right? Genetics play a very important role when it comes to your height. So what is the target height of an individual? Target height definitely depends upon the height of the mother and father. So how do you calculate target height? Take the height of mother and father, average it out, and if it is a boy, you add 6.5 centimeter, and for a girl, you minus 6.5 centimeter. This gives you a target height. This target height is the height of the expected height of the child, right? So this is target height, right? So examiner is going to give you mother's height, father's height, and ask you to calculate target height of that child. If it is a boy, add 6.5 centimeter. If it is a girl, subtract 6.5 centimeter. With that, now let's move to types of short stature. We have two major types broadly. So short stature can be physiological short stature or it can be pathological short stature. फिजियोलॉजिकल क्या है? जो एक तो मैंने आपको बताया, आई एम शॉर्ट बिकॉज माई पेरेंट्स आर शॉर्ट, दैट मीन्स familial short stature. That short stature is running in the family, right? This child is going to have short parents, that's why the overall height of this child, the final height of the child is also less, right? जब भी हम population distribution की बात करते हैं, there is bell shape curve. तो कुछ नॉर्मल लोग भी तो होंगे ना जो अनफॉर्चुनेटली इस स्पेक्ट्रम में है. तो these are nothing but familial short stature. इनको कोई प्रॉब्लम नहीं है. दिस इज जस्ट फिजियोलॉजिकल शॉर्ट स्टेचर. सेकेंड कैटेगरी आती है वो बच्चे जिनका थोड़ा डिले होता है. दिस इज नोन एज कॉन्स्टिट्यूशनल डिले इन ग्रोथ एंड प्यूबर्टी. Their parents also have such history that they attained height sometime later, they attained puberty sometime later, right? These are usually short children till class 9th or 10th, right? And suddenly one fine day, summer vacations of class 10th, and they come back and their height is too much, right? So from the first two position in the assembly, they move to the last two position of the assembly, right? So this is how the growth spurt happens in these. The puberty is slightly delayed, comes slightly later on, राइट. सो दिस इज कॉन्स्टिट्यूशनल डिले इन ग्रोथ एंड प्यूबर्टी. तो फिजियोलॉजिकल शॉर्ट स्टेचर में दो चीज़ें हैं, फेमिलियल शॉर्ट स्टेचर एंड कॉन्स्टिट्यूशनल डिले इन ग्रोथ एंड प्यूबर्टी. यहाँ पेरेंट्स की भी हिस्ट्री होती है कि उनको भी जो प्यूबर्टी हिट की थी वो थोड़ी लेटर ऑन हिट की थी. तो यही दोनों फिजियोलॉजिकल कॉजेज ऑफ शॉर्ट स्टेचर को हम डिस्कस करते हैं. वी हैव फर्स्टली द कॉन्स्टिट्यूशनल डिले इन ग्रोथ एंड प्यूबर्टी एंड देन वी हैव फेमिलियल शॉर्ट स्टेचर. दोनों हम डिस्कस कर चुके हैं. तो वन बाय वन लेट्स जस्ट कैपिटुलेट द डिफरेंसेस बिटवीन द टू. फर्स्टली इन कॉन्स्टिट्यूशनल डिले इन ग्रोथ एंड प्यूबर्टी, व्हाट विल बी द हिस्ट्री विल बी delayed puberty in parents. The final height of these children will be normal, obviously, right? Sooner or later they are going to catch their height. Puberty here is delayed. On the other hand, in familial short stature, there will be history of short parents. The final height, unfortunately, here is going to be short only. Their puberty is normal, puberty at the normal time, right? Now, when we differentiate constitutional delay in growth and puberty and short stature, there is an important concept of height age, bone age, and chronological age. अभी क्या होती हैं तीनों चीज़ें? So we have height age. हाइट एज मतलब अगर मैं आपको बोलूँ द हाइट ऑफ द चाइल्ड और द लेंथ ऑफ द चाइल्ड इज 75 सेंटीमीटर, ठीक है? तो आप थोड़ा पीछे जाओ, हमने पढ़ा था बर्थ पे 50 होती है और 1 ईयर पे 75 होती है. तो यू कैन टेल ना दिस चाइल्ड मस्ट बी अराउंड 1 ईयर क्योंकि इसकी लेंथ जो है वो 75 है. सो दिस इज नथिंग बट हाइट एज. सो हाइट एज इज दैट एज एट विच दैट पर्टिकुलर हाइट इज एट 50th सेंटाइल, राइट? तो तो अगर मैं वो आपको बोलूँ 75 सेंटीमीटर, आप बोलोगे 1 ईयर, राइट? सो दिस इज नथिंग बट हाइट एज. देन यू हैव क्रोनोलॉजिकल एज. क्रोनोलॉजिकल एज तो बहुत सिंपल है, दैट इज द एक्चुअल एज ऑफ द चाइल्ड. सो आज कौन सी तारीख है? आज है 19th August 2026. So if I say a child was born on 19th August 2025, so what is the chronological age? आज उसका birthday, first birthday. So the chronological age is 1 year. So chronological age is nothing but the age since the birth. That is chronological age. And lastly, you have something that is known as bone age. Bone age क्या होती है? We take the X-ray of the child, compare it to the atlas कि कितने ossification centers आपको मिलते हैं that particular age पे. Depending upon the number of ossification centers, we label the child of that particular age. Compare करते हैं हम different X-rays से उस atlas को कि कितने ossification centers होने चाहिए. So that is bone age. सो अगर प्यूबर्टी ऑन टाइम है तो ये हमें बोन एज जो है नॉर्मल दिखेगी, लेकिन अगर प्यूबर्टी डिलेड है तो अभी ऑसिफिकेशन सेंटर फ्यूज नहीं हुए होंगे तो हमें बोन एज कम दिखेगी, राइट? सो वही कॉन्सेप्ट यहाँ लगता है. तो सबसे पहले हाइट एज की बात करते हैं. हाइट एज तो दोनों में ही कम है, दोनों ही शॉर्ट स्टेचर के साथ आए हैं, राइट? सो हाइट एज तो दोनों की कम है, दोनों की हाइट अपनी एज के हिसाब से कम है. What about bone age? Constitutional delay में bone age would be less because अभी puberty delay है. On the other hand, bone age will be normal in familial short stature, right? Now the examiner asked the relationship between this chronological age, height age and bone age. So very simple. देखो दोनों ही सिनारियों में हाइट एज इज डेफिनेटली लेस देन क्रोनोलॉजिकल एज. जो एक्चुअल एज है बच्चे की उससे तो कम ही है उस बच्चे की हाइट. सो क्रोनोलॉजिकल एज से हमेशा कम है हाइट एज. हाइट एज इज लेस देन क्रोनोलॉजिकल एज, राइट? सॉरी उल्टा लिखते. हाइट एज, chronological age is less than the height age. ये तो डिक्टम है ही. हाइट एज इज लेस देन chronological एज. अब देखो दोनों में ही कॉन्स्टिट्यूशनल डिले में दोनों ही लेस है, हाइट एज भी लेस है, क्रोनोलॉजिकल एज भी लेस है, तो यहाँ पर आ जाएगा आपका बोन एज. ऑन द अदर हैंड यहाँ पे बोन एज नॉर्मल है तो यहाँ पर आ जाएगा आपका बोन एज, राइट? सो वेरी सिंपल. आप ये तो ध्यान में रखना है हमेशा कि मेरी हाइट एज कम है एज कम्पेयर टू क्रोनोलॉजिकल एज. बोन एज में अब कहाँ बोन एज कहाँ कौन सी डायरेक्शन में लगाना है वो आप डिसाइड कर लोगे, राइट? सो दिस रिलेशनशिप इज वेरी इंपॉर्टेंट. नाउ लेट्स मूव टू पैथोलॉजिकल शॉर्ट स्टेचर. एंड पैथोलॉजिकल शॉर्ट स्टेचर में हम पहले डिस्कस करेंगे डिस्प्रोपोर्शनेट शॉर्ट स्टेचर. डिस्प्रोपोर्शनेट शॉर्ट स्टेचर. So whenever we are measuring the height of the child, एक बहुत important ratio होता है, that is the upper segment lower segment ratio. So if this is the child out here and I am taking the height of this child, बहुत important ratio है. What is the upper segment? Upper segment is from the top of the head till pubic symphysis. This is upper segment. और बचा हुआ हो गया हमारा लोअर सेगमेंट. अगर हाइट में से आप अपर सेगमेंट माइनस कर दोगे दैट इज योर लोअर सेगमेंट. वाई दिस इज इम्पॉर्टेंट? क्योंकि हमारी जो डायरेक्शन ऑफ ग्रोथ होती है दैट इज सेफेलोकॉर्डल. मतलब पहले हेड ग्रो होगा, फिर मेरा ट्रंक ग्रो होगा, फिर मेरे लिम्स ग्रो होंगे, सेफेलोकॉर्डल. तो एट द टाइम ऑफ बर्थ ऑब्वियसली मेरी अपर सेगमेंट बड़ी होगी, लेकिन जैसे ही बच्चा ग्रो करेगा उसके लिम्स ग्रो करेंगे, लोअर सेगमेंट भी ग्रो करती रहेगी, तो ये रेशियो चेंज होता रहेगा विद एज. सो इनिशियली my upper segment is big, so my upper segment lower segment ratio would be high, and as the child ages lower segment will increase, so this ratio will decrease. So normal upper segment lower segment ratio की बात करें तो at the time of birth it's 1.7:1. By the age of 3 years it becomes 1.3:1. By the age of 6 years it becomes 1.1:1. At 10 years it becomes 1:1, and after that the adult ratio of 0.9:1, या फिर हम इसको लिख सकते हैं 1:1.1 is reached, राइट? अब ये रेशियो हमारे लिए क्यों इम्पोर्टेंट है? क्योंकि अगर ये रेशियो अपनी एज के हिसाब से सेम नहीं आ रहा है तो मतलब कुछ तो अफेक्टेड है, कुछ डिस्प्रपोर्शनेटली छोटा है ये बच्चा, या तो इसकी ट्रंक कुछ गड़बड़ है या तो इसके लिम्स कुछ गड़बड़ है। तो वही देखते हैं हम। दो सिनारियो है हमारे पास। पहला सिनारियो इज कि अपर सेगमेंट लोअर सेगमेंट रेशियो इंक्रीज्ड है। मतलब अगर मैं आपको बोलूँ एक 3 साल का बच्चा है, लेकिन उसका अपर सेगमेंट लोअर सेगमेंट रेशियो मेरे पास 1.4 आ रहा है, मतलब अपने रेशियो से ज़्यादा आ रहा है। इसका क्या मतलब है? इसका मतलब इस बच्चे का अपर सेगमेंट बड़ा है और लोअर सेगमेंट छोटा है। दैट मीन्स दिस चाइल्ड हैज़ big upper segment and short lower segment. That means big trunk and short limbs. So this is nothing but short limb dwarfism. और जब भी आपको ये शॉर्ट लिम्ब डॉर्फिज्म दिखे, इंक्रीज्ड अपर सेगमेंट लोअर सेगमेंट रेशियो दिखे, थिंक अबाउट दीज 3 कंडीशंस। ये सपने में भी आपको नींद से भी कोई जगा के पूछे कि कौन सी 3 कंडीशंस है वेयर यू सी शॉर्ट लिम्ब डॉर्फिज्म, यू शुड बी एबल टू टेल दीज 3 कंडीशंस। व्हाट आर दीज 3 कंडीशंस? दीज आर rickets, achondroplasia and congenital hypothyroidism. Congenital hypothyroidism, right? So these are the conditions with increased upper segment lower segment ratio or short limb dwarfism. On the other hand, अगर उल्टा हो जाता है, एज के हिसाब से आपको कम मिल रहा है अपर सेगमेंट लोअर सेगमेंट रेशियो। फॉर एग्जांपल अगर ये 3 साल का बच्चा और इसका अपर सेगमेंट लोअर सेगमेंट रेशियो 1:1 आ रहा है, मतलब अपनी एज से कम आ रहा है। इसका क्या मतलब हुआ? अगर एज से कम आ रहा है अपर सेगमेंट लोअर सेगमेंट रेशियो, मतलब अपर सेगमेंट छोटा है, मेरी ट्रंक छोटी है, देयर इज शॉर्ट ट्रंक डॉर्फिज्म। शॉर्ट ट्रंक डॉर्फिज्म। और इस शॉर्ट ट्रंक डॉर्फिज्म का मतलब क्या है? आपकी ट्रंक किस चीज़ से बनी है? मेरी स्पाइन है, रिब्स है, मेजरली वही है ट्रंक का पार्ट। तो जब भी आपकी स्पाइन, आपके वर्टिब्रास अफेक्टेड होंगे, यू विल सी दिस शॉर्ट ट्रंक डॉर्फिज्म। सो एनी वर्टिब्रल anomalies, if any, TB spine, that is spot spine, if there are any storage problems, mucopolysaccharidosis, spondyloepiphyseal dysplasias, in all of these conditions you will see affected trunk, short trunk dwarfism, or decreased upper segment or lower segment ratio, right? So this is about short stature assessment. Now let's move to the second aspect, that is developmental milestones, right? ये developmental milestones बहुत important है। Just before the exam please go through these charts, because ये ऐसे factual points हैं, और अगर एग्जामिनर पूछ लेता है, you should not be the one marking wrong or thinking कि मैंने last time पे क्यों नहीं revise किया, right? So firstly let's move towards वन बाय वन ईच ऑफ द डोमेन्स। पहले स्टार्ट करते हैं विद ग्रॉस मोटर माइलस्टोन्स। ग्रॉस मोटर मतलब ग्रॉस बॉडीली मूवमेंट्स। सबसे पहले 3 मंथ्स पे द चाइल्ड अचीव्स हेड कंट्रोल, and then child starts to sit. Firstly the child will sit with the support, that means in tripod position at 6 months of age, and then without support, sit without support at 8 months of age. At 9 months, the child will try to stand with support. And by 12 months, the child stands without support. So, मतलब by the 12 months, मतलब अपने first birthday पे the child will stand independently and cut the cake. But what about walking? The child will walk with the support over here. Walk with support. तो अपने गिफ्ट्स लेने के लिए, देने के लिए उसको वॉक सपोर्ट के साथ करना पड़ेगा अपने पेरेंट्स के। देन 10 मंथ्स बीच में आता है। इस 10 मंथ्स में बच्चा स्टार्ट्स क्रीपिंग और क्रूजिंग। क्रीपिंग एंड क्रूजिंग। क्रीपिंग एंड क्रूजिंग। क्या होता है क्रीपिंग एंड क्रूजिंग? क्रीपिंग आप समझते हो, मूविंग ऑन 4 लिम्स। मूविंग ऑन 4 लिम्स, तो बच्चा ऐसे 4 पैर पे अपना चलेगा, दिस इज क्रीपिंग। अगर एब्डोमेन टच कर रहा है ग्राउंड पे, दिस इज क्रॉलिंग। अगर एब्डोमेन लिफ्टेड ऑफ है ग्राउंड से, दिस इज क्रीपिंग। सो क्रॉलिंग जिसमें एब्डोमेन टच कर रहा है वो आता है 8 मंथ्स पे, and creeping जहाँ पे abdomen lifted off है ground के, that comes at 10 months of age, right? नहीं पढ़ा था 12 months पे walk with support आएगा, walk without support comes at 15 months. उसके बाद बच्चा भागेगा, उसके बाद भागेगा 18 मंथ्स पे, जैसे हम घर से 18 ईयर्स पे जैसे सब भागने लग जाते हैं ना, अडल्ट हो गए अब तो हम आज़ाद हैं, भागते हैं घर से सब, वैसे ही 18 मंथ्स पे भी बच्चा रन करना लग जाता है, रनिंग, एक्सप्लोरिंग वो सब शुरू हो जाती है, राइट? By 2 years of age, the child will be jumping, climbing the furniture. 3 years पे child would be riding a 3-tire cycle, tricycle. 4 years पे child hops and skips on one foot, and finally the child will skip on both the feet by 5 years of age, right? So these are the important gross motor milestones. Gross motor milestone में एक stairs का milestone है जो एग्जाम में बहुत पूछता है, और इसको मैंने सेपरेटली इसलिए बनाया है ताकि आप मेन टेबल में कंफ्यूज ना हो जाओ, ये stairs milestone आपको याद रहने चाहिए. So what is easier when we climb up and down the stairs? Going upstairs or downstairs? Remember, upstairs jana is easier, downstairs coming is difficult. So initially what the child does? Child goes upstairs, learn to go upstairs, then downstairs. पहले child uses both the feet on one step, मतलब एक step पे दोनों पैर रखेगा, फिर दूसरे step पे दोनों पैर रखेगा, ऐसे करके ऊपर चढ़ेगा. That means 2 feet on one step. And thereafter the child learns to use 1 feet per step, जैसे हम चलते हैं as adults. So initially 2 feet on one step, the child learns to go upstairs and downstairs by 2 years of age. After that, the child will learn to go upstairs with 1 feet per step. This is by 3 years of age. And finally, by 4 years of age, the child will learn to come downstairs as well. Right? So 2, 3, 4 याद रखना. पहले 2 feet on one step, upstairs and downstairs. Then 1 feet per step upstairs, and then 1 feet per steps downstairs. So this is the important stairs milestones. Now let's move to your fine motor milestones. Fine motor milestones. So to attain new milestones, the child should lose the primitive reflexes. So one of the primitive reflexes is palmar grasp. If you stroke the palm of the child will involuntarily grasp it. This is palmar grasp. अब ये palmar grasp जब जाएगी तभी बच्चा voluntarily grasp कर पाएगा चीज़ें. So ये palmar grasp जाती है around 3 to 4 months, और उसी टाइम पे बच्चा can voluntarily grasp the toy or rattle, और फिर ग्रास्प करने के बाद वो माउथिंग शुरू कर देता है बाय 4 मंथ. जो भी हाथ में आया उसको पसंद आया वो अपने मुंह तक ले जाएगा, राइट? बाय 5 मंथ्स द चाइल्ड हैज़ बायडेक्स्ट्रस ग्रास्प, मतलब दोनों हाथों से बच्चा ऐसी चीज़ों को पकड़ेगा. फिर उसको रियलाइज होगा मैं एक हाथ से भी पकड़ सकता हूँ. देन कम्स यूनिडेक्स्ट्रस ग्रास्प बाय 6 months. Then the child will realize God has given me this thumb and I can hold objects like this with my thumb and finger, right? So the child will have pincer grasp by 9 months. This pincer grasp is initially immature pincer grasp. That means the child will need support of the accessory fingers as well. But this okay sign will be the mature pincer grasp that the child attains by 12 months of age. So now the child has good pencil grasp, the child can hold a pencil and scribble. So by 15 months, the child will start scribbling. Then the child will be making different things. Firstly, 2 years of age, the child will make this line. The line has 2 points, right? So remember, 2 years of age, line. Then 3 years, the child will make a circle. 4 years, the child will be able to make a cross. 4.5 years, the child will be able to make this rectangle. Triangle at 5 years. Diamond at 7 years. Cylinder at 9 years. And cube at 11 years of age. Right? So these are the drawing skills that the child achieves. Now in fine motor skills, fine motor हम समझते हैं अपना fine distal actions है. तो इसमें एक ऐसा एक्शन है जो हम डेली यूज करते हैं, दैट इज ड्रेसिंग अनड्रेसिंग, राइट। सो ये बहुत ही इंपॉर्टेंट फाइन मोटर स्किल है। ड्रेसिंग अनड्रेसिंग में क्या ज़्यादा ईजी लगता है आपको, ड्रेस अप करना या अनड्रेस करना? अनड्रेसिंग इज ईजियर। तो पहले चाइल्ड अनड्रेसिंग सीखता है, अनड्रेसिंग करेगा विदाउट हेल्प, फिर ड्रेसिंग करेगा विद हेल्प, without help, right? So this happens one by one, right? So undressing happens first and then dressing. पहले with help सीखेगा, फिर without help सीखेगा. So सबसे पहले undressing आता है with help, this comes by 2 years of age. Then come dressing with help, 3 years of age. Then dressing undressing without help, 5 years of age. और ड्रेसिंग अनड्रेसिंग में भी सबसे इंट्रिकेट फंक्शन होता है बटनिंग अनबटनिंग. सो पहले अनबटन करना सीखता है 3 years and then button करना सीखता है 4 years. So remember the sequence 2, 3, 5. This is about undressing and dressing, और unbutton button 3, 4 years, right? So मैंने ये टेबल सेपरेट बनाया ताकि मेन टेबल में आप मिक्स मत करो और फटाफट आपको स्ट्राइक कर जाए एग्जाम में जब ये क्वेश्चंस एग्जामिनर आपको पूछ रहा है. After this gross motor and fine motor milestones, now let's talk about the social milestones. Social milestones मतलब बच्चा आसपास अपनी सोसाइटी में कैसे इंटरैक्ट कर रहा है. Initially, the child has this social smile. That means he will respond to everyone with a smile. This is social smile. This comes at 2 months of age. Then the child will realize, I have to give special regard to my mother. She gives me milk. She cares for me. So by 3 months, the child will have mother regard. Mother regard. At 6 months, the child looks in the mirror and play with it. So now the child has seen self also. Mirror regard, mirror play. So now the child knows the mirror, child knows the mother. So everyone around who is a new face, who is a stranger, will give him anxiety. So the stranger anxiety comes by 7 months. So stranger anxiety will come by 7 months. Then comes 9 months, which is a very important milestone. Here object permanence comes. Object permanence comes. Object permanence means before 9 months, what happens if you show a toy to the child and then hide it? The child will not look for it because now it is not in the universe. Whatever is in the front of the child is a part of his universe. Whatever is not in his visual field, that is not in the universe. So what a good place to stay, right? किसी की याद नहीं आएगी, जो दिख रहा है बस वही उसी प्रेजेंट में जी रहे हैं. बट 9 months में ऑब्जेक्ट परमानेंस आ जाती है. द चाइल्ड नाउ रिकॉग्नाइजेस दैट इफ यू हैव हिडन समथिंग, इट इज स्टिल देयर इन दिस यूनिवर्स. ही विल ट्राई टू फाइंड इट. सो इफ यू आर हाइडिंग अ टॉय बिहाइंड योर बैग, द चाइल्ड विल ट्राई टू लुक फॉर इट. दिस इज ऑब्जेक्ट परमानेंस. एंड एट दिस टाइम ओनली द चाइल्ड विल लर्न टू से बाय बाय, वेव बाय बाय, right? So at this 9 months, the child will waves bye bye. Right? Now after that, all the drama will start. The child will start imitation, mimicry, right? So imitation, mimicry of the acts, all of these come. Domestic mimicry, that means copying the day-to-day actions, that will come by 18 months. By 3 years, the child can tell you 3 things, that is name, age and sex. Lastly, at 4 years, the child can do left to right discrimination. Left to right discrimination. In the social milestones, interaction and playing is very important. So at 15 months, the child hugs the parents. 18 months expresses love by kissing the parents. And obviously now the child is too attached. The child will have this separation anxiety when mother and father will go to work. The child is going to have this separation anxiety. And the child will realize now I have to go out and play. So by 2 to 2.5 years of age, he will go out and do parallel play. What is this parallel play? Two child sitting together, but they're not playing together. Each of them is involved in their different task. Remember the first day of your college, you must be sitting with your best friend, but at that time you were strangers, so you were involved in your phone, he was involved in his phone or doing something else. This is parallel play, that they are sitting together but they're not playing together, they're not interacting, they're just sitting together and playing on their own. After that, there will be cooperative play where they will be interacting. So this comes around of 3 years of age, there is cooperative play. And finally, by 4 years of age, there will be this group play or role play. Group play or role play. Right? So these are about the important social milestones. And finally, let's wrap up with language milestone. That is how the child uses the voice to interact. Firstly, what happens? The very first milestone, the very first words that come out the mouth of the child, these are the cooing sounds, right? So these cooing sounds come at 2 months of age. Then at 4 months of age, the child will laugh out loud. 6 months, the child says monosyllables like ma, ba, da. Then the child joins to form, say, bisyllables, that like mama, papa, dada. By 9 months of age, the first real word, that means the meaning of that word child knows, the first real word the child will say on its first birthday, that is 12 months of age. By the age of 15 months, the vocabulary would be of 4 to 6 words. 18 months, it would be 8 to 10 words. Then by the end of 2 years, vocabulary will increase very rapidly, 50 to 100 words. And by the end of 2 years of age, the child can say 2-word sentences like khana do, pani do, give water, give food. So these 2-word sentences the child can form by 2 years of age. At 3 years, the child can use pronouns like I, me, you, tum, main, aap, I, me, you. Right? By 4 years now, the child would be able to tell you story, poem. And how does the story starts? Once upon a time, that means the child can also refer into the past tense. So here the child will be able to use past tense कि ऐसा हुआ था, ऐसे गए थे. And 5 years, the child will now be using future tense कि हम कल जाएंगे, करेंगे, ये सारी चीज़ें उसको समझ आने लगेंगी. And also the child will be asking about the meaning of the words. Meaning of the words. Right? Okay, so this is how language milestones happen. Now there are two special milestones, vision milestones and hearing milestone. Vision आपके लिए बहुत important है. So vision milestone में क्या करते हैं, एक red dangling ring ऐसे लेंगे और बच्चे को वो ring follow करनी है. So we keep this ring around 8 to 10 inches from the child. If the child can follow up till 45 degrees, this is a newborn. Till 90 degrees, this is a 1-month-old child. And by the end of 3 months, the child can follow up till 180 degree. Another important MCQ point, when does binocular vision come? Binocular vision comes at 4 months of age. 4 months of age, right? So this is all about the general pediatrics growth and developmental section. In general pediatrics, yet another important topic is nutrition, and we are going to start nutrition next. If there is any doubt regarding this growth and development, please drop that down in the comment section and we'll cover that right away. Otherwise, let's start nutrition straight away. Okay, so let's start with nutrition then. So with nutrition comes a very important topic, that is micronutrients in health. That means your vitamins and minerals, important ones, right? So when we talk about vitamins and minerals, PDF will be available on app. This class will stay for will be done 2.5 to 3 hours maximum, and yes, this will be available on YouTube thereafter once the live ends, and you'll get notes on the app. So let's start with vitamins and minerals. Vitamins, you have two types: water soluble and fat soluble. Water soluble vitamins are B complex and vitamin C. Fat soluble are A, D, E, K. So one by one we are going to discuss each of them. Let's start with water soluble vitamins. The very first vitamin, B1, that is thiamine, right? That is thiamine. So deficiency of this thiamine causes beriberi. This beriberi can be dry beriberi and wet beriberi. Dry beriberi, your nerves are affected, and wet beriberi, your cardiac tissue is affected. That's why wet, there is cardiac failure, right? And in alcoholics, you see this thiamine deficiency, where there is a beautiful name of a syndrome, that is Wernicke-Korsakoff syndrome. And what do you see in this Wernicke-Korsakoff syndrome? You see this triad, and you can remember this triad by the mnemonic GOA. In GOA, you have alcohol, right? So alcoholics have this Wernicke-Korsakoff syndrome, deficiency of thiamine. G stands for your global confusion. O is ophthalmoplasia, and A is ataxia, right? Now let's, after B1, let's move to B2, that is riboflavin. So ribo means red, right? So everything will turn red over here. The tongue turns red, जिसको बोलते हैं magenta colored tongue. You can see the inflammation around the mouth. You see angular stomatitis, ciliitis, and also red vessels. So you can see red vessels sprouting in the cornea. Can you see red vessels sprouting in the cornea? This is known as neovascularization of cornea. So all of these things you see in the deficiency of vitamin B2. Then comes B3, that is niacin. Very important for your exam. This niacin deficiency causes the 3D disease, that is pellagra. This pellagra is known as 3D disease. What are these 3Ds? We have diarrhea, dementia, and dermatitis. So you have diarrhea, dermatitis, and dementia. And this particular rash is examiner's favorite. Can you see a typical rash in the exposed areas? Right, this is a photosensitive rash, and the typical rash that you see around the neck, this is known as Casals necklace. Right? After B3, there's nothing like B4. We have B5, which is known as pantothenic acid. And how do you wear pants? From your foot. So here the deficiency causes burning foot syndrome, right? Severe pain in the foot, very tingling, burning pain in the foot. So that's why this is also known as nutritional melalgia. After B5, we have B6, that is pyridoxine. Pyridoxine में a very important thing that you need to remember, that this pyridoxine deficiency is a very important cause of neonatal seizure. So neonatal seizure में आप जब सारी treatment दे देते हो, antiepileptic दे दिया, सारे metabolic problems देख ली, still the seizure is not stopping, we give a trial of pyridoxine, because pyridoxine deficiency can be a cause of neonatal seizures. Then comes B7. B7 is biotin. स्किन केयर एंथुजियास्ट को पता है बायोटिन कितना इंपॉर्टेंट है. बायोटिन सप्लीमेंट्स बहुत यूज करते हैं ना फॉर हेयर फॉल्स, गुड स्किन. सो दिस बायोटिन डेफिशिएंसी इज नोन टू कॉज एलोपेशिया एंड डर्मेटाइटिस. There's nothing like B7. Then we have B9, which is folate, and we all know this folic acid deficiency in mothers, and finally in the baby can lead to neural tube defects and megaloblastic anemia. B12 is cobalamin, and again it causes megaloblastic anemia and subacute combined degeneration. So this was all about the B complex vitamins. Another water soluble vitamin is vitamin C, known as ascorbic acid. Ascorbic acid, vitamin C, is very important in कोलाजन सिंथेसिस. सो कोलाजन तो बहुत इंपॉर्टेंट कनेक्टिव टिश्यू हमारी ब्लड वेसल्स में है, स्किन में है, हर जगह है कोलाजन, है ना. तो बिकॉज ऑफ दिस डेफिशिएंसी ऑफ प्रॉपर कोलाजन फॉर्मेशन यू विल सी ब्लीडिंग, you will see सब perifollicular bleeds, gum bleeds, you will see this corkscrew hair with perifollicular redness, bleeding in the gums, and बोन्स में ये टिपिकल इफेक्शन होता है जो आपको एक्सरे दिखेगा. सारे पॉइंटर्स दिख रहे हैं आपको, सब पेरियोस्टियल हेमरेज, कॉर्बिटिक जोन, रिंग साइन, क्रमरफेल्ड जोन, पेल्कन स्पर, ऑल ऑफ दिस ये आपने ऑर्थो में भी पढ़ा होगा, है ना. सो ऑल ऑफ दीज आर द इम्पॉर्टेंट पॉइंट्स फॉर योर एग्जाम. After water soluble, now let's move to the fat soluble vitamins. In fat soluble, let's talk about firstly vitamin A. Deficiency of vitamin A बहुत सारी जगह पे affect करेगा. सबसे पहले skin, it leads to dryness, phrinoderma, toad-like skin. In eyes, you see different signs. So they ask about the first symptom and the first sign. Symptom क्या होता है जो patient आपके पास लेके आ रहा है, sign क्या है जो आप detect कर रहे हो. So what is the first symptom with which this patient comes? That's night blindness. Night blindness. Night blindness. फर्स्ट आई साइन क्या है जो आप पिक कर रहे हो? दैट्स ड्राइनेस ऑफ द कंजक्टिवा. सो conjunctival xerosis is the first sign. And then you have this WHO classification. This must be covered in opthal also. XN is for the night blindness, then comes X1, X2, 3, S and F. So XN night blindness है. X1A में you see conjunctival xerosis. B is this bite-dot spot. Can you see this white flake over the eye? This is bite-dot spot. B for bite-dot spot. X2, now you see corneal xerosis. Then you see corneal ulcer. If this ulcer is covering less than 1/3 of the cornea, this is X3A. If it is covering more than 1/3 of the cornea, this is X3B. Then you have scarring, and lastly there are fundal changes. Right. So this is an important classification of vitamin A deficiency. Now let's move to vitamin D deficiency. If there is vitamin D deficiency in growing bones, we label it as rickets. Important clinical feature, what is the first sign that you see with vitamin D deficiency in pediatric population? It's craniotabes, that is ping pong skull. Have you seen a ping pong ball, the table tennis ball? If you make indent that ball, give a pressure over one part of that ball, you will see the indentation. Similarly, the same thing happens in the skull of the baby, the vitamin D deficiency baby. If you press hard over a point, you can notice the indentation. This is ping pong skull or craniotabes. After that, coming down to the chest, what do you see? You see pigeon chest. That is also known as carinatus, pectus carinatus. And you see rosary, that is rachitic rosary. And you see a sulcus, that is Harrison sulcus. In lower limbs, you can see different types of deformity, bowing of legs. If this bowing is like this, that means the knees are apart. This is labeled as genu varum. If the bowing is like this, that means the knees are touching each other as if they are glued through a gum. This is gyrus valgum. And if you see both the knees going in one direction, either left or right, this is windswept deformity. Windswept deformity. right? Windswept deformity. And this is the typical x-ray that you see in the exam. What all you can appreciate, can you notice at the ends there is cupping, like this cupping, then these ends are widened up, this is splaying, and then there is fraying. So these 3 points for your exam: cupping, splaying, and fraying. And obviously, because of the vitamin D deficiency, less calcium deposition, you can notice the bones don't look that opaque. The bones have decreased density. So this is yet another feature that you notice on the x-ray. Moving to the lab investigations, because of the vitamin D deficiency, calcium levels would be low, phosphate levels will be low. That will stimulate the parathyroid hormone. Parathyroid hormone will cause the release of the calcium from the bones, and finally this calcium levels can be normalized also, right? So these are the features that you will appreciate over vitamin D deficiency. If the examiner asks which is the most important parameter that tells you vitamin D deficiency, the answer there will be ALP, raised ALP. If there, this is vitamin D deficiency, right? Coming to the treatment, we have two protocols, high dose regimen and the low dose regimen. Which are you going to prefer? I'm going to prefer the low dose regimen. So what is the high dose regimen? In high dose regimen, you give 60,000 international units of vitamin D per week for 10 weeks, right? So total 6 lakh international units, 6 lakh international units. Then you have low dose regimen, where we give 6,000 international units per day for 3 months, Then after the treatment, we check the response to the treatment. How do you check the response? You look at the x-ray. If the child is responding, you can see a white line of calcification, So this is all about vitamin D deficiency. Now let's move to the next fat soluble vitamin, vitamin E. In vitamin E deficiency, you are going to see two problems: hematological problem, neurological problem. In hematological problem, we see hemolytic anemia, and in neurological problem, you see spinocerebellar ataxias. Then comes vitamin K deficiency. Vitamin K is very important for your clotting factor, so that deficiency can lead to bleeding, and in newborns it leads to hemorrhagic disease of newborn. That's why at the time of birth you give this vitamin K injection, right? So after this vitamin section, now let's move to the main core topic, that is malnutrition. So the term malnutrition actually, if you look in the literal sense, means inappropriate nutrition. That means ये ओवर न्यूट्रिशन भी हो सकता है, अंडर न्यूट्रिशन भी हो सकता है, बट अभी इंडिया में अंडर न्यूट्रिशन की ज़्यादा हमारी प्रॉब्लम है, तो इसलिए हम सिनोनिमसली यूज़ करते हैं अंडर न्यूट्रिशन को माल न्यूट्रिशन से. Now there are some definitions जो हमने पढ़ा था, नॉर्मल पैरामीटर्स. तो ग्रोथ चार्ट्स में अगर आप देखो तो वी हैव standard deviations. Anything beyond plus minus 2 standard deviation is considered abnormal. So anything beyond plus minus 3 standard deviation is going to be severely abnormal, right? So ये dictum आपको ध्यान रखना है. Anything beyond plus minus 2 is abnormal. Anything beyond plus minus 3 is severely abnormal. तो अगर मैं वेट बोलूँ, वेट फॉर हाइट, मतलब वेट बच्चे का एज कम्पेयर टू द हाइट इज लेस देन -2, दैट मीन्स लेस वेट है, द चाइल्ड इज अंडर वेट, देयर इज वेस्टिंग. सो द क्राइटेरिया, वेट फॉर हाइट लेस देन -2 बट मोर देन -3, मतलब -2 से -3 के बीच, दिस इज वेस्टिंग. बट इफ आई से वेट फॉर हाइट इज लेस देन -3, दिस इज गोइंग टू बी सिवियर वेस्टिंग. ऑन द अदर हैंड दूसरे स्पेक्ट्रम पे जाते हैं, अगर मैं बोलूँ वेट फॉर हाइट इज इन बिटवीन +2 to +3, दिस इज एक्चुअली द चाइल्ड इज ओवर वेट. बट if it is more than +3, this is obesity. Let's go toward height. Short stature याद है, So if the height for the age is -2 to -3, that means the child has stunting. If it is less than -3, it is going to be severe stunting, severe stunting. Right, so these are the important criteria that you should know for your exam. Now let's move to the next topic, that is acute versus chronic malnutrition. What is acute malnutrition? When you go to a new setting, कोई भी stressful situation आती है, stop eating food, the nutrition is not fine, what is going to happen? You are going to lose weight. Same thing happens with the child. So imagine a child जिसको डायरिया हो गया, लूज stools हो गए, and appetite बिल्कुल डाउन हो गई, वायरल फीवर हो गया. So obviously the child won't eat for odd 7 to 10 days, and because of that the child's weight will fall. This is acute malnutrition. Here the height won't be affected. 7 दिन में बच्चे की height पे कोई effect नहीं आएगा, weight definitely will drop. But if this undernutrition continues for a long period of time, there is chronic malnutrition. Now the height will also get affected, the overall growth will get affected. So the same thing differentiates acute versus chronic malnutrition. Acute malnutrition, obviously your weight for age will decrease, height for age will stay normal. So if you look at a weight for height, it will definitely be decreased. On the other hand, in chronic malnutrition, your weight for age has decreased, your height for age has also decreased. So the weight for height may be normal. So the examiner asks 3 things over here. What is the marker for acute malnutrition? So acute malnutrition में देखो, weight for age तो दोनों में decreased है, height for age तो normal होता है. So what is the best marker for acute malnutrition? It's weight for height. If I say what is the best marker for chronic malnutrition, it's height for age. If I say what is common to both, weight for age is common. So ये 3 पैरामीटर पूछता है एग्जामिनर पूछता है कि what is common. So what is common? Common is weight for age. What is the best marker for acute? It's weight for height. What is the best marker for chronic? It's height for age. Right? These 3 important pointers you should know for your exam. Okay. So with that, now let's move to the extreme form of malnutrition, that is kwashiorkor and marasmus. So अब 2 बच्चे देख रहे हैं यहाँ पे ना, baby A, baby B. Look at baby A. Baby A is just skin and bones baby, right? Fat लग ही नहीं रहा, just skin and bones है, nothing else, right? So this is marasmus child, marasmus child. मरा हुआ दिख रहा है ना, marasmus child. Then you have B child. Look at this child has swelled up. This is kwashiorkor child, which has the better prognosis, kwashiorkor or marasmus? Marasmus has better prognosis, kwashiorkor has poor prognosis. What has happened in both of these children? Both of these children have extreme form of malnutrition. But one child, marasmus child, has used all of the fat sources from his body, has good appetite. Look how alert this child is sitting. If I offer 2 chapati to this child, the child will demand for 4, has very good appetite. As soon as I give enough nutrition, this child will gain weight. This child adapts to the situation very well. On the other hand, look at the child B. This is kwashiorkor child. This child has lost his appetite, is lying down listlessly. If you offer food to this child has zero appetite, won't eat that food. That's why poor prognosis in kwashiorkor child, right? So let's enlist the differences between them. Firstly, in marasmus child, wasting is there. Yes, I can see the child is severely wasted, visible wasting is there, right? I can see the bones of this child. So you see visible severe wasting in marasmus. What about kwashiorkor? Can you see wasting over here? I cannot see wasting. There is swelling. The wasting is hidden, right? So wasting, though it is there, but it is masked by the edema. What about the general appearance? Here the marasmus child is alert. There is severe wasting, and we call this child as skin and bones baby. On the other hand, this kwashiorkor child looks very lethargic, dull, and we call this as fat sugar baby. Appetite of this marasmus child is very high, voracious appetite. So if a child of that particular age can eat 2 chapati, he will eat 4. On the other hand, here the appetite is lost, appetite has decreased. Age of onset of marasmus can happen anytime, but usually kwashiorkor happens after 1 year of age when the diet is switched to a very low protein diet. The prognosis here is good, prognosis here is poor. There are certain skin and hair changes that you appreciate. These are the additional features that you can see actually in both of these, but more commonly seen with kwashiorkor. So what are these changes? Skin and hair changes. You see flag sign in the hair. If you look at the hair, there will be areas of hypopigmentation, hyperpigmentation. This is known as flag sign. You see peeling of the skin. This is known as peeling of the skin, flaky paint appearance, flaky paint dermatopsis. And you see patching skin, that is known as greasy pigment appearance. In liver, when we talk about marasmus, you see shrunken liver. Here you see fatty liver. Fatty liver. So these are the two important extreme forms of malnutrition. Now let's move to an important segment for your exam, that is severe acute malnutrition. इसमें सबसे important चीज you should know the definition. How do you define a child as a SAM child, right? So remember, for defining or the criteria for SAM includes any one out of these. The first thing, weight for height less than -3 standard deviation, that means severe wasting. In the age group 6 months to 5 years of age, MUSE less than 11.5 centimeter. And lastly, nutritional edema. That means no other cause of edema to be there, nutritional edema. I need anyone to label this child as a SAM child, right? So this covers our nutrition section. Any doubt regarding this nutrition segment? If not, then we will move to our genetics part. Any doubt? If no doubt is no problem is there, then we can move towards genetics. Okay, let's start with genetics then. In genetics, we will start with Down syndrome, right? So what is Down syndrome? You all understand? Trisomy 21. We have one extra 21 chromosome. What are the features that you appreciate in a Down syndrome child? Look, I have shown the face of the typical Down faces. Can you notice a flat facial face, as if someone has flattened the face? There is a flat facial profile. Small low set ears. In eyes, you see epicanthal folds. Open mouth, protruding tongue. In hand, you can see this crease, single crease, that is known as simian crease. Which question was asked in last session? I think I missed it. Can you repeat that question again, Parth? Okay, so what is important for you in the exam regarding the Down syndrome are the associations of Down syndrome, right? Examiner is very fond of asking which things are more common in Down syndrome as compared to the general population. What are the cardiovascular problems that you see? What are the GI problems that you see? What are the hematological problems that you see? So one by one, let's discuss. If we talk about cardiovascular system, which congenital heart defect is associated with Down syndrome? Remember, the most common one is endocardial cushion defect. Endocardial cushion defects. पार्थ, आपका ये क्वेश्चन ना हम एकदम एंड में लेंगे, आप तो बहुत पीछे चले गए शॉर्ट स्टेज पे, है ना। तो अभी हम ये कंटिन्यू करते हैं। जेनेटिक्स का मोस्ट कॉमन एंडोकार्डियल क्वेश्चन डिफेक्ट कार्डियोवास्कुलर सिस्टम होता है। इन जीआई यू सी ऑब्स्ट्रक्शंस, ड्यूडेनल ऑब्स्ट्रक्शन, ड्यूडेनल एट्रियासिस, एनुलर पैनक्रियास। Hematological problem में they ask which is the most common hematological cancer or malignancy that you see. So remember, in Down syndrome, ALL is more common than AML. But in chota bachas, less than 3 years of age, AML is more common. And which AML? M7 type, that is megakaryocytic. Endocrine issues में they are more prone for congenital hypothyroidism. In skeleton issues, they have atlantoaxial instability, atlantoaxial instability or dislocations. That's why we go for their neck X-rays from time to time. And in neurological, remember, they have mental retardation or intellectual disability. Mental retardation is universal in the Down syndrome patients. So मतलब Down syndrome का बच्चा essentially हमेशा लो IQ होने वाला है. That's why it's very important that you antenatally screen for Down syndrome and let the parents decide whether they want to continue the pregnancy or not. Right? So antenatal screening की बात करते हैं. फर्स्ट ट्राइमेस्टर में हम करते हैं डुअल टेस्ट इन विच वी गो फॉर बीटा HCG एंड पैप ए राइट. ये तो हो गए ब्लड टेस्ट. व्हाट अबाउट अल्ट्रासाउंड? अल्ट्रासाउंड पे वी चेक फॉर न्यूक्ल थिकनेस और ट्रांसल्यूसेंसी, दैट इज NT-NB स्कैन. If the nuchal thickness is more than 3 mm, it is suggestive of Down syndrome. सेकेंड ट्राइमेस्टर में आता है ट्रिपल टेस्ट. इस ट्रिपल टेस्ट में हम करते हैं बीटा HCG, अनकॉन्जुगेटेड एस्ट्रायोल एंड अल्फा फीटोप्रोटीन. क्वाड्रपल टेस्ट जिसमें आपका ट्रिपल टेस्ट आएगा प्लस इनिबिन. एग्जामिनर आज के कौन सी चीज़ बढ़ती है इनमें? ट्रिपल टेस्ट, क्वाड्रपल टेस्ट, ये सब हम करा रहे हैं. कौन सा मार्कर बढ़ेगा, कौन सा मार्कर कम होगा? तो वेरी इजी टू रिमेंबर दो चीज़ें इंक्रीज होंगी. I for inhibin, I for increase, and H for HCG, H for high. Right? So ये याद रखना है, ये दोनों बढ़ेंगे, रेस्ट ऑल विल डिक्रीज. So बीटा HCG हाई आएगा, बाकी सब लो आएंगे. यहाँ पे भी बीटा HCG हाई आएगा, बाकी सब लो आएंगे. इनिबिन इंक्रीज आएगा. राइट? So this is all about Down syndrome. Why AML is more common in less than 3 years? ये तो डेटा है ना. अब छोटे बच्चों में हम ज़्यादा देख रहे हैं. यूजुअली ALL ज़्यादा है, लेकिन लेस देन 3 years में तो फैक्ट है. इसमें क्या एक्सप्लेनेशन देना है? इट्स अ फैक्ट दैट यू नीड टू रिमेंबर फॉर द एग्जाम. अब बोलोगे आप मुझे एंडोकार्डियल क्वेश्चन डिफेक्ट क्यों होता है डाउन में? अब दिख रहा है वही हम पढ़ रहे हैं. बहुत सारी चीज़ों के आंसर नहीं है. मेडिसिन तो पेशेंट टू पेशेंट हमने स्टडी की है, उससे एसोसिएशंस निकाली हैं. Right? So all this is about Down syndrome. Now let's move to yet another important two syndromes, that is Turner and Noonan syndrome. So what is Turner syndrome? Monosomy exo. Right? So 45 exo है, एक क्रोमोसोम हमारे पास कम है. Because Y क्रोमोसोम नहीं है, these are always and always going to be females. Usually they have normal IQ, but their gonads are not functional. They have streak ovaries. So amenorrhea होगा. On the other hand, Noonan syndrome, जिसको मेल टर्नर्स भी बोलते हैं, but मेल टर्नर्स is a गलत term. Why? Because this is not a chromosomal problem. This is a genetic problem. Autosomal dominant है, जिसमें PTPN11 जीन की प्रॉब्लम है. तो ऑटोसोमल इशू है इसमें. तो सेक्स क्रोमोसोम तो हमारे पास है ही नॉर्मल, तो एक्स भी हो सकता है और वाई भी हो सकता है. सो दिस कैन बी अ मेल ऑल्सो, दिस कैन बी अ फीमेल ऑल्सो. राइट? सो इसको मेल टर्नर जो नाम है वो गलत है नूनान सिंड्रोम का. IQ here is low. On the other hand, the gonads are functional या फिर थोड़ी सबफर्टिलिटी हो सकती है. Both of these have associated cardiac defects. टर्नर सिंड्रोम में रिमेंबर लेफ्ट साइडेड हार्ट डिफेक्ट्स होते हैं और नूनैन सिंड्रोम में राइट साइडेड हार्ट डिफेक्ट्स होते हैं. लेफ्ट साइड पे क्या है आपका? लेफ्ट एट्रियम, लेफ्ट वेंट्रिकल, एओर्टा, सो कोआक्टेशन ऑफ एओर्टा, बायकस्पिड एओर्टिक वाल्व, ये सब यहाँ दिखता है. राइट साइड पे क्या है? राइट एट्रियम, राइट वेंट्रिकल, पल्मोनरी आर्टरी, सो पल्मोनरी स्टेनोसिस दिखता है यहाँ पे. राइट, सो नाउ लेट्स रिवाइज इंपॉर्टेंट फीचर ऑफ टर्नर सिंड्रोम. ये फिनोटाइप है जो आपको दिखता है. शॉर्ट स्टेचर, लो हेयर लाइन, ब्रॉड चेस्ट, शील्ड लाइक चेस्ट भी उसको बोलते हैं, वाइड कैरिंग एंगल, स्ट्रीक ओवरीज, शॉर्ट मेटाकार्पल, ये सारे पॉइंट्स एग्जामिनर आपसे पूछ चुका है. ठीक है, सो वेरी इंपॉर्टेंट दैट यू गो थ्रू द फिनोटाइप ऑफ टर्नर सिंड्रोम. मूविंग टू नेक्स्ट जेनेटिक सिंड्रोम, दैट इज द डाइजॉर्ज सिंड्रोम. यहाँ पे थर्ड और फोर्थ फेरेंजियल आर्च का डिफेक्ट होता है. And what are the features that you see? You see catch 22. ये mnemonic है, catch 22. C is for cardiac problems. What are the most common cardiac issue that you see in Dijor's syndrome? The most common defect that you see over here is conotruncal defect. Conotruncal defect. Then we have A. A is for abnormal facies. Then you have T. T is thymic problem, thymic aplasia. Then you have C. C is for cleft lip, cleft palate. H is hypocalcemia. And T is the chromosome. यहाँ deletion होता है on the chromosome 2211 deletion. This is the micro deletion syndrome. So these are the features of Dijor's syndrome that you need to remember. ठीक है, so this is about genetics. Now let's finally move to your neonatology. Neonatology में 3 important conditions, respiratory distress की problems. एक प्रॉब्लम होती है प्री टर्म में, एक प्रॉब्लम होती है टर्म में, एक प्रॉब्लम होती है पोस्ट टर्म में, या जो इन्फेंट्स जो बच्चों में स्ट्रेसफुल इंट्रायूट्राइन कंडीशन है. सबसे पहले आते हैं प्री टर्म्स में. प्री टर्म बेबी समय से पहले पैदा हो गए, सो देयर लंग्स आर नॉट मैच्योर, दे हैव डेफिशिएंसी ऑफ द मैच्योर सर्फैक्टेंट. सर्फैक्टेंट क्या करता है? जब बच्चा ब्रीथ करता है तो एल्वुलाई शुड एक्सपैंड, एंड देन जब एक्सपायर करता है बच्चा तब एल्वुलाई शुड कॉन्ट्रैक्ट. To prevent the complete collapse of the alveoli, we have surfactant. The surfactant decreases the surface tension. But because premature baby इस mature surfactant के साथ नहीं पैदा हुआ, what will happen? Their alveoli will collapse. There will be no air in the lungs. No air in the lungs means when I look at the X-ray, I won't see black air. I will see complete white out. No air. This is white out lung. So this is a problem of preterms that is known as respiratory distress syndrome or highly membrane disease. This happens because of the deficiency of mature surfactant. Right, X-ray you will see white out lung or air bronchogram appearance. The treatment here is nothing but going for CPAP therapy or giving surfactant via Ensure technique. We can prevent this condition by giving antenatal steroids to the mother. Then comes the second condition. This you see in term babies, especially term male babies who are born via cesarean section. So normally what happens when the child is born through a vaginal canal, the baby is squeezed through that narrow vaginal canal and then comes out. So in utero, the baby's lungs are not functional. They are filled with fluid. So during this passage, what happens? Whatever fluid is there in the lungs is expressed through the mouth, nose, and squeezed out. But when the child is born through a cesarean section, the child is taken out through a cut in the uterus. So some amount of fluid might be retained in the lungs, and because of that fluid retention there is respiratory distress. Sooner or later this fluid gets resorbed and the child turns out to be fine. So this is a very transient condition, right? That's why the name transient tachypnea of the newborn. This happens because of the retention of lung fluid. And here on the x-ray you will again see this fluid. This fluid you will see around the hilum. This is known as perihilar infiltrates. You will see this fluid in the horizontal fissure, so fluid in the interlobar fissure. All of these things you are going to see. You will see this perihilar infiltrates, which is known as sunburst appearance, and you will see fluid in the interlobar fissure. The treatment here is nothing, just supportive treatment. Sooner or later, the child's fluid will be resorbed and the child will turn out to be fine. Lastly, you have meconium aspiration syndrome. In meconium aspiration syndrome, what happens? If the child is a post-term child, or the child has some stressful situation in utero, what will happen? The child will pass meconium. This meconium will be aspirated. This meconium can block my airway, can cause partial obstruction, can cause pneumonitis. So all of these things can happen, right? So one by one, let's look at the x-rays. Let's look at the first image. What can you see over here? Can you see that complete white out is there? White out lung is there. This is white out lung that you see in RDS. Then you see this situation. Can you see fluid in the interlobar fissure, perihilar infiltrate, sunburst appearance? This is nothing but transient tachypnea of newborn. And this third condition where you see this fluffy things, you see an area of collapse, hyperinflation. This is nothing but meconium aspiration syndrome. So what do you see in meconium aspiration syndrome? There can be collapse, जहाँ पे एयर पूरा ब्लॉक हो गया वहाँ कोलैप्स हो जाएगा. जहाँ पार्शियल ब्लॉक हुआ वहाँ हाइपर वेंटिलेटेड पिक्चर दिखेगी, हाइपर वेंटिलेशन दिखेगा. And there can be pneumonitis like picture. Pneumonitis like picture. Here also the treatment is supportive. Sooner or later this meconium would be cleared out. Right? So these are the conditions with respiratory distress in newborns. So जब भी distress में कोई बच्चा आता है हम उसको एक distress score देते हैं. This is known as Silverman-Anderson score. This you go for in preterms. 5 parameters हैं: upper chest retractions, lower chest retractions, xiphoid retractions, nasal flaring and grunting. जितना ज़्यादा score उतना ज़्यादा distress, right? So ये हम distress क्यों कर रहे हैं हम scoring? क्योंकि जब बच्चा मेरे पास आएगा मैं score लगाऊंगी. अगर score बोल रहा है बहुत severe distress है तो मैं उसको intubate करके ventilator पे रहूंगी. अगर score बोल रहा है बहुत कम distress है तो मैं observe कर सकती हूँ just oxygen पे. अगर moderate distress है I would need a CPAP machine. राइट. सो दिस स्कोर इज वेरी इंपॉर्टेंट. ऑन द अदर हैंड जब मुझे दिख रहा है कि डिस्ट्रेस अभी कम हो रहा है, इसका स्कोर कम हो रहा है, तो मैं इसको वेंटिलेटर से हटा के सीपैप पे रख सकती हूँ. सीपैप से फिर मैं रूम एयर पे शिफ्ट कर सकती हूँ डिपेंडिंग अपॉन द स्कोर. दैट्स वाई दिस डॉक्यूमेंटेशन ऑफ स्कोर इज इंपॉर्टेंट. ठीक है. तो 1 0 1 2 करके स्कोरिंग होती है. जितना ज़्यादा स्कोर मतलब ज़्यादा डिस्ट्रेस. वेरी इजी टू रिमेंबर. देखो लोअर चेस्ट, जिफॉइड और नेजल फ्लेयरिंग तो बहुत आसान है. अगर रिट्रैक्शंस नहीं है तो 0, माइल्ड है तो 1, सिवियर रिट्रैक्शंस है तो 2. राइट. सेम गोज फॉर नेजल फ्लेयरिंग. अगर कोई विजिबल नहीं है तो 0, माइल्ड है तो 1, सिवियर विजिबल है तो 2. फिर आते हैं ग्रंटिंग पे. ग्रंटिंग क्या होती है? ग्रंटिंग साउंड होती है एट द टाइम ऑफ एक्सपिरेशन आती है. दिस इज एक्सपिरेशन अगेंस्ट द पार्शली क्लोज्ड ग्लॉटिस. सो अगर कोई ग्रंटिंग साउंड नहीं है तो 0. अगर ग्रंटिंग साउंड आपने जैसे स्टेथोस्कोप लगाया आपको सुनाई दे रही है, वैसे नहीं सुनाई दे रही नेकेड एयर से, बट स्टेथोस्कोप लगा के चेस्ट पे सुनाई दे रही है, इट्स ऑडिबल ऑन स्टेथ, दिस इज 1. एंड व्हेन यू डोंट इवन नीड अ स्टेथ, मम्मी को भी सुनाई दे रही है, आपको भी सुनाई दे रही है, इट्स ऑडिबल बाय नेकेड ईयर, इट्स 2. राइट. सो ऑडिबल विथ नेकेड ईयर इज 2. फिर फाइनली आते हैं हम अपर चेस्ट रिट्रैक्शंस पे। नॉर्मली क्या होता है, अगर बच्चा ऐसे लाई डाउन है तो चेस्ट और एब्डोमेन की मूवमेंट एट द टाइम ऑफ ब्रीथिंग बहुत सिंक्रोनस होती है। देयर विल बी राइज ऑफ द चेस्ट, देयर विल बी राइज ऑफ द एब्डोमेन एंड सो ऑन, राइट। सो सिंक्रोनस ब्रीथिंग होती है। जब बच्चा डिस्ट्रेस में होगा, बच्चा तेज तेज सांस लेगा, सो चेस्ट की मूवमेंट डिसोसिएट हो जाएगी आपके एब्डोमेन मूवमेंट से। इनिशियली आपको नोटिस होगा कि इंस्पिरेशन पे एक लैग आ रहा है, उसके बाद कंप्लीटली अपोजिट ऐसी मूवमेंट जाएगी, दैट इज सी सॉ मूवमेंट ऑफ द चेस्ट, राइट। सो अगर सिंक्रोनस आपको दिख रहा है तो 0, अगर आपको इंस्पिरेटरी लैग दिख रहा है 1, अगर सी सॉ मूवमेंट दिख रही है चेस्ट की तो इट्स 2। ऐसे हम ग्रेड करते हैं प्री टर्म्स रेस्पिरेटरी डिस्ट्रेस को बाय द सिल्वरमैन एंडरसन स्कोर। अब टर्म बेबीज में वी हैव दिस डाउनी स्कोर। डाउनी स्कोर के जो भी 5 कंपोनेंट है, अगेन 0 1 2, रिमेंबर बाय दिस निमोनिक ग्रेस। G is for grunting, R is your respiratory rate, A is air entry, C is cyanosis, and R is retractions. सो ये ग्रंटिंग और रिट्रैक्शंस तो सेम है पीछे वाले स्कोर जैसा। अगर ग्रंटिंग नहीं है तो 0, अगर स्टेथ से ऑडिबल है तो 1, नेकेड ईयर से ऑडिबल है तो 2। सेम गोज फॉर रिट्रैक्शंस, नो रिट्रैक्शंस 0, माइल्ड 1, सिवियर 2। What about respiratory rate? Normal respiratory rate newborn का होता है 40 to 60. So if it is less than 60, 0, 60 to 80, 1, more than 80, obviously breaths per minute, then it's 2. फिर आते हैं एयर एंट्री पे। अगर एयर एंट्री आपको नॉर्मल सुनाई दे रही है 1, अगर एयर एंट्री डिक्रीज्ड है, नॉर्मल सुनाई दे रही है 0, डिक्रीज्ड है तो 1, अगर एब्सेंट है तो 2। फिर आता है साइनोसिस। अगर बच्चे को कोई साइनोसिस नहीं है, मतलब बच्चा पिंक है, तो 0। लेकिन अगर बच्चे को साइनोसिस है और जैसे ही आपने ऑक्सीजन दिया वो साइनोसिस चला गया, मतलब साइनोसिस सिर्फ रूम एयर पे है, तब 1। सो साइनोसिस ऑन रूम एयर इज 1। एंड लास्टली साइनोसिस है, आपने ऑक्सीजन दिया, 30% ऑक्सीजन भी दे दिया, अभी भी साइनोसिस नहीं जा रहा है, इट्स 2, राइट। सो इफ द साइनोसिस इज देयर on 30% oxygen as well, then it is 2, right. So this is about Downey score. With that, now let's move to an important topic, neonatal sepsis. So neonatal sepsis, what is the most important cause of neonatal sepsis? Most important cause, remember India में the most important cause is Klebsiella, whereas worldwide it's group B streptococcus. This neonatal sepsis is divided into early onset neonatal sepsis and late onset neonatal sepsis. अब इसको early neonate और late neonate से confuse नहीं करना है। Early onset means बहुत जल्दी हुआ है after the birth. Within 72 hours अगर बच्चा contract करता है, this is known as early onset sepsis. After that you label it as late onset sepsis. Right? SpO2 तो आपकी saturation है ना, जो आप चेक कर रहे हो, और FiO2 जो आप सप्लाई कर रहे हो। FiO2 इज इंस्पायर्ड ऑक्सीजन, जो मास से अगर मैं 30% दे रही हूँ तो दैट्स FiO2, जो मैं चेक कर रही हूँ बच्चे की, दैट इज SpO2, राइट। सो अर्ली ऑनसेट सेप्सिस कब होगा? बच्चे को इतनी जल्दी इन्फेक्शन, विद इन 72 आवर्स ऑफ इन्फेक्शन कहाँ से हो जाएगा? यूजुअली मदर से आएगा, है ना, मटर्नल कॉज होता है। तो क्या क्या रिस्क फैक्टर्स होंगे? अगर मदर को प्रोलॉन्ग्ड लेबर है मोर देन 24 आवर्स, if prolonged rupture of membrane है, there is spontaneous prematurity, right, chorioamnionitis, if there is birth asphyxia, बहुत सारे PV हुए हैं मदर के ना, per vaginal examination we say even a single unsterile or 3 or more sterile PVs is a risk factor. फिर आते हैं लेट ऑनसेट सेप्सिस। आफ्टर 72 आवर्स हो रहा तो यूजुअली कम्युनिटी और हॉस्पिटल एक्वायर्ड होगा। सो पुअर हाइजीन, पुअर कॉर्ड केयर, bottle feeding, all of these would be the risk factor for late onset sepsis, right? जब भी हम सस्पेक्ट कर रहे हैं सेप्सिस, वी गो फॉर दिस इन्वेस्टिगेशन. द गोल्ड स्टैंडर्ड ऑब्वियसली बिकॉज इन्फेक्शन है वुड बी ब्लड कल्चर, बट स्क्रीनिंग इन्वेस्टिगेशन विल बी सेप्सिस स्क्रीन. सो सेप्सिस स्क्रीन एग्जाम के लिए बहुत इंपॉर्टेंट है. तो जब ब्लड कल्चर तो टाइम लेगा आने में 3 से 4 दिन, उससे मीनवाइल हम एक सेप्सिस स्क्रीन करते हैं जिसके बेसिस पे हम अपनी ट्रीटमेंट डिसाइड करेंगे. 4 कंपोनेंट्स होते हैं, इसमें से अगर 2 पॉजिटिव आ रहे हैं, एबनॉर्मल वैल्यूज आ रही हैं, वी से दैट सेप्सिस स्क्रीन इज पॉजिटिव. तो क्या 4 कंपोनेंट्स हैं? सबसे पहले है आपका एब्सोल्यूट न्यूट्रोफिल काउंट. इफ इट इज लेस देन 1800, एबनॉर्मल. नॉर्मली हम में जब इन्फेक्शन होता है न्यूट्रोफिल्स बढ़ जाते हैं, बच्चों में कम हो जाते हैं. तो एब्सोल्यूट न्यूट्रोफिल काउंट लेस देन 1800. देन वी हैव इमेच्योर टू टोटल रेशियो of these neutrophils. If it is more than 0.2, again abnormal. Then comes CRP. If it is more than 1 nanogram per ml, abnormal. Then you have your micro ESR. If it is more than 15 mm per hour, this is abnormal. ठीक है. Treatment, you obviously are going to use antibiotics. We start with the empirical antibiotic ampicillin plus gentamicin. अगर feature of meningitis है तो you will go with 3rd generation cephalosporin cefotaxime. अभी जो ड्यूरेशन है डिपेंड करता है हमारी सेप्सिस स्क्रीन और ब्लड कल्चर पे. अगर सेप्सिस स्क्रीन पॉजिटिव है देन वी गिव इट फॉर 7 days. अगर ब्लड कल्चर पॉजिटिव है वी गिव इट फॉर 14 days. अगर meningitis है तो 21 days. So very easy to remember, 1 week, 2 week and 3 weeks, right? So this is about neonatal sepsis. A very important and severe form of neonatal sepsis is necrotizing enterocolitis. उसमें आपके एग्जाम पॉइंट ऑफ व्यू से इंपॉर्टेंट है बेल स्टेजिंग. So what is happening in necrotizing enterocolitis? देखो नाम में ही है, इन्फ्लेमेशन ऑफ द कोलॉन, एंट्रोकोलाइटिस. और उस इन्फ्लेमेशन की वजह से नेक्रोसिस हो रहा है, उस नेक्रोसिस की वजह से ही आगे प्रॉब्लम्स होंगी. तो इसकी स्टेजिंग जो है वो बहुत इंपॉर्टेंट है. बट हैपन्स इन स्टेज 1 में आपको सिर्फ क्लिनिकल पिक्चर्स दिखेंगी, इमेजिंग में कुछ नहीं दिखेगा. क्या इंपॉर्टेंट होगा क्लिनिकल फीचर में? बच्चा विल बी डल, लेथार्जिक, फीडिंग प्रॉब्लम्स आएंगी, वॉमिटिंग होगी, एब्डोमिनल डिस्टेंशन होगी. इसको डिवाइड करते हैं हम 2 स्टेजेस में, स्टेज 1A एंड 1B डिपेंडिंग अपॉन द क्लिनिकल फीचर. 1A में आपको दिखेगा ओकल्ट ब्लड एंड स्टूल. दैट मीन्स स्टूल में आपको वैसे ब्लड नहीं दिख रहा है लेकिन माइक्रोस्कोप के अंडर आपको आरबीसी दिख रहे हैं, ओकल्ट ब्लड है. देन will see gross blood in stool. matlab you don't need any microscope, mother can also see there is blood in the stool. So this gross blood in stool in stage 1B. Then comes stage 2. In stage 2 you see this important triad that is known as the metabolic triad. kya dikhta hai isme? We see metabolic acidosis, thrombocytopenia, that is decrease in the platelet count, and hyponatremia. This is metabolic triad. यहाँ पर हमको इमेजिंग फाइंडिंग्स दिखती हैं और डिपेंडिंग अपॉन दोज इमेजिंग फाइंडिंग्स वी हैव डिवाइडेड द स्टेज 2 into 2A and 2B. 2A में आपको दिखता है एयर इज ट्रैप्ड बिटवीन द इंटेस्टाइनल लाइनिंग. दिस इज नोन एज नीमोटोसिस इंटेस्टिनालिस. नीमोटोसिस इंटेस्टिनालिस. And in 2B you see this air gets trapped in between the portal vein. This is known as pneumatosis portalis. Pneumatosis portalis. Right? Then comes stage 3. Stage 1 को हमने डिवाइड किया था into 1A and 1B on the basis of clinical feature. 2 को किया था into 2A and 2B on the basis of imaging. In 3, stage 3, we have stage 3A, that is a clinical feature, and stage 3B which is a रेडियोलॉजिकल फीचर. 3A में आपको दिखेंगे फीचर्स ऑफ पेरिटोनाइटिस, जो हम पढ़ते हैं ना, रिबाउंड, टेंडरनेस, रिजिडिटी, वो सारी चीज़ें दिखेंगी. एंड स्टेज 3B में आपको दिखेगा एयर अंडर डायफ्राम, दैट इज नीमोपेरिटोनियम. नीमोपेरिटोनियम. Right? So these are the important points for your Bell staging. जैसे जैसे आप आगे जाते जाओगे ऑब्वियसली प्रीवियस स्टेज के भी फीचर्स आपको दिखेंगे बट ऐड ऑन होते जाएंगे, सिवियर होते जाएंगे. ओके, सो this is all about necrotizing enterocolitis. Now, one topic, if you have to read out of neonatology, I would say read this and go into your exam, because at least one question, even if they're asking just about your ambu bag, this is out from neonatal resuscitation. आपके MBBS time में भी in viva this had a separate table, and this is a very important topic indeed for your exam. तो basic concept हमको पता है. We will start with ventilation. The cornerstone of neonatal resuscitation is ventilation. तो आप ये पूरा फ्लो चार्ट ऑब्वियसली पूरा पढ़ चुके हो डिटेल्स में, हमारी पूरी डेडिकेटेड वीडियोस हैं, सेक्शंस हैं. यहाँ पर हम वन बाय वन लाइनर के थ्रू कवर करेंगे इस टॉपिक को. Right? So as I told you, what is the most important step in neonatal resuscitation? What is the most important step? It is ventilation. This is the cornerstone of NRP. देन वी हैव गोल्डन 1 मिनट. गोल्डन 1 मिनट क्या कहता है? अगर बच्चे को वेंटिलेशन रिक्वायर्ड है तो विद इन 1 मिनट ऑफ बर्थ स्टार्ट हो जानी चाहिए. दिस इज द गोल्डन 1 मिनट, दैट इज विद इन 60 सेकेंड्स और 1 मिनट देर शुड बी इनिशिएशन ऑफ दिस इफेक्टिव वेंटिलेशन. तो इनिशियल स्टेप्स आपको विद इन 60 सेकेंड्स कर लेने. इनिशियल स्टेप विद इन 60 सेकेंड्स कर लेने. व्हाट आर द इनिशियल स्टेप्स? वार्म द चाइल्ड, सक्शन, पोजिशन. तो पैड्स का निमोनिक है. पोजिशन एयरवे ड्राई स्टिमुलेट. राइट, पोजिशन एयरवे ड्राई स्टिमुलेट. क्या होती है पोजिशन हेड की? वी हैव द सेमी एक्सटेंडेड पोजिशन, दैट इज नोन एज द स्निफिंग पोजिशन. रिमेंबर, ट्रेकियल सक्शनिंग हम कभी नहीं करते. रूटीन ट्रेकियल सक्शनिंग इज नॉट रिक्वायर्ड. नो ट्रेकियल सक्शनिंग एंड नो routine suctioning even if meconium is present. नॉर्मल हार्ट रेट न्यूबॉर्न का कितना होता है? इट्स 100 to 160 beats per minute. And if you notice that the heart rate is less than 100 per minute, you start with this positive pressure ventilation. Once you start this positive pressure ventilation, within 60, after 60 seconds of, sorry, after 30 seconds of effective positive pressure ventilation and the heart rate is still less than 60, then you start chest compressions. The compression to ventilation ratio is 3:1. So you do 1, 2, 3 breath, 1, 2, 3 breath. And what is the site of this chest compression? Just below the nipples. Just below the nipples. Just below the nipples. Right? And you do this at the junction or the lower 1/3 of the sternum. Lower 1/3 of the sternum. And how deep? How deep it should go? It should be 1/3 of the AP diameter of the chest. AP diameter of the chest. What is the best indicator for the effective ventilation? Remember, increase in the heart rate is the best indicator. This is a very important MCQ that the examiner asks. What is the important indicator? बहुत सारे options होंगे. Change in the color, breathing rate. Remember, increase in the heart rate is the best indicator of effective ventilation. Right? Okay, so whenever you start with this positive pressure ventilation, if it is a term baby, you start with room air, that is 21% oxygen, room air. If it is a smaller child, you can start with 30%. So 21 to 30 is the range that is given. If the baby has labored breathing but heart rate is more than 100, so you don't give positive pressure ventilation, you shift the baby to CPAP. Now coming to this drugs that they ask. So after 60 seconds of chest compressions, if the heart rate is still less than 60, if the heart rate is still less than 60 after total 1 minute or 60 seconds of ventilation and chest compressions, you go and give epinephrine. What is the preferred route? Intravenous route is the preferred. We use this umbilical vein catheter and give epinephrine through that. What is the dose that we give? We use 1:10,000 dilution of this epinephrine, 0.2 ml per kg. If IV access is not there, you use intraosseous access or intratracheal access. If despite giving epinephrine the child is not responding, we suspect two conditions. Either the child is in shock or the child has pneumothorax. If there is pneumothorax, you are going to do thoracotomy. needle thoracotomy. If suspected shock is there, we use volume expander like normal saline. What is the indication for this volume expansion? It's suspected bleed. Suspected bleed, right? So this is all about your neonatal resuscitation. Some of the special situations that the examiner can give you is the meconium. This meconium concept is an older concept, but because this is given in your textbook and examiners have been asking time and again, I have mentioned this over here. So remember, whenever there is a meconium baby, that means the liquor is stained with meconium, we divide these babies into two categories: vigorous babies and non-vigorous babies. Vigorous babies means this baby is very active, the heart rate is above 100, and non-vigorous baby means the baby is not active, poor activity is there, and the heart rate is less than 100. So in both of these categories, you don't need to do anything different, right? If there is a non-vigorous baby, you start positive pressure ventilation and deal as per the protocol. Delayed cord clamping, remember, is not done if the baby requires immediate resuscitation. Usually, by the books, delayed cord clamping has to be done. We wait for 60 seconds before clamping the cord. This allows extra blood to reach to the baby, and this extra blood will help this baby in later life so that the baby cannot do not develop the chances of anemia, right? At that particular moment there is no benefit, but later on it is seen that wherever we do this delayed cord clamping, chances of anemia are less. So if the baby needs resuscitation, you don't do this delayed cord clamping, you immediately cut the cord, right? With that, we end the neonatology section. Now let's move towards the systemic pediatrics, and we will start with important infections. If there is any doubt regarding this neonatal resuscitation, please drop that down in the comment section and we will discuss that. Okay, so let's start with congenital infections then. So three important infections, three important triads. What are the three important infections? Toxoplasmosis, rubella, and CMV. So we have congenital toxo, congenital rubella syndrome, and congenital CMV. तीनों में triad है, right? कॉफी बीन साइन भाई आपको सिग्मोइड वॉल्वुलस में दिखता है ना? कॉफी बीन साइन ये तो सर्जरी में पढ़ते हैं ना? Okay, so congenital toxoplasmosis. What are the triad? Firstly, you see in eye, chorioretinitis, chorioretinitis. Then brain, diffuse calcifications. Then again brain, big head, hydrocephalus. So ये triad है ना, brain में दो चीजें हो गई, hydrocephalus, big head, calcification and eyes में chorioretinitis. This is toxoplasmosis. Then comes congenital rubella syndrome. यहाँ पे भी हमें triad दिखता है. What is the triad? Heart problem, PDA, sensory neural hearing loss, and eyes में cataract. Then you have congenital CMV. अब congenital CMV को ना toxos देखो. CMV का CMV है mnemonic. C is for calcification, C is for chorioamnionitis. M is for head का microcephaly. टॉक्सोप्लाज्मो में बड़ा हेड था, यहाँ पे छोटा हेड है, माइक्रोकेफली. एंड V is for calcification. वहाँ कैसी कैल्सिफिकेशन थी टॉक्सोप्लाज्मा में? डिफ्यूज कैल्सिफिकेशन या वी कैल्सिफिकेशन है, मतलब पेरीवेंट्रिकुलर कैल्सिफिकेशन, वेंट्रिकल्स के अराउंड कैल्सिफिकेशन, पेरीवेंट्रिकुलर कैल्सिफिकेशन, राइट? सो ये तीन इंपॉर्टेंट कॉन्जेनाइटल इन्फेक्शन है जिनके तीन ट्रायड है, ये बहुत अच्छे से आप याद करें. When we're talking about infections, how can we forget diarrhea? The most common cause of diarrhea in pediatric population is nothing but viral diarrhea, है ना? वायरल डायरिया ज़्यादा कॉमन होते हैं एज कम्पेयर टू बैक्टीरियल डायरियाज. तभी कोई भी आपके पास डायरिया के साथ बच्चा आएगा उसको एंटीबायोटिक नहीं देना है ना, बिकॉज मोस्ट कॉमनली इट्स वायरल डायरिया. बहुत स्पेसिफिक सिचुएशंस होती हैं जहाँ पे हम देते हैं एंटीबायोटिक. यूजुअली वी ओनली फोकस ऑन डिहाइड्रेशन ट्रीटमेंट एंड गिविंग जिंक. सो वायरल डायरियाज आर मोर कॉमन, और अगर बैक्टीरिया की बात करें तो बैक्टीरिया में ओवरऑल मोस्ट कॉमन इज इकोलाई। अगर आपको डायरिया के साथ साथ ब्लड दिख रहा है, मतलब डायरिया के साथ साथ ब्लड है, ब्लड एंड स्टूल्स है, सो वी कॉल दिस एज डाइसेंट्री, एंड डाइसेंट्री का मोस्ट कॉमन कॉज इज शिगेला, मतलब बैक्टीरियल है। ओवरऑल आई टोल्ड यू मोस्ट कॉमन कॉज इज वायरल। तो मैनेजमेंट में दो ही एस्पेक्ट होंगे, फर्स्ट इज द डिहाइड्रेशन करेक्शन एंड सेकेंड इज एडमिनिस्ट्रेशन ऑफ जिंक, एडमिनिस्ट्रेशन ऑफ जिंक। सो हम लर्न करते हैं अब कि डायरिया कैसे हम, मतलब डिहाइड्रेशन कैसे हम असेस करेंगे और ट्रीट करेंगे। सो वी हैव थ्री टाइप्स, थ्री डिग्रीज। नो डिहाइड्रेशन मतलब कोई डिहाइड्रेशन नहीं, सम डिहाइड्रेशन मतलब सम लेवल ऑफ डिहाइड्रेशन स्टार्ट हो गया, एंड सिवियर डिहाइड्रेशन मीन्स नाउ द चाइल्ड हैज सिवियर अमाउंट ऑफ डिहाइड्रेशन। सो नॉर्मल डिहाइड्रेशन का बच्चा, नो डिहाइड्रेशन का बच्चा कैसा होगा? द कॉन्शियसनेस विल बी नॉर्मल, thirst would be normal, matlab if you will offer water to the child, very gentleman like the child will drink normal thirst. If you do skin pinch, the skin is well hydrated, so it will go back, so there will be instant recoil. Eyes will look normal. Here I will give plan A. We will discuss what plan A is. Now moving to some dehydration. Now this child has some degree of dehydration, so this child will be very irritable, very irritable child. If you offer water to this child is very thirsty, very increased thirst, he will rapidly drink that water, very eagerly drink water. Skin pinch here is delayed because some degree of dehydration is there, but recoil would happen within 2 seconds. Eyes would look sunken over here. Oral mucosa will also be dry. Here I am going to give plan B. Then you have severe dehydration. Now the child has very high degree of dehydration. So the child will be comatose, lethargic, comatose. Obviously, if you offer water to this child, this comatose child won't drink water. So thirst would be decreased, or the child would be unable to drink. Here the skin pinch will be very delayed, more than 2 seconds. Eyes would be very sunken. Oral mucosa would be very dry. Here I am going to give plan C. Plan C. Right? So now let's see what are these plan A, plan B, and plan C. So plan A is for no dehydration. That means the child does not have any form of dehydration, just losses are happening from stools. So here the child can drink orally. So I am going to give ORS for this dehydration correction, oral rehydration solution. Here I just need to replace the losses. So how do you do replace the losses? Very simple rule, 10 ml per kg per loose stool. So you ask the mother, whenever the child is passing a stool, give him 10 ml per kg. So for example, if the child is 10 kg, I am going to give 100 ml, 10 into 10, right? So ask the mother, whenever the child is passing passing a stool, remember, give 100 ml of ORS, right? Moving to plan B. In plan B, the child has some degree of dehydration, but the child is very easily able to drink, eagerly drinking in fact. So here again, I am going to give oral ORS, oral rehydration solution. Here the rehydration I will do first because some degree of dehydration is there. So how do you do the dehydration correction? That is rehydration. We give 75 ml per kg over the next 4 hours, over 4 hours. So for example, if the child is 10 kgs, so I am going to tell the mother that give 75 into 10, that is 750 ml over the next 4 hours. This is for rehydrating the child. Some degree of dehydration is there, right? So this is for rehydration. Apart from that, whenever the child is passing stools, you have to replace that losses too. So replacement of losses is going to happen 10 ml per kg similarly, right? And while all this, needless to say, maintenance fluid should be given. You all know what are maintenance fluids, that you, me, everyone, we all need some amount of fluid to be daily consumed depending upon our weight. This is maintenance fluid, right? Moving to plan C. In plan C, the child is not able to drink, so here I am going to give IV fluids. The choice is Ringer lactate. Here the rehydration we do as 100 ml per kg. This we do in two parts. Initially 30 ml per kg bolus and then 70 ml per kg. If the child is less than 1 year, I am going to give this 30 ml per kg over 1 hour and rest over 5 hours. If it is an older child, I give it over 30 minutes and then over 2.5 hours. And needless to say, replacement of losses and maintenance fluid have to be given. Right? So sometimes maintenance fluid calculation is asked in the exam. How do you calculate maintenance fluids? We calculate by holiday cigar formula. So for the first 10 kgs, 100 ml per kg. For the next 10 kg, it's 50 ml per kg, and thereafter 20 ml per kg for every kg. Right? So if we have a 21 kg bacha, so let's calculate together. For the first 10 kg, it would be 100 into 10, that is 1000 ml. Then for the next 10 kg, it would be 50 into 10, that is 500 ml. And then for 1 kg, it's 20 ml. So total maintenance fluid of this child would be 1520 ml. So this is something that the child should take every day. ठीक है, ये अलग होगा ये तीनों सिचुएशंस में. नो डिहाइड्रेशन, सम डिहाइड्रेशन, सिवियर डिहाइड्रेशन, ये सबको चाहिए आपको, मुझको, सबको चाहिए अपने वेट के हिसाब से. दिस इज द मेंटेनेंस फ्लूइड रिक्वायरमेंट ऑफ एवरीवन, right? So this is about holiday cigar formula. Now that we have covered the gut infections, let's move up to pneumonias. Pneumonias में सबसे important है आपकी IMNCI guidelines. जैसे हमने डिहाइड्रेशन में पढ़ा, नो डिहाइड्रेशन, सम डिहाइड्रेशन, सिवियर डिहाइड्रेशन, जैसे निमोनिया में भी दिया हुआ है नो निमोनिया, निमोनिया, सिवियर निमोनिया. तो ये गाइडलाइंस में हम जाने से पहले दो इंपॉर्टेंट चीज़ें मैं आपको याद कराना चाहती हूँ. व्हाट इज़ फास्ट ब्रीथिंग एंड व्हाट आर द डेंजर साइंस. सो कब बोलेंगे कि बच्चा फास्ट ब्रीथ कर रहा है? सो डिपेंडिंग अपॉन द एज कट ऑफ्स है हमारे पास. नॉर्मली लेस देन 2 months the cutoff is 60. So if the breathing rate is more than 60 breaths per minute, this is fast breathing. 2 to 12 months more than 50, and after that more than 40. Right? फिर आते हैं danger signs. What are the danger signs? If there is a stridor in a calm child, this is a danger sign. If there is severe respiratory distress signs, that means retraction, cyanosis. If there is inability to drink, if the child is lethargic, if the saturation is less than 95, if the child has convulsions or seizures, this is all are the danger signs. So कभी भी कोई danger sign आपको दिखे, straight away this is severe pneumonia. हमें danger sign नहीं चाहिए. ठीक है, तो बहुत सिंपल सा कॉन्सेप्ट है यहाँ IMNCI गाइडलाइंस में. अगर बच्चे को फास्ट ब्रीथिंग नहीं है और कोई डेंजर साइन नहीं है, जस्ट कॉफ, कोल्ड, फीवर, ये सब है, सो दिस इज जस्ट कोल्ड, दिस इज नो निमोनिया. अगर फास्ट ब्रीथिंग है या रिट्रैक्शंस है, दिस इज निमोनिया, पर कोई डेंजर साइन नहीं होना चाहिए. जैसे ही डेंजर साइन आया, दिस इज सिवियर निमोनिया. सो नो निमोनिया मीन्स there is just cold, cough, no danger sign, no retractions, no fast breathing. यहाँ पे you just give supportive treatment, soothing agents. In pneumonia, you will see fast breathing, जो हमने अभी कट ऑफ पढ़ी थी, and or retractions. Here, pneumonia है बच्चे को, है ना, pneumonia है, that means infection is there. So here treatment will include giving antibiotics. But here, because severe pneumonia is not there, no danger sign is there, you can give oral amoxicillin and send the child home and ask to review. Mother को danger signs भी बताने. Severe pneumonia में you are going to have danger signs. Here, because the child has severe pneumonia, you have to give IV dose of antibiotic. We usually give ampicillin and gentamicin and refer this child to higher center for management. Right? So this is about IMNCI guidelines. आपको कोई केस सिनेरियो देंगे और उस पर फिर पूछेंगे कि नो निमोनिया है, निमोनिया है, सिवियर निमोनिया, और उसकी ट्रीटमेंट क्या हो. राइट? पिंक, येलो, ग्रीन. राइट? With respiratory problems, one topic needs thorough discussion, that is cystic fibrosis. वेरी फेवरेट ऑफ एग्जामिनर्स, आईएनआई में तो इम्पॉर्टेंट है ही, नीट पीजी में भी काफ़ी बार पूछा गया है, मेडिसिन में भी पढ़ाते हैं. सो फटाफट हम इंपॉर्टेंट पॉइंट्स यहाँ पे रिकैप करेंगे. सो यू ऑल नो दिस इज अ जेनेटिक प्रॉब्लम. कैसे इनहेरिटेंस है? ऑटोसोमल रिसेसिव इनहेरिटेंस है. सो यू विल सी इन कंजैंगुइनस मैरिजेस, you will see this a lot, right? Which is the gene involved, which is the chromosome involved? The gene we all know, this chloride channel. Chromosome is chromosome number 7, CFTR protein. And the most important mutation is deletion of phenylalanine on 508. बहुत सारी mutations recognize की हुई हैं. This is the most common mutation that you see. The common pathogen by age, initially छोटे बच्चों में और infancy में it's the staph aureus, and in adults what do you see? You see Pseudomonas is a common pathogen, right? बहुत सारे क्लिनिकल फीचर्स हैं. पैनक्रियाटिक इनसफिशिएंसी, मेकोनिया मिलियस, रेक्टस प्रोलैप्स, लिवर इन्वॉल्वमेंट, मेल इनफर्टिलिटी. डायग्नोसिस इज important for you. The gold standard here is the sweat chloride test, diagnostic test. Here you see if the chloride levels in the sweat is more than 60 millimole per liter on at least 2 or more separate occasions, this is diagnostic. उसके अलावा अगर जेनेटिक डिफेक्ट आपने ले लिया, डिटेक्ट कर लिया, दैट्स कन्फर्मेटरी. देयर इज नो परमानेंट क्योर, बस सपोर्टिव ट्रीटमेंट करते हैं टू थिन आउट द सिक्रीशंस, फिजियोथेरेपी एंड ऑल. फार्मा में आपने पढ़ा होगा, नाउ मॉड्यूलेटर्स आर अवेलेबल, कैफ्टर्स, ईवा कैफ्टर, ल्यूमा कैफ्टर, ये सब आपको फार्मा साइट पे बहुत अच्छे से बताया हुआ है, राइट? Now coming to the one-liners that the examiner is going to drop in cases of cystic fibrosis. They ask what is the earliest manifestation. Earliest manifestation neonatal period में आपको दिखेगी meconium ileus. Thick meconium है, stuck कर गया meconium ileus. Earliest pulmonary pathogen staph. Most characteristic Pseudomonas. Most fatal Burkholderia Cabacea, right? So important one-liners regarding this. Now moving to serious, एक्सरेज पूछते हैं और डिफरेंट अपीयरेंस पूछते हैं. दो टेबल्स हैं जो बहुत इंपॉर्टेंट है. हमने पढ़ा था कि जेनेटिक सिंड्रोम्स में देयर आर सम एसोसिएशंस. वी हैव रेड अबाउट डाउन सिंड्रोम, टर्नर सिंड्रोम, नूनान सिंड्रोम. So let's revise that again. I told you Down syndrome, what is the most commonly associated cardiovascular problem? It is nothing but endocardial cushion defects. So endocardial cushion defect is the most common. Endocardial cushion defect. Then you have Turner syndrome. I told you left-sided heart defects. Left side पे आपका क्या है? Left atrium, left ventricle, aorta, bicuspid aorta wall. You have coarctation of aorta. Noonan syndrome में you see right-sided problem, pulmonary stenosis. Then you have Edward and Patau. What is Edward? Trisomy 18. Patau is Edward. Patau is trisomy 13. In both of these, the commonly associated congenital heart defect is VST. In DiGeorge, we discussed the most common is conotruncal defects. In Williams syndrome, there is supravalvular aortic stenosis, supravalvular AS, right? Then we have certain maternal conditions जहाँ पे आपको दिखता है congenital heart defect. एक हमने पढ़ा, remember, rubella, PDA, congenital rubella syndrome में था. वैसे ही gestational diabetes अगर mother को है, there are chances that the baby can have congenital heart defect. Which one? Most commonly आपको दिखता है VST. If they ask most specific, that's transposition of great arteries. In rubella you see PDA. If mother is alcoholic, the baby will have fetal alcohol syndrome. If the mother continues to take phenytoin throughout pregnancy, baby will have fetal hydantoin syndrome. In both of these conditions, the most commonly associated congenital heart defect is VST. If the mother has bipolar disorder and is taking lithium, baby can have Epstein anomaly. If the mother has SLE and anti-Ro, anti-La antibodies, baby will have heart blocks. neonatal heart blocks. Neonatal heart blocks. Right? Now let's see different x-rays of different congenital heart defects. Firstly, let's look at this. What can you see? Snowman appearance, also known as figure of 8 appearance. Where do you see this figure of 8 or snowman appearance? Anyone? Snowman appearance or figure of 8 appearance? Yes, you see this in supracardiac TAPVC. Supracardiac TAPVC. Then we have this famous boot-shaped heart. Boot-shaped heart. This boot-shaped heart, where do you see? We see this in TOF, tetralogy of Falley. Tetralogy of Falley. Right? Then you have this box-shaped heart. This box-shaped heart you see in Epstein anomaly. Then you have the string, egg on string appearance. Egg on string appearance. Where do you see this egg on string appearance? Very classical egg on string appearance. You see this in most specific CHD that you see in gestational diabetic infant or diabetic mothers. That is TGA, transposition of great arteries. Right? So with that, we finished the cardio and moving on to the last topic of discussion, that is pediatric epilepsy syndrome. आपका भी recent UPSC CMS एग्जाम में भी पूछा गया, INI में भी पूछा गया, अभी NEET PG में भी पूछा जाएगा जरूरी है क्वेश्चन, राइट? My prediction from this session कि ये पूछा जाएगा in some form, right? So let's discuss पीडियाट्रिक एपिलेप्सी सिंड्रोम वन बाय वन. तो जब भी एपिलेप्सी सिंड्रोम पढ़ते हो ना आप, ये 3 चीज़ें आपको पढ़नी है. एज ग्रुप, कैसा सीजर दिखता है, दैट इज द सेमियोलॉजी ऑफ द सीजर एंड द टिपिकल ईजी फाइंडिंग इफ एनी, एंड लास्टली द ट्रीटमेंट. सो स्टार्ट करते हैं वेस्ट सिंड्रोम से. वेस्ट सिंड्रोम यू सी इन इन्फेंट्स, दैट मीन्स लेस देन 1 ईयर. क्या दिखता है यहाँ पे? आपको टिपिकल ट्रायड दिखता है. ट्रायड में क्या होगा? सबसे पहले सीजर फाइंडिंग. कौन सा सीजर है? इन्फेंटाइल स्पैसम. इन्फेंटाइल स्पैसम है. ठीक है. और क्या दिखता है? सेकंड, डेवलपमेंटल फाइंडिंग. देयर इज डेवलपमेंटल रिग्रेशन, मतलब लॉस ऑफ प्रीवियसली एक्वायर्ड माइलस्टोन. तो बच्चा अगर बैठना चलना सीख गया था, खड़ा होना सीख गया था, अब नहीं बैठेगा, देयर इज रिग्रेशन. एंड लास्टली देयर इज एन ईईजी फाइंडिंग. व्हाट इज दैट ईईजी फाइंडिंग? ईईजी फाइंडिंग बहुत क्योटिक हाई वोल्टेज हमें चेंजेस दिखता है. जैसे किसी ने बच्चे ने स्क्रिबल कर दिया मेरी ईईजी की शीट पे. दिस इज नोन एज हिप्सरिथमिया. हिप्सरिथमिया. Right? So these are the 3 features of West syndrome that you need to know. What is the treatment over here? Remember, drug of choice is ACTH or steroids. अगर 1 choose करना हो आपको, choose steroids. Right? And if you see this West syndrome associated with tuberous sclerosis, then the drug of choice here is Vigabatrin. Vigabatrin. Right? Vigabatrin. Then we have Dravet syndrome. This you see after 1 year of age. After 1 year of age, usually 1 to 2 years of age. Dravet syndrome में क्या होता है? इनिशियली बच्चे को होगा फेब्राइल सीजर, फीवर के साथ सीजर. उसके बाद लो फीवर के साथ भी सीजर आने लगेंगे. फिर ए फेब्राइल सीजर आने लगेंगे, मतलब विदाउट फीवर सीजर आने लगेंगे. एंड फिर फाइनली after the age of 1 year, multiple different types of seizures honge that are difficult to control. Right? Here the drug of choice is valproate. The pointer for your exam is we have a new drug now for Dravet syndrome, that is teripental. Teripental. Right? After that, now let's move to Lennox-Gastaut syndrome. ये दिखता है after 2 years of age. स्पेक्ट्रम है, यहाँ पे भी आपको वैसा ट्रायड दिखेगा. फर्स्टली सीजर, multiple different types of seizures, atonic seizures, absent seizures, बहुत different types से seizures होंगे. Developmental delay, because इतने सारे seizure हो रहे हैं, difficult to control seizures है. And lastly, typical EEG finding. क्या EEG finding है? Slow spike and wave complex दिखता है. What is spike and wave? ऐसे spike wave, spike wave, spike wave. तो यहाँ पे usually 2 to 3 hertz spike and wave complex दिखता है. हेयर ट्रीटमेंट इज नथिंग बट एंटी एपिलेप्टिक्स. मल्टीपल चाहिए होती है यहाँ पे, तो वैल्प्रोएट ड्रग ऑफ चॉइस. फिर आते हैं जैन syndrome, that is juvenile myoclonic epilepsy. This you see in adolescent age group, adolescent girls specifically 12 to 18 years. नाम में ये myoclonic epilepsy, so यहाँ you see myoclonic jerks, and these myoclonic jerks मतलब shock like contractions. These are precipitated by sleep deprivation and alcohol consumption. यहाँ पे EEG में आपको दिखता है 4 to 6 hertz spike and wave complex. Drug of choice here again valproate, but obviously if this female wants to plan pregnancy, then you shift to a safer drug, because valproate is teratogenic. You can shift to levetiracetam, right? But if she is pregnant and she comes to you that I am pregnant, you don't change the anti-seizure medication now, right? Because I don't want a pregnant female to throw a seizure. So if she is planning and she comes to you beforehand, we can try and switch to a safer drug. Otherwise, you continue on valproate and monitor, do scans regularly. Lastly, you have absence seizures. This you see in school-going age group, right? After 6 years. So school-going child, 4 to 10 years, they say, right? What do you see over here? There are brief episodes of vacant staring look. So there are complaints of daydreaming, decline in the scholastic performance. So very brief episodes of vacant staring look, right? So here the typical EEG is 3 hertz spike and wave, and drug of choice here is ethosuximide. Remember, if ethosuximide is not in the option, choose valproate, right? So this is about the important pediatric epilepsy syndromes, and with that we come to the end of this section. If there is any doubt or anything you want me to repeat, please drop that down in the comment section. I'll be more than happy to cover up. If not, let's have our dinners finally and have a good night's sleep. Any doubt, please? ये रिवर्सल ऑफ शंट जो टॉपिक है, ये आप जो मेन वीडियोस हैं उसको जाओ, क्योंकि पूरा समझाना पड़ेगा। साइनोटिक हार्ट डिसीज़, ए साइनोटिक हार्ट डिसीज़, पीडीए में क्या होता है, तो ये सारा अभी लास्ट मोमेंट आपका ये कॉन्सेप्ट क्लियर करने का तो नहीं है ना, तो आपको वो एकदम डिटेल में आपको समझाना पड़ेगा। अभी यूट्यूब चैनल पे भी पूरा सीवीएस सेक्शन जो है वो फ्री पड़ा हुआ है, आप प्लीज़ वो जाके रेफ़र करो, आपको रिवर्सल ऑफ शंट समझना है तो। ग्रोथ सेक्शन में आपका डाउट था एक, शॉर्ट स्टेचर में क्या पूछा गया था कि हाँ, ये रिलेशंस लाइफ लॉन्ग वैलिड रहेगा या अडल्टहुड में भी बोन एज क्रोनोलॉजिकल एज बिकम्स सेम इन द क्रोनोलॉजिकल डिले चाइल्ड। तो क्रोनोलॉजिकल डिले क्या होता है? मेरे हिसाब से आप ये पूछना चाह रहे हो कि अडल्टहुड में भी ये रिलेशनशिप रहती है या नहीं रहती। ऑब्वियसली नॉट यार, ऑब्वियसली नॉट। एक पॉइंट पे तो सारा बोन एज आ ही जाएगा ना, नॉर्मल हो जाएगा वो तो अभी आपके पास बच्चा आया है, इवेंचुअली बोन्स आर गोइंग टू फ्यूज, वो प्यूबर्टी तो आ ही जाएगी कॉन्स्टिट्यूशनल डिले में, है ना। अडल्टहुड में नहीं रहेगा ये सेम रिलेशन, ऑब्वियसली बोन फ्यूज हो जाएंगी, ठीक है। सो आई थिंक ऑल ऑफ द डाउट्स आर डन। ऑल द बेस्ट फॉर योर एग्जाम, प्लीज़ रिवाइज स्मार्टली इन दिस लास्ट लेग ना। बहुत कॉम्प्लिकेटेड कॉन्सेप्ट्स में नहीं घुसना है, टू द पॉइंट रहना है। प्रीवियस ईयर क्वेश्चंस, प्रीवियस ईयर टॉपिक्स अच्छे से रिवाइज करने हैं। कोई जीटी देने की अभी गलती मत करना, बिकॉज बिल्कुल वेस्ट ऑफ टाइम होगा जीटी देना। प्लीज़ गो थ्रू द प्रीवियस ईयर क्वेश्चंस, टॉपिक्स अपने ही नोट्स बार बार रिवाइज करने हैं, वही आपके काम में आएंगे। ओके, थैंक यू, हैव अ नाइस डे, विल मीट सम